we start with this gene, MIFR1, which has in N-terminus a signal peptide, which we removed it and replaced it with a 6-histidine tag to facilitate the purification. This modified gene was cloned into a plasmid vector. The protein was expressed in Escherichia coli cells, and the protein was purified by 3 chromatography methods. Here we can see a size exclusion chromatograph. This curve represents our protein. And on SDS page we can see a single band of our protein. So our protein now is very pure, and we decided to crystallize our protein. We tried 5 commercial kits in 2 different temperatures, using protein concentration from 5 to 25 milligram per ml, but no crystals were observed under these conditions. But we want to characterize this protein, and our protein is very pure, so we proceed in circular dichroism spectroscopy with thermal shift assay. Circular dichroism provides information about the secondary structure of the proteins. Here we see on the black curve our protein at 20 degrees Celsius before thermal shift assay, and these 2 minima indicated that our protein consists of alpha helices. We heat our protein and from 20 to 105 degrees, and here we can see that progressively our protein loses its alpha helical content. So there is a thermal unfolding. But when we cooled our protein from 105 to 20 degrees, here with the Kian curve, we saw that this flow closely overlapped with the initial at 20 before the thermal, and we found out that our protein refolds reversibly. Here is another plot from circular dichroism at 222 nanometer at 20 degrees before the thermal, and we see that the melting temperature is very high, it's at 90 degrees Celsius. So we have not crystallized yet our protein, and we decided to proceed in silico studies. We used Swiss model. We insert our protein, the sequence of our of MIFR1, and we used as template model the crystal structure of another FAR protein, NAFR1. And we took this predicted structure for our protein. We superimposed these 2 structures and we transfer remotely this palmitate to this position here with the same coordinators in the predicted protein. But why did we do that? We observed that here in crystal structure the palmitate has a polar group that is stabilized in binding site by lysine 96, which has a positive charge. And we wanted to see if this happened in our protein. Here is our protein, the palmitate with the same polar group, and arginine 97. And the palmitate is also stabilized with the positive charge of arginine. And we wondered if this happens and in other FAR proteins from other nematodes. And we proceed in multiple sequence alignment of various fat proteins. And we saw that these basic residues, adenine and lysine, are conserved at this position. So it may be possible to this positive charge of these basic residues may be very important for the stabilization of fatty acids in binding site. So, we have learned many things about this protein, but we want more. So, we're gonna increase the protein concentration in order to find which is the 3D conformation of our protein with crystallization. We're gonna extract thermodynamic data from circular dichroism and Van Comp equation. We want to find a suitable model in order to do ligand docking and molecular dynamics simulations. So we want to do that with Swiss model, but with AlphaFold and Modelite in order to select the correct one. So at the end, I would like to thank my supervisor, Kostas Bethanis, for his help and his support. Dr. Petros Giastas and Dr. Zouridakis for teaching me all the techniques in molecular biology and biochemistry, and especially Dr. Zouridakis for welcoming me in his group in Hellenic Paris Pasteur Institute. Dr. Karkouzas and Dr. Christoforidis for their help in computational approaches, and Dr. Panagiotopoulou for his help and guidance in circular dichroism. And of course, all of you for your attention. Thank you.
Okay, thank you, Maria. Any questions?
Do you have the possibility to try in vivo if by mutating the site of the protein you identify, if it can reduce the effect on the expression of the defense gene of the plant?
It's very early to do some in vivo experiments. We want to determine its role in vitro, and then we will try it in nematology.
Okay, thank you.
Okay, anybody else?
When you say it's quite hard to do some in vivo like tasting, is it because the purification of the protein when you mutate these amino acids implicated into ligand interactions like are not viable, or is it just the are you trying like into the nematode? Like the nematode mutation of this protein is like is essential, and so the nematode is not viable, and like the infection model is not.
This step, we will do that. We think that we can do a mutation in order to characterize somehow this protein.
Thank you for the interesting presentation. Two questions. One is, well, I think it's unusual for a protein to heat up to 105 degrees and then when you cool it down, reform completely, especially on alpha helix. So is that a characteristic that is observed in that family, or is it sort of a particular characteristic of this protein? And the other question is, what's your overall aim? What would you use that protein after you crystallize it? And, what do you see long term?
Okay, for the first question, we have information about a far protein from C. elegans that has also melting temperature at 90 degrees Celsius. But it's only this. For the second question, our aim is to find blockers, ligands, that block nematode, this protein, so the nematode, because the nematode has not the ability to produce fatty acids for its membranes. So we block that protein, which binds fatty acids, and we block the survival of nematodes. So this is our big aim.
Yeah. Thank you.
Okay. Right, Maria, a very quick question before we move on. Great presentation. I think you have your hands full for the rest of your studies. Just very quickly, I mean, you produce the protein in E. coli, and excuse me, I don't know a lot about plants and parasitism, but does glycosylation potentially come into play? Is that something you have to consider about its normal function, or do you know anything about it, or is it something you look into?
Yes, in EXPASY, I think it's EXPASY program, I'm not sure, we saw that there is a glycosylation site. So we want to express our protein in Pichia pastoris on another yeast cell in order to see this very important for the function of this protein.
Yeah. Okay, great. Thank you very much.
Thank you.
All right. Now let's move on to Lorenzo Panellati, who also comes from the Pasteur Institute in Paris. Lorenzo is a research engineer in the Neural Circuit Dynamics and Decision-Making Unit. And he will talk to us about exploring the dynamics of interhemispheric integration in the barrel cortex with calcium imaging and expansion microscopy. Thank you.
Thank you very much for the introduction, and hi everybody. Thanks for being here. So I'm Lorenzo. As you heard before, I'm a research engineer at Pasteur Institute, and today I'm going to talk about the dynamics of interhemispheric neurons at the level of the barrel cortex with a study using calcium live imaging and expansion microscopy. So in our lab we focus mostly on the rotor facial hair called vibriscus system. We exploit this system because there is a clear one-to-one somatotopic representation, which is a pattern well represented across several nuclei at the level of the brain, which is a clear landmark for studying neuronal integration. Because of that, there are like more than 50 years of research about the matter. So even though there are like already a lot of studies about that, there are still some limited understanding on how transcallosal activity contribute to tactile processing. So here is a rapid representation how the information reaches the cortex. So from the vibrissa at the level of the snout, there are like two main pathways. The lemniscal pathway, which reaches the somatotopical, the primary somatosensory area, and it reaches the columns that you can see here. So each column is representing a single whisker at the level of the snout of the mice. And the paralemniscal pathway, mostly innervating the layer 1, layer 5, and also the infrabarrel space of layer 4. When looking at the interhemispheric projection, a nice paper has been recently published, and it addresses some of our questions. So in particular, there are 3 different layers, layer 2, 3, 4, and 5, that are projecting on the contralateral side. But despite that, this paper doesn't show in which layer they are projecting to. So in our lab, we question ourselves how long-range interhemispheric projection contribute to the sensory processing. To answer that, we will first investigate the anatomical projection, and to do that, I will try to convince you that we need a new technique of histological analysis And then we will see also the functional identity of those neurons. So the experimental setup consists in a double transduce of the neurons. In particular, we will inject on the left side a retropropagating virus that will encode for Cray recombinase. And on the contralateral side, we will inject a second virus encoding for GCAM, a calcium sensor. So only the double transduced neuron will then provide the expression of the calcium sensor. After 1 month, that is the time required for the protein expression, we will perform chronic imaging, calcium live imaging, and then we will collect the brain and perform some histological analysis, in particular using iDisco technique and expansion microscopy. After which we will acquire the data and then we will trace the neurons. So I will start now with the morphological characterization of this ensemble of neurons. From our preliminary data, we identified that the system was working and we were having a good expression of neurons in layer 4 of the cortex, and the same results were also showed at the level of an iDisco brain. Furthermore, there are also double transduced neurons in layer 2-3 and in layer 5B and 6. So now I will focus on layer 4 because it's the one where there are the majority of the cell. So perform projection of the layer 4, and as you can see here, there is the somatotopical representation of the whiskers primary somatosensory, where each structure here corresponds to a vibrissa, and when looking at the specific signal coming from the double transduced neurons, there is a negative pattern compared to the autofluorescence. And when looking at the colocalization, we can see that none is happening, suggesting that our neurons, double transduced neurons, lays in the infrabarrel space, which is mostly, we suppose, is part of the paralemniscal pathway. When we start, we then try to trace the neurons and segment them, but despite the fact that we were able to identify some somas and to trace some dendrites, none of the axons projecting on the contralateral side were possible to be observed, and that's mostly due to some intrinsic limitation of the light sheet microscope, because it's the axon diameter is going even below 700 nanometers. So for that reason, we decided to change to a different type of technique, an histological technique, which is expansion microscopy. Compared to iDisco, indeed, we will completely physically expand the sample, and we will surpass the diffraction limit of the traditional light microscopes. Since the sample is bigger, we also decided to have a bigger microscope. Otherwise, it would take a lot of time to acquire the entire data frame. So for that, we will rely on a camera with a larger field of view, with a bigger sensor, and a detection objective with larger working distance and larger field of view. All of that is to minimize the need for tiling. This has the work being done by Bahar, which is the postdoc of my team, and she presented a nice poster yesterday. Now I will show you the comparison between a commercial light sheet and our setup. In particular, to acquire an entire brain of an iDisco brain, we will need 6 different volumes that then needs to be stitched together, and this is a process that is power consuming and also is error prone. Whereas for the expansion, for the XSP microscopy, the custom built in our lab, an entire mouse brain can fit in a single field of view. That's not only with the expansion microscopy, we also need the tiling system, but that will increase the resolution. But on the other side, we also have to deal with large data sets. Going back to the morphological analysis, we saw that there are neurons located in the infrabarrel space, but mostly in layer 2-3, which is the is the canonical source of callosal projection. For that reason, we will focus for the functional analysis on this cluster. As you can see here, this is a wide field of view of a bright field microscope, and on the right there is what we observe with a calcium with the two-photon microscopy. Thanks to a software called C2P, we were able to identify like some ROIs and then to extract the fluorescence, which is in this case is the spontaneous neuronal activity. So as we want to understand like the integration process, we will provide like we are providing some stimulation at the level of the snout of the animal, at the level of the vibrissa, and then we will record the activity in the exact same position at the level of the barrel cortex. In particular, there are like the stimulus is presented on the contralateral side only, ipsilateral, and the bilateral stimulation. This slide is just to highlight the fact that there are neurons that are they are responding all the time that the stimulation is provided, and others that are not. And some increase the fluorescence with the contralateral, and others decrease the fluorescence. So we have to deal with a heterogeneous responses. And those neurons that respond to the ipsilateral stimulation, a latency has been observed of approximately 100 to 100 milliseconds, meaning that the signal is coming back and forward from the contralateral side. And when we provide a bilateral stimulation of both contra and ipsilateral, here in green, synergistic integration happens because there is a huge increase in the fluorescence. So we decided then to try to differentiate and identify like some different roles in the neurons, in our population of neurons, and there are like just few of them that are silent, and most of them are acting in a different way. In particular, there are some that are modulated by the contralateral stimulation, and others that are inhibited by the contralateral, suggesting that there is a heterogeneous response. And for that reason, we suppose they are mostly they are important for the processing of the stimulation and like the sensation of the stimulus. For that reason, we decided to move to the behavioral analysis. This is a work that is still ongoing, so I don't have data to show, but we rely on a two-force alternative choice task, where we provide a different stimulation on one side and a distractor on the opposite side, and then the mouse needs to understand on which port he has to lick. So to conclude, I presented some technical innovation, in particular the expansion microscopy and this custom-made setup that is the only second one in the entire world. From the morphological analysis, we identify 3 different clusters at the level of the whisker that are projecting on the contralateral side, and this population of neurons shows heterogeneous responses. From the behavioral analysis, I can tell you that the mouse learned the task, and we are now dealing with the next step. So next steps, it would be like to acquire the first expanded brain and to register to the common data frame of Allen Brain Institute. And after that, we will link the functional identity to the morphological. And finally, I want to thank you for the attention and all my team for the work we have done together. And with that, I'm doing.
Thank you very much, Lorenzo. Any questions? No? Okay, a very quick question for my general question. So you describe methods to study signal integration. Okay, are there any conditions, except trauma, I guess, where there might be the communication between the two hemispheres is impaired, or that becomes relevant?
So in particular, it's known that for autism, for example, there is an impairment of the transcallosal activity, and for that reason, it's important to study it. But we focus mostly on the interhemispheric connection because, for autism, but also because we are a lab working on fundamental biology, so we really want to understand what is happening at the level of the neurons and how the signal is integrated.
Thank you very much. Anyone else?
Thank you. Maybe it's a stupid question, but when you do the behavioral test, you have the mouth constrained, right? To then stimulate the whiskers. Do you think that constraint should have some impact on what you see in the connection?
So for sure, we know that the level of the of the primary somatosensory area, a lot of integration happens. So that's true, but since the mouse is restrained, is also a way to deal with interference coming from other type of stimuli. So since he cannot move, there should be a minimal integration coming from the motor sensory area. And that's also the reason why when we perform recordings, we perform recordings also of the movement of the animal, and then we extract this movement with the Deep Black Cat, which is a software Python written code that allows to track the animal movement. And we will exclude those trials where the animal was moving too much, because you can't really understand if the activity of the neurons is related with the motor activity or with the stimulus you provide. So to answer your question, yes, for sure there will be interference coming from outside, and also because of our setup. We are trying to simplificate as maximum as possible the stimulation, because we are just interested in the integration of the signal we provide.
Thank you. Hi, thank you for the talk. I'm not into neuroscience, but I was wondering, you registered different pattern in neuron stimulation. So have you ever seen a specialization of this pattern? One neuron we're gonna respond to the stimuli in a specific way, and the other surrounding neurons are going to respond differently?
So in this case, it's not from what we observe, since these are really sparse, our neurons are really sparse, so there are not clustering all together. We can't really understand if there is it's not we can't really understand. We haven't observed that there is a specific pattern that is repeated across different areas. What we observe is the neurons is mostly responding the same way at the stimulation. To make an example, the neurons here is always firing at when the contralateral stimulation is provided, and across all the session he will respond in that way. Another neurons is consistent, and it will decrease the fluorescence, suggesting that they are involved with a specific role, but they are doing different, they are involved in different integration processes.
Okay, I think we should move on. Thank you, Lorenzo, again. And I'll pass you over to Mario for the final 3 talks.
Okay, the next speaker is Evgenia Politi. Evgenia is a technologist from the Agricultural University of Athens, and she's carrying out her PhD thesis at the Hellenic Paris Pasteur Institute in the group of structural neurobiology, and she will talk about her efforts to study the structure of human tyrosinase and development of novel inhibitors.
Good morning, everyone. My name is Evgenia Politi, and today I'd like to share with you a structural perspective on tyrosinase inhibition. My work focuses on how we can better understand this enzyme and create new models that can help design or study inhibitors more effectively. So, tyrosinase is a metalloenzyme. It contains 2 copper ions in its active site, and it's the key catalyst in melanin production, the pigment that gives color to our skin, hair, and eyes. It carries out the very first step in melanin biosynthesis by converting L-tyrosine or L-dopa to dopaquinone. Because it sits at the top of this pathway, even small changes in its activity can lead to pigmentation disorders. Oh, sorry. Okay. When tyrosinase is downregulated or absent, melanin is not formed at all. On the other hand, when it's overactive, excess melanin accumulates and we see hyperpigmentations, which are major cosmetic and dermatological concerns. That's why tyrosinase is a major therapeutic target for developing inhibitors, especially to treat hyperchromias. In our project, we have 2 main goals. First, to understand the molecular mechanism of tyrosinase inhibition, and second, to design a structural model that mimics the human tyrosinase active site, since working directly with a human enzyme is extremely challenging. Many structures of tyrosinases from different organisms already exist in Protein Data Bank, but the problem is that none of them fully mimics the human enzyme. The most commonly used protein, the tyrosinase from Agaricus biosporus, is easily accessible since it's commercially available. However, this commercial tyrosinase is characterized by batch-to-batch differences and impurities, and also the most important drawback is that it has very low similarity to the human enzyme. A closer example, a bacterial tyrosinase from Bacillus megatherium, shows much greater similarity to the human enzyme, and also it is easily expressed, purified, and crystallized, and due to these facts, there are already many PDB depositions of its structures. No structure is available to date, though, for human tyrosinase. One issue that might affect the lack of these structures is the fact that human tyrosinase is a glycoenzyme, which means that extra deglycosylation steps are required before subjecting the protein to crystallization. In our lab, we work with both Bacillus megatherium and human tyrosinases. BM tyrosinase was expressed successfully in E. coli and gave us a very high yield, around 80 milligrams per liter. Human tyrosinase was also expressed successfully in a stable HEC 293 cell lines we constructed, but in this case, yield was much lower, only about 1 milligram per liter. Human tyrosinase is a much more delicate protein and far harder to produce in the amounts we need for structural studies in the lab. That also highlights the fact that there are no structures available yet. When we analyzed both proteins in size exclusion chromatography, both of them eluded at the estimated molecular weights and showed satisfying purity when the peak fractions were analyzed in SDA spades. Furthermore, to make sure we had in hand fully functional tyrosinases, we ran enzymatic assays by measuring dopachrome, a brown tyrosinase product, over time. Both enzymes showed the expected catalytic activity and substrate conversion profiles. Then we proceeded to screen on both tyrosinases a group of compounds synthesized at the University of Athens by Professor Zoidis' group. One compound, named PP25, immediately stood out because it produced the same effect on both tyrosinases and an effect also comparable to that of kojic acid, a known tyrosinase inhibitor. After that initial screen, we measured its potency more extensively and found an IC50 of 0.9 micromolar using the bacterial enzyme. When we soaked crystals of the bacterial enzyme with a PP25 solution, the crystals visibly changed color. This was This is a classic sign that a reaction was taking place in its active site. This was our first clue that PP25 was not just an inhibitor, but also a substrate of bacterial tyrosinase, a dual behavior that we then confirmed with in vitro assays. Although it's obvious by the color that PP25 was soaked into the crystals, when we obtained the crystallographic data of these crystals, we found out that the active site cavity was empty. That is due to the fact that PP25 was consumed by the enzyme since it's a substrate. Therefore, to obtain a crystal structure of the compound and enzyme complex, we crystallized the bacterial tyrosinase inactivated by switching the ions of its active site with zinc instead of the catalytic copper. So indeed, we obtained a crystal structure of 1.96 angstrom resolution with great statistics. In the structure, we could see the PP25 approaching the active site, although we couldn't see in full detail the second part of the compound. However, this still gives us valuable insights on how the compound interacts with the enzyme. Now, when we compared the crystal structure of the bacterial tyrosinase with the AlphaFold predicted structure of the human enzyme, we found some regions that differ. You can see here in green the regions of the bacterial enzyme, and in pink the predicted regions of the human enzyme. We found that there were 4 amino acid residues and 3 flexible loops that were not conserved. To overcome this, we designed a chimera, a Bacillus megatherium tyrosinase backbone with 7 humanized regions, the 4 amino acid residues and the 3 loops that differed. This scaffold could become the closest structural surrogate to date of human tyrosinase. This is still a work in progress, so we're going to have to update you on how this will go. And to conclude, we first we identified PP25 as a novel substrate with inhibitory properties, and second, we designed a chimeric model that will help us bridge the gap between the bacterial and the human tyrosinase structures. We believe it's a promising step towards better understanding tyrosinase inhibition and help design new inhibitors effectively. Now I'd like to thank especially my supervisors Petros Giastas and Marios Zouridakis, all of our collaborators at the Agricultural University of Athens, the Hellenic Pasteur Institute and the University of Athens, of course all the members of the Structural Neurobiology Research Group here at Pasteur, and also I'd like to acknowledge these funding bodies that help support this project, and all of you for your attention.
Well, thank you for being in time. So questions, please. Okay. So thank you very much. Okay.
Thank you, Eugenia. Very nice talk. So I wanted to ask you, do you know also the KD of binding of the drug to the
We
protein?
now want to we now want to conduct experiments using ITC with an inactivated enzyme because we wouldn't get results using the active enzyme. So it's still, these results are very recent, so we have to, will be the first we will do.
Right. Okay.
Happen on that.
Yeah, great.
But we guess that the KD will be much lower than the original substrates. We want to conduct both using the substrate and the natural substrate and PP25.
Hi, thank you for the presentation.
So I have two questions. The first one, I'm not sure, maybe you already covered it. I didn't understand how did you, so you got the crystals of the bacterial one.
Mhm.
How did you superimpose the human one? I mean, you didn't crystallize it, so how could you compare the two structures?
It's the AlphaFold prediction of human tyrosinase. We only had that to compare it, but it's not the structure that the structure will differ because there are different amino acids. We compared the fact that there were different there were different amino acids as far as their properties were in thought.
You answered my question. My second question is, so you plan to perform some sort of screening, right?
Yes.
Will you do this simulation and how, like the screening, will you use compounds based, like the chemical structure of the compounds will be based on that compound that you found out, or you have a library, or?
We don't have a library in this scale. We have the collaborators that design inhibitors, so we're going to give them feedback about this and design the new generation. As far as the screening, we will do it in vitro and then proceed to in cristallo experiments. We won't do virtual screening.
Thank you.
Another question, please.
Hi. So does E. coli have a tan when you produce this protein and you add the right compounds?
Excuse me, I didn't hear the first part. Did E. coli?
Does E. coli have a changed color when you produce this protein?
The clones on the agar plate turned brown in the middle.
Could you use this property to screen them?
I don't think I could easily use it to have results that would not be easily affected by the conditions. I don't know if I can do this on a plate, having the same conditions exactly every time. I think it would require special media to make sure we don't have even differentiations of the media of the plate, or not have a false positive inhibitory effect. So we can see the clones that have a different color, but not all of them turn brown. It's not a characteristic of the E. coli, more of an induction of the expression without an inducing agent. It's not always the same.
Thank you.
Okay, thank you, Eugenia, once more. So we proceed to the next talk from Professor Angeliki Giannouli. Angeliki is an assistant professor of chemistry at the University of Crete, a member of the Royal Society of Chemistry. She has held her postdoc research in prestigious institutes such as the Weizmann Institute of Science, the CNRS, and the National High Magnetic Field Laboratory in the USA. Her work has been recognized with multiple international awards and fellowships, and today she will share her recent findings on protein conformational dynamics explored through state-of-the-art EPR, a project in collaboration with the Marseille University and the CNRS.
Hi everyone, thank you very much for the kind introduction and for having me here. I'm very happy to be here and very honored, I have to say, And I have to point out that the last time I gave this talk was at the Pasteur Institute in Paris in June. So if you were there, apologies if you hear this talk twice. So today I'm going to be talking about a protein called HTPG, and is a bacterial homologue of HSP90. And HSP90 is a very important protein of the cells existing in all organisms, in human, bacteria, and yeast. And is important because it's interacting with many other proteins called clients, facilitating their proper folding, maturation, and degradation. And it does so by using energy from ATP together with magnesium ion as an effect, important cofactor of the ATP hydrolysis. So when HSP90 works, it is processing the client, and it does so by undergoing conformational changes related to the binding and hydrolysis of ATP, and also the structural changes are further regulated by other proteins called co-chaperones. So to give you a perspective, HSP90 is very flexible homodimer, adopting open and closed conformations, and you will appreciate the difference here for the E. coli homologue crystallized many years ago from the agar group. And also you will also notice that the closed structure of the yeast homologue is very different from the E. coli homologue. And the protein, as I mentioned, is a homodimeric with its monomer consisting of 3 consecutive domains. The N-terminal domain, which is the binding site for magnesium and ATP, and the middle domain, which is the binding site of the client protein, and the C-terminal domain, which is always constitutively dimerized and is also a binding site for some other proteins, for some other co-chaperones. And we wanted to look at the structure of HSP90, of this homologue, in vitro, but also in the cells. And the way to do that is by using electron paramagnetic resonance spectroscopy, which is a fancy biophysical technique where you can have your protein and you need to introduce to it so-called spin labels, which are paramagnetic molecules. And we typically introduce them into cysteine residues. So if you have native cysteine and you know that the labeling will not affect the activity of your protein, you can exploit them, or you can do site-directed mutagenesis, introduce cysteine at desired positions, and then spin label them. And then we can measure the distance between these two spin labels by putting the protein in a magnetic field, and we can get 2 types of information. First of all, we can derive oscillations in the frequency of which are encoded the interspin distance from this equation here, or we can look at the CW, the continuous wave EPR spectra, which can already tell us if the spin label is found in a rigid region or in a loop. So the spectra of the spin labels are very distinct on the side of the protein. This is particularly important, let's say, for intrinsically disordered proteins where X-ray crystallography is very challenging. And another important parameter of the technique is a so-called modulation depth parameter, which gives information about the multimerization degree. So it has been applied to find out if a protein is homopentamer, hexamer, heptamer, and so on. So there are a lot of studies on that by now. And as I mentioned, we're going to be working from the bacterial homologue from Salmonella oneidensis in collaboration with Professor Olivier Zenest at CNRS, who is a guru of HSP90, and we want to introduce them into bacterial cells that have about 70% homology. So we're not exactly on the same organism, but we are very close to it. And we did site-directed spin labeling on HSP90, and for better seeing the system, we have the native cysteine residue here at the C-terminal domain, which we exploited for mutation. And then I also created 2 different single system mutants, one at a time, at the middle domain and the N-terminal domain. And because the protein is homodimeric above nanomolar concentrations, and for this technique we work in the micromolar concentration regime, we automatically have 2 cysteine residues, and we can obtain 1 distance from each of these mutants. And here are the positions in the open conformation. I just put it here to see how much its structure changes. And these are the continuous wave EPR spectra that I mentioned before, where I did the labeling with 2 different spin labels. And you shouldn't worry about what they say now, but from this spectra we can quantify the spin labeling efficiency, and we can also derive information about how much of the protein is rigid and how much is more flexible, and we're going to exploit them soon. And here I just saw the difference between the two spin labels, and the importance of this spin label is that it's stable in the reducing cellular environment. So if you put this in the cell, this is getting easily reduced to the diamagnetic hydroxylamine and is not EPR active anymore. So we have to be smart about the labels we use as well. So initially we looked at the distance between the different domains. The E62 is on the N-terminal domain, and the middle domain, the C-terminal domain, with the two spin labels. And we see that we obtain a very good agreement for these two positions when we compare it with the prediction from the X-ray structures that I showed you before. However, we have a discrepancy here for the N-terminal domain mutant. And we also tested the effect of the nucleotides in the conformation, and just give you here one example, but we have done it for all of them, and we see that, and this is the primary data, this data here in the presence of magnesium AMP-PMP, which is a non-hydrolyzable ATP analog, or with magnesium ADP, and we see that the distance and the oscillations do not change. So from this data we can say that in vitro, the N-terminal and middle domains of HSP90 adopt broad distance distribution, as you see, with one of them matching the X-ray structure. The C-terminal domain is well structured and dimerized in agreement with some data we also have published on the yeast homolog, and we observe no changes in the presence of nucleotides. And one important feature of this data is the modulation depth parameter, as I mentioned, which is very low and not typical for this sort of systems. Typically we observe modulation depth values of 20 to 50%, and this is very low. So this already tells us something. It tells us that the protein is found probably in an equilibrium between the open conformation, and this distance here is very far. It's very long to be measured with our technique. You see here, this is about 14 nanometers, and we cannot obtain such long distances with EPR. We can go typically, if we are lucky, up to 8 nanometers. So it means that we lose information, so we don't really see this open conformation, and what we see is a small fraction of this closed conformation. And this is for both these positions because the depth is very low. So from these in vitro measurements, we can not only get information about the conformation of the protein, but also say that we are observing an open-closed equilibrium, but we can only see the closed state. And then we wanted to put the protein in the cell and look at it in the cell, and the way to do that, we have developed some protocols for internalizing labeled proteins. So we have our protein, we overexpress it, purify it, we attach a spin label, we again purify it from the excess of the spin label, and we can use, for example, electroporation or heat shock to reintroduce it back into the bacteria. We've also done work on human cell lines. I worked also with HeLa cells, working with the yeast homolog, but this is for another day. But for this case, we use heat shock. I used heat shock to introduce it back in the cell. And these are again the CW spectra that I showed you before. And again, you will appreciate the difference between the in vitro data and the in cell. We have massive changes in the spectra, and we can now quantify them and see how much of the protein has shifted conformation. And this is in work in progress. We can also use this spectra to quantify how much is the concentration of the protein in the cell, and it's about 100 micromolar, as you see. And I would argue that this is actually physiologically relevant because HSP90 is very abundant inside the cell. But the technique is very also sensitive to much lower concentrations up to the high nanomolar range. So if someone has a protein that physiologically is few micromolar, you can easily apply this methodology to look at it in the cell. And then I measure the distance between the middle domain mutant and the C-terminal domain mutant here in the cell this time, after reintroducing the protein in the cell. And now we see that the modulation depth that before was very miserable has increased, and now the distance we get matches very nicely the one that is predicted for X-ray for the closed conformation. And the C-terminal domain mutation is very similar to the one obtained in vitro and matches also the model distance. So we were able to actually find and see that we have a closure of the M domains once we put the protein inside the E. coli cells. And we also saw that the C-terminal domain remained well structured and dimerized in agreement with the in vitro data. But what is interesting is the shift of the conformational equilibrium to the closed conformation inside the cell. So in conclusion, we can say that in vitro we have an equilibrium of open-closed conformation with the conformation shifted to the open one. But we have a shift towards the closed state once we put the protein in the cell. And from calibrating the modulation depth values against references, we can even quantify the amount of open and closed conformations in vitro. So you see it's mostly open in vitro and then it's mostly closed in the cell. So with this, I will finish my talk, and I would like to thank Mosbury for funding my research visit at CNRS for 1 month, CNRS and Marseille X, Elisabetta, Valerie, and Olivier Zenet for collaborating we had a great collaboration on this project, and of course, University of Crete, where I have my lab now, and we are continuing all these research independently. Thank you very much.
Thank you, Akiliki, for this very interesting presentation. So any questions, please? Yes?
Thank you very much. I wanted to know what are these, the knowledge that you acquired regarding the physiological function of this protein, or the regulation of the physiological function of the protein inside the cell. Can you make hypotheses?
So far, the only information we have about, so when ATSP9 is working in the cell, we know that it's dimeric. But we have no other clue what the conformation is about. So actually, we were also puzzled why this structure changes, and we assumed it's because of molecular crowding. But then we did some experiments with a crowder here, no, not this one, this one. But now we are continuing with more crowders, and we see that the effect we observe in the cell is not because of the molecular crowding, it's actually because of interactions with client proteins. And we have some other data on the yeast homolog that corroborate with that, but we don't know which, for example, client is responsible for this. So the only thing we can say is that in the cell, the protein finds its partners and is changing to accommodate, to perform its function. So this is a result of the function of ATSP9 in the cell. But we cannot say more about, which client protein this is and so on. We cannot say more about that at this stage.
Thank you. I may be wrong on this, but usually heat shock proteins, they tend to form higher oligomers.
It's true.
How can you be sure that this is a dimer? You did mention at some point the concentration, but do you have a control?
So we have a paper from last year on Biophysical Journal where we measured the KD of the dimer. So we know it's dimer, and we measured it with this sort of distance measurements. We measured it with MST and with native mass spectrometry. So we're very sure we are on the dimer, up to actually 300 micromolar concentration of protein. Our protein is dimeric and does not form higher order oligomers.
Sorry, and a side question. Where did you perform the native MS experiments?
Where?
Yeah.
In Weizmann Institute.
Because I come from the native MS world, so just
Okay.
Thank you.
So Michal Saron.
Thank you for the talk. It's clear that you don't know which client triggers this conformational shift. But did you try in your in vitro setting, in your in vitro experiments, polypeptide sequences that could trigger also this?
Yes. So what?
And the result was again shift to the closed conformation?
So what we did, but not for this one, for the yeast homolog, is we did mass spectrometry with trypsin digestion, and we found more than 300 candidates that can bind, and then we scored them, and I produced one of them and put it in vitro together with the protein, and we see the same effect. and we are actually working on publishing that now. But yeah, it was 4-year story and we still haven't finished it. So it's very difficult. So we see with one client we can reproduce the effect we see in the cell, but maybe there are other clients that do the same thing, But we can just say for one now.
Yes, one more question. Professor Fenopoulos.
Is it possible that following the initial binding at the C-terminal domain, clients and HSP90 interact also through other domains? Are there any data on this?
Yes, so there are cryo-EM structures. There are 3 or 4 cryo-EM structures where they show binding of the client, not only on the C-terminal domain. So the binding of the clients is done on the middle domain, but also the C-terminal domain has a flexible loop which is not resolved in the X-ray structure. And it has the MEVD motif, which is very standard for TRP cochaperons. So these cochaperons bind on the MEVD motif to HSP90 and then lock also on the M domain. So we also know that there is some cooperation with the middle and the C-terminal domain in the loading of the client. Yeah, this is true. This is true.
Is it possible that the opening of the dimer participates in that secondary bindings?
I don't know. I don't know. Maybe it could. It could, but so what we did, but again I don't know, so I'm just saying we did. So I truncated the other domain. So I only have the C-terminal domain. And we see that the behavior of the C-terminal domain only is different from the full-length protein. So this suggests that the other domains, are important for the client loading and the processing. But I think it would be an overstatement to say something more. Like we don't know much about what happened because, with the technique we only look at specific regions. We cannot map the whole protein at the same time. But it would be interesting to look, let's say, if we bind the client, what happens to the N domains and the M and so on, independently. Yeah.
Okay, thank you very much
Thank
once
you.
again. And now we move to the last speech of this section. We welcome Alexei Kubrenko, who is a PhD candidate at Agricultural University of Athens, and he's conducting his PhD research within our group here in HPI. And Alexei is going to talk to us about his structural studies on the ACE2 catalytic domain and efforts for drug screening.
Thank you, Dr. Zogdakis, for your kind introduction. So at first I would like to make a quick overview of what I'm going to present you in the next few minutes. Now, the main name of this study was in discovering ACE2 allosteric sites and lead compounds that could enhance the catalytic activity of angiotensin converting enzyme 2. Now, at first, I'm going to talk to you about why ACE2 activation matters, what was the structural strategy we adopted. To be more specific, we isolated the structure of this protein with a transition state analog in it, and also in its free state. Then I'm going to show you some screening and heat and how we identified those, some structural and kinetic evidence, and finally the challenges, the limitation, the current status, and the future steps of our study. So first and foremost, let's see what the angiotensin converting enzyme function in the organism is. Now, here is a really simplified presentation of the renin-angiotensin-aldosterone system. Some of you might know it as it implies many antihypertensive drugs, such as ACE inhibitors and angiotensin type 1 receptor blockers. But my presentation is not about this. So the main function of ACE2 in the organism is to hydrolyze this angiotensin 2. It's a tiny octapeptide. that cause vasoconstriction acting through the angiotensin type 1 receptor that causes blood pressure raise. So what ACE2 does is it hydrolyzes this vasoconstrictive octapeptide into angiotensin 1,7 that has vasodilative and anti-inflammatory properties that acts through the mass axis, and those causing decrease in blood pressure. Also, as some of you may know, ACE2 is also the entrance gate into the cell for SARS-CoV-2 and some other viruses I will not mention in this presentation. And since 2019, it became an object of intensive studies and prospective pharmacological target. So functionally, ACE2 is a monometallocarboxypeptidase. It digests a single amino acid from the carboxy terminus of its substrate, containing this characteristic zinc binding motif in the active site, and structurally it is comprised of 4 domains: the catalytic domain, the electrolyte domain, the transmembrane, and the cytoplasmic domains. After expression, it's being anchored to the cell membrane with forming dimers from these correctrin light domains. Now, in this study, we expressed and studied and investigated the role of the ACE2 catalytic domain. So we didn't express the whole protein, we just expressed this catalytic domain. It's the part that does the catalysis. So here you can see also an animation of how it is, how the dimers are in the cell membrane. A few words about materials and methods we used. The protein was expressed in Pichia pastoris cells, a well-established expression system in our laboratory. Then purified the immobilized metal affinity chromatography, deglycosylated for crystallization purposes, and finally purified using size exclusion chromatography. Then it was co-crystallized with some compounds that mimics that are transition state analogs of the native substrate. Then we shoot those crystals with X-ray, obtained the data, and obtained our crystal structure. Also, we performed some enzymatic and functional experiments that I will describe you in the next few slides. So here on the left panel, you can see some structures obtained by our research group. So this panel basically shows the subdomains of ACE2 catalytic domain, and its 2 conformations. The green one is the substrate receptive one, or as we call it, the open one. Without it is the apo conformation, and the yellow one is the conformation that we call active-like, that the protein takes in presence of the substrate or a competitive inhibitor. And here on this animation, you can see the active site. It's this groove over here, and the animation shows a proposed transition from open to closed state, to active-like state. Now, from a pharmacological point of view, there is a need in novel compounds that could enhance ACE2 catalytic activity, as they could be promising in treatment of hypertension and related pathological conditions. Also, we believe they could at least partially alleviate some of the COVID-19 symptoms by compensating enzymatically the depletion in available ACE2 molecules. Despite that, despite the 3 years of 2-3 years of pandemia, there is no experimental data on ACE2 allosteric sites, and there is a limited number of ACE2 activators reported in the literature, with those having low potency and often controversial mechanism of action. As on the allosteric sites of ACE2, there is only some in silico and molecular docking studies, so there is no wet experiment on ACE2 allosteric sites. And as I mentioned in the beginning, this study aims at discovering ACE2 allosteric sites and some lead compounds that could act as activators. So our first step was to perform a conventional enzymatic screening of an FDA-approved drugs library using this tiny fluorogenic ACE2 substrate as, and throughout this screening, we managed to determine at least 3 activating compounds with micromolar range EC50 value. Although, at this point of time, we were really happy because we get some results on what we wanted. But when we went to some HPLC assays, we done these assays in order to verify our results. And we used the native substrate of ACE2. Here you can compare them and notice all the differences in chemistry. The compound A2 that had the highest EC50 value, it was the most potent of the 3 activators, showed to be an inhibitor. Here you can see the IC50 curve. And that's really impressive on how the size and the shape of substrate you use in an enzyme assay can affect the classification of modulators you're looking. Either they are activators or inhibitors. And here we understood the value the structural studies offer us, as we have the chance to obtain electronic density map of our protein and look if there is any binder near or far from the active site. So in order to do that, we wanted somehow to stabilize our protein in a conformation that it takes when the substrate is bound upon. So we wanted something that could mimic our native substrate, although it should be something non-hydrolyzable. So the protein stays stabilized in its active-like conformation I showed you before. And for this task, it's a great opportunity for increased allofragment screening, for looking for modulators in crystal. And for this task, we went to our collaborator from the University of Athens, to Professor Dimitris Georgiadis, who has a great experience on synthesizing transition state analogs. Now, you can see he offered us this compound here, the DG81, or as you can see from the chemistry, it's the phosphinic angiotensin 2 analog. I remind you that angiotensin 2 is ACE2 substrate. So it mimics the substrate and it has the ability to bind to the active site and lock the enzyme in its active-like conformation. So it exhibited low nanomolar affinity for our enzyme, the phosphinic angiotensin, a great thermostabilizing effect, which agrees fully with the obtained crystallographic, with the obtained crystal structure. As you can see, the pseudo-substrate revealed and allow us to map some substrate specificity pockets, and we were able to build all the 8 residues of the substrate, beginning from the S1 prime position to the S7 position. Here you can see the obtained structure in really good resolution of 1.6 angstrom, and here you can see that the pseudo-substrate has an extended network of interactions with structural waters and substrate specificity pockets residues. And this gray cavity in here is basically the cavity, this gray space in here is basically the cavities that are available after the enzyme it's being in its active-like state. So here you can see, here you can get a closer look at this cavity. It is not isolated from the around environment, so it is connected chemically from the N-terminus communication channel and the C-terminus communication channel. So in simple words, what you can do from this, you can just take your crystal as you have them and take a library with small molecules with or any compounds you want, soak the crystals into the solution of your compound of desire, and then shot it with X-ray and see if this compound enters some cavities in here. So you can structurally identify if there is any interaction between your compound and the active-like state of protein. And concluding, the drug library screening showed us some contradictory results depending on the nature of the substrate. And also, as I told you previously, we succeeded in obtaining high-resolution structure of ACE2 catalytic domain. Also, it is the second available structure of the ACE2 in its active-like, in its closed state, beginning from 2001 or 2002. And this complex constitutes the ideal case for crystallographic fragment screening. Now, our future perspective is to utilize this complex, for screening of small molecule libraries, in aim to find potential allosteric binding pockets in the structure. Then further enzymatic screening of some HPLC assays will be performed to investigate any obtained compounds. And also there is a possibility of structure-based optimization of those binders to increase their potency. I would like to bring my acknowledgements to my supervisors, Professor Petros Giastas and Dr. Marios Zviridakis, to the members of our research group, the HFRI for supporting me financially, to our collaborators, especially Professor Georgiadis, Professor Stratikos, and Professor Spiroulias, and to Dr. Anastasia Bakali and to Assistant Professor Anastasia Aragiriou. And of course to all of you for your attention, and to the organizing committee for organizing this wonderful event.
Well, thank you, Alexei, for this very nice presentation. Any questions, please?
Congratulations for your work.
Thank you.
I wanted to ask you, why did you target allosteric sites and not the catalytic site?
Oh, no, probably I didn't explain that very well due to the lack of time. But as I showed you, if you allow me to show you some previous slides, if we have time also, do we have? Yes. Okay, thank you. Wonderful, wonderful. Okay, I guess this will do the job. Now, you can see the ACE2, maybe the whole ACE2, not only the catalytic domain. It's a single unit enzyme, so there is no multiple units that we could target to find some allosteric effect. And as about the active site, I guess it's a bit obvious. I don't know how obvious it is, but any compound that targets those substrate specificity pockets would probably act as an inhibitor. So we could have the exactly opposite effect if we target the active site. Did I answer your question?
Thank you very much. Can I make another one, or there is somebody else?
Yes, proceed with the next question.
There was a discrepancy between the data that we get when you use a fluorogenic peptide versus the endogenous substrate.
Yes.
But it could also be discrepancies if you do these experiments in vivo or ex vivo. Do you have in your plans some experiments to go in vivo or ex vivo?
Oh, no, of course. That's a very good question. If we're doing drug development, of course, the next step after the structural development, it will be the in vivo testing. I don't know, some human embryonic kidney cells tests. So, of course, we're not going to identify the substrate without testing it in vivo and do all the proper processes. So yes, we're planning to test that in vivo, but I have to tell you, the thing is we've not managed to obtain those crystals outside of our laboratory. So currently we're doing some crystallization. We're doing some optimization. So the work is a bit on pause now. We don't have any novel results because of that troubleshooting. And yes, after the structural determination of after the identification of any activators, they will also be tested in vivo.
Okay. I think that due to we have passed the time, if anybody wants to ask Alexei any question, he may visit him outside. So we proceed to the. Thank you, Alexei, for your presentation.
So, good morning. Now we have the round table. I would like to ask everybody to speak, to talk, and to sell, let's say, to tell your ideas about what we can do in the network in order to become better, to travel more as students and share ideas. So don't be afraid to raise your hand, say something if you have anything to say. So you can stay for the next round table. Thank you. Okay, let's do a 5 minutes break in order to stand up, blood to go to the brain.
You can have one. We are the same. Okay, we'll take it out. If you want, we can take it out now. Ή αύριο με την ή σε τώρα, δηλαδή και τώρα, μετά από το διάλειμμα. Δεν είναι. Άμα είναι με mail τότε καλύτερα σήμερα, αλλιώς όλα είναι σχετικά. Κυρία Θυμάμαι εδώ το λέμε στις σχέσεις μας. Όχι, όμως θέλω να το κάνουμε και στο διάλειμμα. Ναι, στο διάλειμμα. Dörre. Je cherche. Ouais, et bon, sur français ça va, mais en anglais, il y a un truc genre. Si, il y a un truc.
télécharges
à moins de faire une vidéo qui tourne bien. J'allais dire oui, comment tu fais pour avoir la présentation? Merci. Je sais que j'avais réussi. Après, j'ai de l'eau. Est-ce qu'on voit? Parce que il faudra toujours aller.
like guide us. Yeah. We can select the photos. It will be just the same. So okay.
Okay, everyone. Good morning, Kalimera. Welcome for this session with this roundtable that we call Towards a Network of Young Researchers in the Pasteur Network. I'm Hisham. I'm working for the Pasteur Network as a knowledge sharing officer, and I'm really happy to welcome you to this roundtable. It's really wonderful to be here in Athens, my first time, and I hope not the last one. I want to thank all the organizers, and mostly the UP and the STEPA organizer. It is really happy. The Pasteur Network is really happy to be part of this agenda, and we hope that next year, as Cecilia said yesterday, it's going to be much bigger. Our session is part of a bigger movement. We tried last year. We did actually a first event between STAPA and the Stefan Angelov Institute in Sofia. In last March, we did the first webinar about empowering young researchers within the Pasteur Network with experiences from Paris, Tunis, and here Athens, with Athena, Leah, and Dora. And now we wanted to make it a little bigger and to try to reflect on what could be this network of young researchers, inviting 3 new people from Sofia, Brussels, and Lille. And that will be the starting point, we hope, of a network of young researchers within the network, within the Pasteur Network. I want to insist on the fact that we wanted to invite people who are already organizing or coordinating collective of association of students that they are doing it by themselves within their institution, and we want to learn from this experience as Pasteur Network. And I think this experience could be very useful for all the students and the postdocs and the young researchers in general in the network. And we decided to begin with the Euro-Mediterranean region, but there are many others collective and association within the network, and we hope that we are going to meet them very soon. Okay, very quickly, why this network is important. We wanted to push for this network of young researchers because one of the main pillars of the Pasteur Network is knowledge communities. Is to push people, to encourage people, to give them the tools and the space to interact and to exchange. And we thought that because you are the future of science, it is the right moment from the very beginning to put you in contact and to push you exchange on all the opportunities that this network could offer to you. And when I say the network, it's more your network than our network. Okay, so I'm going to present our 6 speakers, 6 outstanding young researchers and also leaders and building communities. The first one is Athena Boutou from the Hellenic Paris Pasteur Institute. Athena recently earned her PhD in neuroimmunology at the Laboratory of Molecular Genetics at the Hellenic Paris Pasteur Institute. Her research focuses on the role of microligand, sorry if I'm misunderstanding, in CNS pathology and explores therapeutic strategies to enhance remyelination and CNS repair in gene-liking neurodegenerative diseases. Athena studied biology at the University of Athens, including an Erasmus Plus exchange at the Université Paris Diderot in France, and completed an international master in neuroscience as valedictorian, conducting her thesis at the Hellenic Pasteur Institute and Netherlands Institute of Neuroscience in Amsterdam. She also received a lot of awards from the Hellenic Academy of Neuroimmunology, FENS, EMBO, Hellenic Society of Neuroscience, and GLIA 2023. Athena also values international collaboration and she's actively engaged in science communication and a lot of initiatives, including leading the organizing committee of this event, the 8th Hellenic and Pasteur and Paris Pasteur Institute Young Scientists Conference, which fosters connection between young researchers across international Pasteur Network members. Our second speaker is Leah Manke from the Institut Pasteur in Paris. Leah is the second year PhD student at the Unit Cell Polarity and Migration at the Institut Pasteur in Paris. She's studying the effect of intermediate protein on the biology of glioblastoma cells. Recently, she was elected to the STAPA, the Young Researcher Association in Paris, to the STAPA direction as the new STAPA Pasteur Network representative. As such, she's actively promoting the exchange and collaboration between the young researchers and the different Pasteur Network members through the realization of scientific and social events. Leah studied overseas for the entirety of her higher education, notably in Netherlands, Sweden, Singapore, and Japan, and she's highly aware of the benefits of international scientific collaboration and exchange, and that exchange can bring to research and education. For her PhD, she came from Germany to France to study as part of the International Pasteur Paris University program. Our third speaker is Dora Wider from the Institut Pasteur in Tunis. Dora is a PhD student in oncogenetics at the Institut Pasteur in Tunis, researching the histological, immunohistochemical, and genetic profiling of colorectal mucinous and adenocarcinomas. I'm sorry. She's the president of the Young Researcher Association at the Institut Pasteur in Tunis, where she leads also initiatives to support collaboration and skill development among early career researchers. Dora was also the manager of several research projects like Alliance SHS Afrique, mostly on social science and in health, and also contributed to the Per Medina project on personalized medicine in North Africa. As a member of the Communication, Science and Society unit at the Institut Pasteur in Tunis, she works also on science communication, public engagement, and participatory research to make science more accessible and socially relevant. On my left, my fellow colleagues and Sofia, Lydia Kecitsira, from the Stefan Engelhoff Institute. Lydia is a first-year PhD student and assistant in experimental immunology at the Stefan Engelhoff Institute in Sofia. She's doing research in epigenic regulation of systematic lupus erythematosus in different mouse models of the disease. Before that, she defended both her bachelor and master thesis in the same lab, first as an intern and then as a specialist. So she has 5 years of experience in this field. In those 5 years, she worked with students at all stages on higher education, including high schoolers on their way to applying for university. Being so close to their age really solidified the idea in her that a proper support system is important, not only for the technical part, but also for advancing their career, but for keeping their love of science alive. This is why she's here to represent the young scientists of the Stefan Engelhoff Institute and their desire to build such an association of their own. Our fourth speaker, we are 6, is Elise Delannoy from the Institut Pasteur de Lille. Elise is a multidisciplinary scientist with expertise in microfluidics, organ-on-chip technologies, and biomimetic cellular models. She completed a doctoral thesis at Lille University focused on developing microfluidics system for 3D blood vessel culture and assessing vascular permeability, followed by postdoctoral research at the Institut Pasteur in Lille, when she established innovative accessible fabrication method for gut-up-chip devices using 3D printing. Her work explores host microbiome interaction and mechanical forces in intestinal models. Elise's cross-disciplinary background empowers her to create robust, cost-effective platforms for advancing biomedical research and personalized health solutions. But Elise is also the junior president of the Association of Young Researchers at the Institut Pasteur de Lille. She is actively invested in mentoring, outreach, and gender equality advocacy, and brings the dedication and teamwork on a high-level rugby player and coach to both science and community engagement. I know I'm a little long, but we still have a last speaker, which is on my right, Sharon Dill from Sint-Sano in Belgium, Brussels. Sharon is a medical doctor specialized in public health with a particular focus on cancer epidemiology. She works as a senior scientist and epidemiologist at Scienziano in Belgium, within the unit of cancer epidemiology in Brussels. Her research focuses on the prevention of and control of HPV-related cancers, with expertise in HPV-based cervical cancer screening and the clinical validation of HPV assays. She is currently in the final stage of her PhD, which centers on the secondary prevention of cervical cancer through improved screening strategies and test evaluation. In addition to her research work, she is also an active member of Scienziano PhD Group, an initiative launched in 2024 to advocate for and support PhD candidates. The group offers a peer-led safe place for sharing experiences, challenges, and advice, fostering a supportive community among early career researchers. Okay, now that you met all our panelists, I'm going to ask them a first question. So, we will begin by presenting your institute, but also the role of the students and the postdocs, mostly the young researcher in the institute. And please take a moment also to present your collective / association and your main activities. And on our back, we have some pictures of the different association and collective and their activities as well. We will begin by Athena, and then, yeah, I will give you the microphone.
You can hear me?
Yes.
Okay. So, Hellenic Paris Pasteur Institute is founded in 1919. It's the oldest public health and research center in Greece. Hellenic Paris Pasteur Institute is a founding member of the Institute Pasteur International Network, and the mission of our institute is built on three pillars: research, public health, and education. Research activities are organized in departments. We have three main departments: the microbiology, neuroscience, and immunology department, and also infrastructures, including bioinformatics, biotechnology, animal models for biomedical research, and transgenic technology unit. In the field of public health, the Institute plays a very important role in Greece through its diagnostic department, national reference centers, vaccine unit, and quality control unit, which are widely recognized in Greece, especially during the COVID pandemic. As part of its educational mission, Hellenic Pasteur Institute hosts undergraduate students and PhD students and master students, and also postdoc researchers. It's actively involved in organizing seminars. Also is part of master program and collaborates closely with the universities in Greece. So, which is the numbers of the young scientists in our institute? Right now we have 19 postdocs, 24 PhD students, 21 master students, and 38 undergraduate. So in total we are 83 young researchers right now, and together with early career researchers and staff scientists, we are around 100 young scientists that were active in our institute. So about the organization, the UPIs. UPIs were established in 2015 as a collective of young researchers, including masters, PhD students, postdocs. The aim is to build a collaborative and inspiring environment for young scientists. The founding team included Georgina Kouroupi and Antonia Evstathiou, and the first activities were some pizza lectures. We have monthly or sometimes weekly seminars in this amphitheater together with pizza afterwards as a networking and more fun activity. Also, they participate in many initiatives, including the Athens Science Festival, European Researchers Night, the Castelloris Opasi Festival. Dimitra Toubanaki and Antonia Evstathiou, together with Dr. Konomopoulou, were very active in these initiatives. However, during COVID pandemic, UP became inactive, And now we're doing some recent efforts to be active again and revitalize the community. We had an open call a few months ago for all the young researchers that they may be interested in participating in UPs again. Also, I have to mention that there were 2 joint retreats with TAPAS at the past, one in 2018 here in Pasteur Institute in Athens, and in 2019 in Rome, which were closed to only specific groups of UPs and TAPAS, but it was the first collaborative event. I don't know if I have to say more. Just to mention our conference as we're here. This was an initiative again of the UPs. It started in 2015. It was a one-day conference for the students within only the Hellenic Pasteur Institute to present their work in their colleagues. And again, this stopped during COVID, and the first event that happened again was in 2023, which was stronger than before. More people were participating, and it was the first comeback after the pandemic in the amphitheaters. Last year, that we also were involved in the organizing committee, we decided to expand it in a 2-day event. Also, we changed the language in English, which we thought it was a first step to be more open and more international. And we also invited the other institutes in Greece. And this was the first step, so this year we decided to open it even more. We invited also STAPAS and the Pasteur Network, and we think this is the first step for a bigger and larger initiatives. Yeah, thank you.
Thank you, Asina. Leah? So from Paris.
Okay. Yes, so I'm here to represent STAPA, which many of you have heard. We are the Young Research Association from the Pasteur Institute in Paris. And STAPA was founded in 2003, so it's quite a few years now. And we are every year growing. So now we are around 800 people on our mailing list. The main activities that we are doing are really the weekly social hours, which are a great opportunity because in the end, the Pasteur Institute in Paris is quite big. We have a large campus, we have a lot of students, a lot of postdocs, a lot of PhDs. So it's easy if you arrive in Paris to feel a bit lonely. So the STAPA social hours that we have every Friday are really a great way to just come for a drink and meet other postdocs, other PhDs. And it's always nice to see the same faces, but also always new faces, because Paris, Pasteur Paris is really a dynamic place. People come, people leave. So it's really nice to have those social hours every Friday to feel like a part of the community. And I feel like their STAPA is really nice because we have this mailing list where people can ask questions about life in Paris, but also about like scientific doubts or questions. So their STAPA is also really there to help every one of their members. And then in addition to this, STAPA is of course also very focused on scientific outreach. So we are doing several, I don't know, the slide is not up, but we are doing several events where we try to talk about our science to the general public. We're having, for example, the Pint of Science, which is like you are giving talks about your project in a more relaxed setting, which is really nice to bring everyone to the table and to discuss science in a really relaxed setting. And then we are also really trying, and then you can also see that we are really trying to, we are trying to do a lot of exchanges as well. So this year we went to Cambridge for one event with the young researchers in Cambridge, and we are very honored to be here in Athens now to have this conference with the UPs, and I think it's a great way to discuss science and be among other young researchers. And yes, STAPA is growing, and we are doing quite a lot of events. So yeah, if you want to know more.
Yeah, we'll discuss it maybe when the roundtable will finish. Thank you, Leah. Thank you. Dora or Tunis?
Hello everyone. So I'm Dora. I'm representing I'm the chair of the Association of Young Researchers at Institut Pasteur de Tunis. Institut Pasteur de Tunis was founded in 1893. It's the most ancient public health institution in Tunisia, and its missions are public health and diagnostics, vaccine production, research, education and training, and we have also some supporting units like grant office, knowledge transfer, and communication science and society. To talk about our association, maybe let's start from the beginning, the origins of our association. In fact, in 2016, a Young Researchers Colloquium was created or organized, and it was an initiative of our communication, the responsible of communication, and the challenge was that this event will be fully organized by young researchers, and this one of him, so I think he's our godfather for the association, I mean. So this event was a huge success. We were organized in several committees, like sponsoring committee, symposium, scientific symposium committee, social and cultural committee, etc. And since this event was a huge success, we earned, first of all, the trust of our leadership at Institut Pasteur and all the seniors in there, and also we gained trust in ourselves. I think we had to organize this event to believe that we can do this kind of event, and it's the most important event in Institut Pasteur de Tunis until now, because every 2 years we organize this colloquium, and this was at the end of 2016, but we didn't want to lose this dynamic, so we have the need to create a more permanent structure, and this structure is an association. So we began the procedures in January 2017, so with our first meeting with the founding members, and to create our chart or our vision, what will be our mission, have to write this, and then we go to the Tunisian government presidency, and we get our response from this structure in July, and in September it was the announcement in the official journal of Tunisian Republic. So in 2017, our association was created. What was our vision? What is our vision? Until now, we are dedicated to creating an optimal environment for young researchers and advancing scientific research in Tunisia. So our mission in that optic were divided mainly into volets, let's say scientific and social. But the main mission was to create a community, because we really needed a community where we belong to, as students, as young researchers, because every young researcher knows that it's not easy to have this long academic parcours. So our missions were scientific, like the organization of the Young Researchers Colloquium, workshops, hard skills and soft skills, like leadership, how to write a grant for getting a grant for postdoctoral or PhD students, master students, etc. And hard skills like bioinformatics. We see what are the needs of our association members, and every year we try to planify workshops that align their needs. What else? Scientifically speaking, also we are organizing, or we are part of projects, of international projects, like a TT project where we were interested in the mental health of PhD students, and this project was led by students, for students, And to be more concise, the social and cultural side, where we organize discussions, scientific cafes, we try to bridge more the gap with the society. Also, we host a lot of students, high school students, even children at the Institut Pasteur du Tunis. So we do a lot of scientific communication, science popularization, etc. And we do cine clubs. We watch movies together. We discuss these movies like Frankenstein and what is the dark side of science, etc. So we have a lot of fun, but also we try to advance research and training also. Thank you.
Thanks a lot, Tor. Thanks a lot for this testimony. Yeah, it's your turn, Lydia.
Hi, my name is Lydia. Some of you know me, and those people are actually on one of the. So it's not. Oh, it's coming. Okay, on one of the pictures behind me, because I was part of the organizing committee for last year's staff retreat. And what's interesting is that we managed to organize, in my opinion, a quite successful retreat without having an official scientific organization. And this is why I am here to actually propose our institute as someone who can in the future have such an organization on our own, because the difference is that the Stefan Angelov Institute is not the same as the Pasteur Institute. We are not a non-government organization. We are part of the Bulgarian Academy of Sciences. So we actually function under different regulations, but there is still room for us to make something that can be beneficial for students. So a quick history run. The Institute, as most of the institutes here, is actually one of the oldest institutes that is focused on public health. We were founded in 1947. We have the standard departments: general microbiology, biotechnology, infectious microbiology, virology, immunology, and mycology. And some, I guess, data, scientific staff only. We have people at all stages of their careers, including we have 2 academicians, but unfortunately, only 30% of them are under 35 years old. And this actually raises one of the main challenges, which is making our institute more visible for students who are in university. Because I am the prime example of someone who found my institute, which is now a big part of who I am. Something that I really love doing, but I found it on accident. So what we're hoping to do with finding such an organization is making it more visible. So this includes having like little booths and pamphlets at career forums, at Clemens Days, which is actually organized by Sofia University, which is where most of us are from, and something that actually Athena mentioned, which is the European Scientific Night. So I think that it would be great, considering that we have students who are still in university and are that live link between the academy and the university, to popularize the institute as an organization that is open to having new people that sometimes stays hidden. So another thing also is the networking, because, for example, the 2 other people on the board with me, which are Nicolette and Kalian, some of you know them, we didn't meet before Hisham actually gathered us together to work together. And there are actually quite a lot of other like PhD students that I've only like met on occasion, but we wouldn't have a reason to meet if it was not for such an organization. And actually being here now, it really becomes obvious that it is needed, not only to actually talk about science, but to be a safety net for each other and to be a safety net for the students that I'm already working with. So we can actually make applying for a PhD easier, or even stuff like graduating with a bachelor thesis or a master thesis. I also work with quite a lot of high school students. As you can see, it's still not there. Anyways, we organize summer schools for high schoolers. So this is quite like a career forum, but on a lower level, but it can still be very, very beneficial to them. And I guess I can talk a little bit about like the steps that we have in mind, because we already had like an official, unofficial meeting with all the young researchers in our institute about what we're planning to do. So first we have to make it an official organization, which is just based on the regulation model of the institute itself. As we said, we have like a separate set of rules because we're an NGO. So things like having a president, a vice president, treasurer, secretary, voting terms. So this is actually all things that I have to learn from you guys having already established organizations. And I think the main thing is how we would define young researchers based on the set regulations that we have in our institute. But in the end, I think that we can, as a first timers, we can start with a group of, a relatively smaller group of between 10 to 15 people. But still, that's because the main focus is actually recruiting more young people and making sure that they have a reason to stay here. And if we become more visible to the Pasteur network, that will be actually a great motivation to know that you can go even beyond the institute. So the idea is to make our stay in the institute, even though it's just for a bachelor's or a master's or for a PhD, even more enjoyable and more exciting knowing that you have all those opportunities. And yeah, we have some smaller things planned for the end of the year, which is the same way that we as PhD students have to have our yearly conferences when we like present our work, making that but without our PIs, so we can actually get a sense of what our colleagues are doing and share some ideas or just words of encouragement. And we'll see long term how things will work out, but I'm actually quite excited to present ourselves as also future collaborations for all of you guys.
Thanks a lot, Lydia. Yeah, Elise?
Hi. Hi everyone. So I'm from Institut Pasteur de Lille. So the institute was founded in 1894, so it's quite an old institute as well. But the Young Association is way more young, in fact. Actually, it was founded in 2021, but it was concerning only one lab of the campus, so it was not a general association. It was very dynamic. But last year, they decided to extend it to the whole campus and to the LB lab, so it became the Young Scientist of Pasteur Lille. And we even changed it more for the name of Youngs of Pasteur Lille, because our institute is quite has different purposes. So you have the research purposes with infectious disease, cancer, metabolic research, neurosciences. But we also have like a prevention department, like focusing on public health and receiving public on the institute. And we wanted to include them in this association because they are part of our identity. And we wanted also to include people from supporting departments and also all the technician engineers that usually wasn't really included in the events. So we have also a lot of young researchers, more than 135 PhD students and more coming with the years, a lot of postdocs, and also some young engineers and technicians. So we are kind of in a transition from the association from one lab to a general campus association. What they did in the past was mainly alumni career events, so they can share tips or the way they move through their careers. Organize also some social afterworks. And one main big event we want to focus on in the future is the PhD days. So it was organized for one lab, but in the coming years it will be organized for the whole campus. And also people that are not in the main campus, because like Lille is a separate research units. So the PhD days will really work to welcome new PhDs and give them a sense of community. So they will get some sweatshirts with colors so they can identify themselves on campus. Since we are some big units, we think it will really help them have a sense of belonging to the campus. We organize also some scientific conference and pizza gathering to share scientific experiences. And also more scientific trainings, because we have like some big platforms in the Lille campus. So we want the youngs to be able to profit on that. We are really glad because we are really supported by the director of our institute. He's really in touch with us and really motivated to give some ideas. The administration actually asked us to maybe organize a gala, so they are really also helping us to thrive. And they used to organize like big afterworks for the whole campus, and they actually give us their hand on this, so the young of Pastorally can really be empowered and organize their own events. So yeah, the idea is really to be open also for non-members of the bureau to be able to organize things, so they can come with an idea, and the idea is to empower them. It's not only the bureau that will organize. So yeah, it's a renewing and starting again dynamic, but we are really happy to be here and to be able to discuss all the initiatives from the other institute of the network. I think it will be really a strength to get connected together. Thank you.
Thanks a lot, Elise, for sharing this experience. Sharon, for Cienzano.
Hello, good morning. I want to thank firstly Cecilia and Hisham and everybody first time to invite someone from Belgium in Cienzano. But yes, Cienzano is the National Public Health Institute in Belgium, and we work on the concept of One Health, and we work with across human, animal, and environmental health. A fun fact, Cienzano in 1900 was known as the Brabant Institute of Pasteur Institute, and the director at that time, Jules Bordet, was a Nobel Prize winner for medicine and physiology, I think, but yes, for his work on immunity. And today we have 5 scientific directorates from epidemiology, public health, human health risk, animal health risk, data governance, and biological health risk, I think. And we have expertise in research, surveillance, as well as crisis response. And we are a community of 1,000 people now, so we have 1,000 employees, and 10% of them are young researchers. So we have 10% PhD students, and there's 3 ways they come to the institute. So they have their PhD either funded by external grants, funded by Cienzano, or they work full-time and they do a PhD part-time. So I'm in the category which is the 3rd category. I work as a senior scientist, and I also do the PhD on part-time. And the PhD support group actually started in 2024, so last year. We're still really in an infancy period, and it's really nice to come here to gain a lot of insights from everybody to see how we can develop this. And it started as initiation from one department. The idea was a lot of PhD students, for example, I'm never on campus. I don't go to the university, I don't see other PhD students. So it was really nice to come together as a PhD group and receive all this support together. And our activities are, we do monthly meetings. We have monthly meetings, but no pizza, but just, yes. And, and we have, for me, what's the biggest help in this is we have an online platform, which is we use WebEx, and there's a group of 100 people on there, and there's always exchange of simple questions. Can somebody read this or proofread this? What's the journal I should target? Where should I go if I have questions about this? It's really nice to have a community that you don't really feel inferior when you ask simple questions, because you work a lot with senior people. And it's nice to have the opportunity to say, okay, it's not really a stupid question. I can ask this and not feel like looked down upon. And it started as an initiative, like I said, in one department. We received a lot of positive feedback, and now it's in the whole institute. And we want visibility. We want to have a voice in high decision making as well, and really have a community, like you said. And it would be nice, I think, to extend this, and maybe we should have this meeting next year in Brussels. But yeah, I'm really interested to see what everybody else is doing, and also to hear your thoughts. Thank you.
Thank you so much thank you so much, Sharon, and thank you everyone for sharing your your experience, and it's really, really interesting. And it's totally different. Actually, you have a common experience, but also with your specificities and your diversity. It's really, really nice. Thank you so much. The second point we would like to discuss about is actually about how what could be the priority actually, according to you, or based on your experience, your common experience, each of your experience, on what could be the priorities and the mission of a network of young researchers within the Pasteur Network. So I'm going back to Athena. We'll try to do it in 1 minute each, if that's possible, or max 2. Thanks.
Yes, so I think the center of attention this initiative should be young researchers, as this is the purpose. The priorities should be to build a community. This can start by doing smaller, like as we started with STAPAS and Hellenic Pasteur Institute, and step by step this can grow to bigger, a larger community with more countries and more research centers. I think one very important thing for the young scientists is that they don't have the opportunity, for example, to travel abroad. So maybe trainships or short-term exchanges or some travel grants should be a priority. As all labs, they don't have the funding, or it's not their main priority to support the young scientists to travel abroad. Yes, so I think I would suggest that.
Thank you. Yeah.
Yes, I think for the Pasteur Institute in Paris, it would be most important to be aware of the existence of the other Pasteur networks, institutes, and then yes, to know what kind of research you're doing, to know that there are other young researchers in those institutes that would like to exchange with us, to share our joint experience in doing this, to do a PhD or a postdoc. So I think it's really just to be aware of the existence of those other institutes, and to also highlight your work, and to really, yes.
Okay, thank you so much. You can have more time. Dora?
I think that this type of event, I mean, this is the first time that I meet other young researchers from other associations in the Pasteur Network, because it widened my perspective. And I think what we have as a community in Pasteur Institute at Tunis, if I project what a community of young researchers among or within the Pasteur Network, it will be huge, as we can share experiences, we can share technical experiences, or also career experiences. So this type of event will be, I hope that it will be held for other years, and maybe one day it will be at Tunis. I mean, also maybe having a platform where we have all the opportunities for young researchers to collaborate, or to have funding, or to do mobility, so we can spread the word in our institution and you in your institution. And there is many opportunities among the Pasteur Network that we didn't hear of, unfortunately. And in our institution, we try to send mails and to communicate in our pages about this type of opportunities. So it will be nice if there is something more.
Thank you. Lydia?
As I mentioned, actually, as I said, is to make sure that our institute is more visible. On one side, that is more visible to students in university to know that they can and come apply and graduate with a bachelor's and a master's and then apply for a PhD in our institute, but also to make sure that everyone else in the network knows that we are here, that we're motivated that we can give you something if you knew that we were like open to exchanging people, and in this way, actually, for all the hopefully newcomers, we'll know that they can also be part of this network.
Yeah, sorry, Elise.
So in LEAD we had like several points that we really think are important to be involved in the network. Mainly the strongest point is about like facilitating visiting opportunities, internship, or for postdoc, like a few months somewhere else in the network. But for that, obviously, you need money, so supporting grants, obviously. Maybe for the Euromed, let's see, with from Erasmus, I think maybe scholarship or things like that could be arranged. But it also comes, I think, with some practical tips for researchers going abroad, so they can really be settled in easier, because sometimes it can be scary to just move from a country. So I guess the association in that could be a strong point. Also about the visibility that you all spoke of, we have like the network websites, but you don't really have like a space for the young researcher to be visible. It would be really nice to have like some siglet that we can really invest, or a LinkedIn page or something that we can really show all the achievements of the young researchers, so they could be visible for the whole world, but also within each others. I guess also about the job offers, they are also accessible on the Pasteur Network website, but you cannot see all of them. You have actually to click on each institute, so it's kind of making it more difficult. So maybe it could be nice to have a general thing where you can afterwards say, oh, it's there, but more maybe by topics. The idea also to have like this conference is really inspiring to us. Congrats again for the organization. It really inspired us, like maybe having like a turning conference that will be held in different institutes each year. So really young researchers could meet and discover the different institutes. It could be, I think, a really nice thing to do and to organize. Also to show people that organizing such event is not a small thing. So I think it's really a great professional experience to do so. And also we spoke about like organizing specific training and technical training on different campuses, because we all have our technicity and specificity. So we think it will be a good way to advocate for everybody's technical skills. And in the end, one thing that is quite important to us, it would be maybe to have a strong commitment from the institute directors. We thought about maybe writing a common manifesto or a letter of intention that will be signed by the directors to really support young researchers, so not discussing money just yet, but as a first step to really getting them involved and to care for the young researcher. So I guess it will be like something very concrete that we can start with all of us to share the values we want for the young researcher in the network.
Love it. Thank you, Elise.
I don't have a lot to add because I think everybody has said what I wanted to say, but I really like what you said, that maybe we need official recognition, a mandate to say, okay, this is an official network and we have a board, like everybody's on the board and all young researchers are involved. But I know funding is an issue, but we could start very easily with low-cost things. We can do frequent webinars. Different people from different organizations present their work. An inventory of current organized, like you said, an inventory of different young researchers open for communication. It should be easy for us to map out and say, okay, Sansano does this, and these are the young researchers there. Reach out to them if you have questions. Hellenic Institute have these people. A platform where it's easy to access, to see, because like you said, it's kind of confusing and you have to really navigate different, you have to do research to see who these young researchers are. So really to consolidate everything we are doing and bringing it together. But a mandate, I think it's really a first step to kind of recognize this officially, but yeah.
Thank you so much. Okay, now I would like to, I will to hear from you, what could be the 3 next steps in order to really create this younger. So you already said a lot of things, but I want you, we are not going to agree, but I would like to hear from you, because the idea is to develop a kind of roadmap for the next months, maybe the next years, together within the Pasteur network. And we want to hear from you what could be the next steps to really set up this network.
Yes, I think the first step starts inside each different institute. The first step is to organize, for example, inside Hellenic Paris Institute, and have an active, well-working association of the young scientists. The second step should be to reach out, start small and then scale up, reach out the other young associations, and maybe do some webinars, online meetings, which is the easiest step for us, a more easy way. And then I think a meeting in different countries each year or every 2 years would be a great idea for meet each other and actually being part of a network. Also, the organizing committee of the meeting could be included from different pasteurs. It doesn't have to be just the host institute. So we will also collaborate to prepare the meeting and do all the scientific input. Yes, so I think starting inside the institute and then reaching the other institute and scale up.
Thank you, Athena. Leo?
Yes, I totally agree. I think it would be nice to maybe organize a meeting of the institutes together. Maybe Paris would be a possible way for next year to start this whole thing, so that we invite a few institutes to Paris to afterwards plan it every year in another institute. Like this, we can really make sure that you get the visibility that you need, and to then afterwards plan other meetings in other institutes. So.
Thanks.
Thank you. I will not be speaking different things from my friends, but I think the first thing is to meet with the member of our institution, internal things, what we are aiming to do within this network, because I am speaking, but I don't know if the other member of our association, maybe they have other ideas, they have other needs. So it will be nice if we have something like internal, then we exchange, we meet with other associations, we try to align these things. And it will be nice that we organize webinars, etc., and activities online. In this way, we keep in touch and we keep this network alive, even online. And we will be happy to join other events like this. And maybe, as I said, one day it will be held at Tunis. But more visibility of this network and what we can do. And maybe also a platform, like Elise said, where we have more visibility of young researchers and where there will be opportunities included in this site. So I think there are, as Cecilia presented yesterday, a platform in the Pasteur network, and it will be nice, yes, and Hichem is responsible of that, I know. So it will be nice if we have a part, an independent part of this network of young researchers.
Thank you, Lydia?
So, yeah, speaking of small scale, I think the main 3 things that we have to do when I come back home is actually first meet with the young researchers that are counting on me to present our institution to you all. And then after okay, it's working. And then we actually have to present ourselves to our scientific council so they can be aware of our plans. and to actually get in talk with them in a way about how they can be useful to us and how we can be useful to them. So because the association is not only useful to us, like among us, but we can actually be like a moving force, because young researchers are the most long-term valuable asset of any organization. So in that sense, we have to like present ourselves, write down some rules and regulations, common manifesto. And then just by the end of the year, I think the first initial meetings to get to know each other on a personal level, but also on a scientific level, but not as formal. So we can actually know what actual work experience we can share, not only personal tips and tricks about surviving PhD.
Thank you, Olivia. No, sorry. Go ahead.
I think for us, so as everybody, we need to go first inward and to really advertise the network to the young scientists of Pasteur Lille, because I don't think everybody of them knows of it and what they can get from it. I think for me, really having a common commitment and values will be very important. So that's why I'm stressing on this kind of manifesto, and also to get support, because we need to gather forces, but we also need support from above to be sure that our initiative will be accepted and followed and supported. And I think one last thing would be also to have a kind of database or professional but social network when you can have access to joint grant possibilities, financial support possibilities, job offers, and also where you can promote what you are doing as a researcher. Maybe conferences you can attend, or prize, I don't know, you can apply for. I know in France, for example, and in a lot of parts of the world, there's the L'Oréal for women, for example, grant, and some of the girls don't know about it. So I guess it could be like a database that all Pasteur network young researcher could access and really gain strength from, because like today, the network is not strength for the young researcher, I think, and we should definitely benefit from it.
Thank you so much, Elise.
Yes, it's the benefit of being last. Everybody has said what I wanted to say, but just echoing on that is really to make it official. I think that's the main thing. And having common ground is the first step, and then we can work towards a goal that, okay, these are the young researchers. We could make a Facebook of young researchers, something like that. Not Facebook exactly, but a community that it's easy to access. No, not Facebook. But my personal experience is, okay, I am a Reddit user. I use Reddit, and there's a PhD support group, and it's international. It really is international. It's anonymous, and you get a lot of advice, a lot of opportunities, grants, discussing all the problems that you have, mental health issues. Something like that would be really interesting. But yeah.
Thank you so much to all of you, and it would be very helpful for us. I mean, when you see us, it's the Pasteur Network as a team and an organization. And for the last question, before giving the floor to the audience, I would like to hear from you. What are you waiting from the Pasteur Network as an organization to help you develop this network? Shoot.
I think, first of all, Pasteur Network should provide visibility. So Pasteur Network can highlight our research work in their website, in Pasteur Network communication, conference, report, has the power to make the young researchers' work visible. Also ensure integration, include young scientists from different pasteurs that are not only in different places. You saw a map with 32 institutes around all the globe. So it's very important to have representatives from all the different areas that have many different problems, different cultures, different way of working. So I think the network should be responsible for providing all young researchers a way to hear their voice. Also to help with the mentorship, for example, create mentoring programs between senior researchers and younger researchers. So, for example, if I'm interested for a postdoc within the Pasteur network, to have access to these labs and find these opportunities. And last, of course, a support like small grants to travel and to participate in Congress. For example, in big congresses, we can have some grants that Pasteur networks could apply, and the best of the applicants could have a travel grant to participate in whatever congress, not only of the Pasteur Institute. Yes, that.
Thank you, Athena. I just want to insist on our need as Pasteur Network to help you develop this network. So of course, what you're talking about, it's important, but how can the Pasteur Network help you formalize this network of young researchers? This is my question. Thanks.
Yes, so I think that, for example, this event right here, this conference, this was is an amazing opportunity for us to really live this community, and I think it would be nice if the Pasteur Network can really support those events, those conferences where we, as young researchers, are really the focus. So it would be great if we could make this an annual thing and to really get also financial support by the Pasteur Network, and so that we can extend it, that we can not only do it maybe in Europe, but also extend it to other areas.
Thank you, Leah. Dora?
Thank you. There is value that, again, Cecilia talked about it yesterday, and it's equity. And coming from North Africa, I think we don't have, unfortunately, the same opportunities. So if the Pasteur Network could support us with mobility to have access to some technologies, some labs that we don't have at our institution, to, as you said, to provide some little grants to young researchers to communicate their work, to have visibility. And I think also supporting these kind of events or other maybe events or that we'll be organizing together is very important. It's kind of more official event as a Pasteur Network Young Researchers Association. So that's it.
Thanks, Dora. Did you?
I actually agreed about the part for equity, considering that we are not an Institute Pasteur, we are associated members. Maybe more opportunities that are focused exactly on countries that are not exactly underprivileged, but we are maybe like a few levels under, to make it easier and more accessible even on a financial level for young researchers that actually have the same level of motivation, but just not the same resources.
The Stefan Golov is a full member of the network. It's not an associate member. It is not called Pasteur, but it's still, you are a member, like Cienzano, for example. You don't have the name, but you are part as any other member. Equity.
For me, I think the one thing Pasteur can do is really coordinate the initiatives, because we all have a lot of ideas, energy, but we don't have a lot of time because we need to be able to do research. Already we are coordinating from our own institute, and it's already taking a lot of time. So I guess we could really benefit from the experience of the network to coordinate such things and to really propose like the initiatives and the ideas. Also maybe to offer like kind of the method that it works for, like the senior parts, and say, okay, we do things like that, what we can take from it. So we can have like concrete example of how things are done, and it can be also used for our own institute and own association, I think. Definitely help on the fundings. I think that's the main thing. If you want to organize things, apart from webinars, we need fundings. So I guess it will be very important as well. And definitely keeping in mind equity, and that all the institute doesn't have the same resources though. So maybe help us on that as well, so everybody could be represented in such in the network.
Thank you, Elise.
Yes, again, really everything has been said, but really Pasteur Institute should act like the catalyst, like really to expand this. I'm really glad that I met everybody. It's an opportunity that was brought by Pasteur Institute, and the network is going to be a node for all these young researchers to come together, but really to build a bridge between everybody and then to form a bigger community. I think Pasteur Institute should be the central and also the catalyst to have all this collaboration as well.
Thank you. It's more the Pasteur Network than the Pasteur Institute.
Sorry.
Yeah, no worries. Just because, yeah, we are living an evolution, and this is the Pasteur Network. Here we are representing the Pasteur Network. Thank you, Sharon, for sharing your advices. Okay, so now we're going to give the floor to the audience for 20 minutes. Yeah, so if you have any question, maybe we can share the mic. Is there any other mic? We want to hear from you because most of you are students, and we would like to hear from you. Is it resonating? Do you have any advices? Do you have any idea that could complement what we already discussed?
It's more like a question for if you know, because I'm kind of new in all this Pasteur thing. But there is chances to do like exchange between pasteurs. For example, if I am in Paris Pasteur Institute and I want to, I don't know, do an exchange to another Pasteur Institute, is that possible? Like as a young researcher trying to maybe look different areas. You know, I'm not sure if it's
no.
clear.
Well, could you please be just a little bit precise on when you say by change, but like do an internship, do your postdoc?
Yeah, it's like, for example, I'm a postdoc and I'm not sure if it's possible to, I don't know, talk to someone that is doing a technique that I need and go there to do that technique, but not a whole postdoc thing, but more tiny things.
Actually, there are some fundings, mostly from Paris. Paris is funding some young researcher to do this kind of internship. And I know that, for example, in Tunisia, because I worked for Institut Pasteur in Tunisia for many years, the government is giving sometimes fundings to do some manipulations, some specific experience for 2 or 3 months. So you can have this, because you don't have the necessary material or the machinery at your institute, you can do it outside, for example. And you can have some funds also, complementary fund. We don't fund as Pasteur Network this type of activities, but maybe that is something that we can. You want to add something, Cecilia? Yeah, no, wait a sec.
So, very practical. Hi. No, actually, just to add what Hisham was saying, is that what you can do, I mean, once you are in a stage, for example, that you are in right now in Institut Pasteur, all activities that you will doing, meanwhile you are doing your research in Institut Pasteur in Paris in that time, it depends on Paris, because you are working like with them. What we can do and where we can interact is when you finished, I mean, and you can just go to another institution, or maybe we can just search together the possibility with you know, and just see what is happening around the world and what could be the opportunities for you, for example. This is something that we can do. But we cannot, I mean, of course we can help, but we need to interact with Institut Pasteur in Paris. Meanwhile, you are doing your research in Paris, because there is a lot of practical assurance and things, details that I'm going to enter into it. But yeah, of course.
You want to add something? I have a question.
Yes, so I was just wondering, so what is the main, how much money does Pasteur Network have, if I can ask the question? And then how much money do you spend on the young researchers? Like, do you have like 10% that you spend on mobility of young researchers, or you don't really have young researchers as a category in your budget?
And maybe I will add something.
Okay. So this is a good question, thank you, because it's really important that everyone can understand how the Pasteur Network is financed. I mean, how it's built, the budget of the Pasteur Network. So basically, because I'm giving you the word, I can just...
You can come here.
No, I'm fine. It's just because I don't want to just stick with my back. So the fundamental money resource of the budget coming from the institutions. So every institution pays the fees for the Pasteur Network, and this is we can say an important percentage of the budget. With this money, what we're just trying to do is just to attend the institution needs, which are mobility, which are project, for example, Spark, which is a call for proposal, which is meetings, kind of, depends on the board of decision, annual meetings.
And courses.
Courses, exactly, courses. So this is coming from the fees. This is what, and of course, what you call the mission, which is us, right? So just give me 1 second, I'm gonna finish. And so this is the big part. And we are waiting, I mean, we are not waiting, sorry, we are like having discussion for raise this amount of money because we are going to have more needs and more needs. I mean, it's coming from the network. I mean, coming from the institutes of the network. So this is a discussion that we are having. And the other part, it's different funders. We receive money from, I don't know, it depends on the project, but Gates Foundation, Rockefeller Foundation, Honky Jockey Club, Wellcome Trust, Durable, I mean, from European Commission. We have different diversity of funding, but because what we want just to keep, it's our independence on decisions. Yeah. I don't want to give you the number of how much money we have put in. This is clear? Okay.
But would it be possible to say, okay, 50% of all the participants that do mobility or do seminars or do, could be young researchers up to 30 or 35, and like this you promote mobility of young researchers?
Yeah, I mean, all related to mobility, it's for researchers, young leaders that we're going to do in the network annual meeting, and young researchers, and that's why we are here. Because what you need just to understand, again, and I will finish with that, is that we, as a Pasteur Network, we had a board. We are not the one taking the decisions. Who takes the decision is the board, and that's what I like your idea. What we need is the commitment of the institutions. We are only just the implementing body of the network. So if we can get the real commitment of the institutions, and that it's became a priority, we are just ready to start working on that. I mean, and we are pushing the idea. Huh? That's why we are here. Otherwise.
Yeah. Thank you, Cecil. Yeah, we have two questions there. Now, can we give him the mic?
I don't need the mic.
No, but we need it because it's recorded. Thanks.
Can you tell us more what mobility is? Because you keep saying mobility, but what activities are within the mobility area?
Yeah, I don't have the slide that I presented yesterday, but actually for us, mobility, it's one concept, which is building community. How we can do this in two senses. First of all, at the level of the network, we have all related to scientific working groups, where we have groups for different, and they will have training courses. And then, for example, we have projects, just for giving you an example. Yeah, okay, I'm just taking it. For example, for the pillar 1, we have, which is epidemic surveillance systems. We have on a project, we can climate and health, and we have fellows. So this program, for example, it's funded by it's a Rockefeller Foundation. Right? Yeah, for the Rockefeller Foundation. And we took young researchers from different institutions, and then we became a specialist, we can say, or the person could be responsible to be the connected between the institutions and governments all around the climate and health. So we have different programs. We don't have just only one. And that's why I agree also that the visibility, but building community also, is that everyone is aware in what we are doing. But again, the link and everything, I mean, something important that we need to understand here is that why the network became an independent association. became because what they want is just to take decisions and response to the need to the all institutions and be part of the network. So for me, it's very important that every one of here make our own homework, coming back to home and say, okay, this is important for Tunis, Lille, Sofia, Cienzano, Greece, Paris, and we can just spread this into the network. We need just to become this in a need, in a priority, and then in a reality. And for that, and just want to finish with that, I took note, really, concrete notes, what all you are discussing here, but we need just to become this in a case. I need numbers. I need impact. I need concrete result of these meetings, because I know what they are going to ask me. So I need to have these answers. I know. I'm just being provocative here, but we need just to think that this is going to be really priority, so we need just to prove it. And this is part. But if we are here, it's because we are really, really open to hear and to the things. So we count on you. I really count on you.
That's my last words. Sorry.
No, that's fine. We still have 10 minutes, so maybe one.
Actually, sorry to intervene.
Yeah,
we're out of time. He sent us we're supposed to stop 20 minutes ago, so
okay. So I thought we were on time. Yeah. So okay, maybe a last question, and then no.
Yeah.
Just
okay,
have a
very quick question.
Okay, 5 minutes. Okay.
In terms of the actual network, so I'm just at Pasteur in Paris for about a year now, and I'm not really too sure because about how to access like contacts in the different network. I don't know if it's possible to set up a mailing list between all the networks, or if any of these meetings, if any representatives, like I don't know what representatives take part in these meetings, like when they are, if they're open forum or not open forum. I think it's not very advertised or discussed, and maybe that could be done or something. I'm not really sure. Yeah. I don't know if that's possible. I know there's like a lot of organizing going on.
You mean how working with a space where everyone can exchange and?
For example, if there's like a network board, I'm not sure like what representatives are in this. If it's like in an open format where like just people a part of the network can access it and watch it, or if there's reports on kind of what is discussed and what progress is made each year, or if there's anything like this. Yeah.
So first there will be the Netherlands meeting. The opening ceremony is open to everyone. You should be able to receive an email we'll find a way to on YouTube. We have an annual report, so you can have and the management report. Everything is on our website, and we are quite clear, I think, on how we are functioning and our responsibilities, but also members' responsibilities. So this is what Cecilia was talking about. That's it. Maybe, Raphael, I don't know if you, is that fine? Yeah. Very, 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 very quickly.
It should be very quick because maybe it's not a question, it's just a remark. And you partially started the remark that I wanted to say is that I think most of us are really unaware of the members, the actual members of the Pasteur Network. And just as an example, I just want to do a little test. You can raise your hand if you knew that there was like a Pasteur Institute in Lille or Suzanne, for example.
The question.
No, this is not really.
Did you know that there is a Pasteur Institute in Lille?
Yes.
Yeah, but not you.
And there are still a lot of people in this room that are not raising their hand because they didn't know until now that there was a Pasteur
Now
Institute.
they know.
And now they know, and the only problem is that we are 35 members. Some of them are getting in, like soon for Tokyo, for example, or some of them are getting out, and we don't know about the life of the Pasteur Network. We don't know about the institute themselves. So the more we know about each other's, even only the existence of what we are doing between each other's, the more we will want to collaborate between each other's. Because, like for example, now we went to Athens. So who wouldn't want to go to Athens to go to a conference between young people, or to go to Paris for a conference between young people? So I think this should be the priority for the Pasteur Network, actually.
Thank you.
Thank you. Okay, so I know we're out of time, so I would like to say three things. First, I was really impressed. I'm sorry, you want to answer? No, go ahead. No, because I had to conclude. No, because okay, but okay. Okay, thank you. But I would like to say just three things very quickly. So, the first thing is that when I was listening to you during the first question, I was really impressed by your commitment that the love, and yes, I would want to talk about love. The love that you have for your institutes and for the things that you're doing, and how much you would like to interact much more, to exchange, and to share your experiences, but also the challenges. We know that being a PhD student, being a postdoc, being a researcher in general, it's hard, it's difficult, and the journey is long. So you need support and not to be lonely. That's the first thing. The second thing is that, of course, yes, there will be a platform, and it will be for free for everyone, and there will be a group specifically for students, postdocs, and young researchers. It's it's a mix between LinkedIn and SharePoint, when you can share files, write projects, etc. And also, it's a social network, and you will have all the opportunities from the network. But this platform is your platform. I mean, if you want something to appear, you can just do it. It's a co-creation. We're working together here. And the last thing is that, and actually it's built on what I just said, and Cecilia said it a lot of times, you are the network. We are here to facilitate the things that I think we consider that could be important for you, but you can do it. Honestly, this is something that you just need to take some responsibilities. I think that we have now a lot of good ideas that we are going to write down on what could be done, like maintaining this event, having a common chart, maybe something at the level of maybe involve the students in the decision making. We'll see, but it's also on you. So thanks again, and see you next year maybe.
But before you leave, Ilya, we have some gifts for you from coming here, all of you, from Tunis, Lille, Belgium, Sofia, and we are very happy to have you here. And we hope that this is the beginning of a new adventure. So, and for you, Cecilia, too.
Thank you very much.
Well, sorry, we don't have a group picture.
We have to take a group picture.
You have to.
No, she needs to be in the picture.
Send this for you. I have the big one.
Yeah,
the big one. Thank you so much. Thank you. Ah, sorry.
And now go to the garden and eat the post.
Βασίλη, γιατί έφυγες?
Έχω πίσω να φέρετε.
Άλλασε στο πλάνο. Yes, it will. Nice.
One more.
Amazing.
One more.
Foster and gardening. Hola, ¿cómo estás? Sí, me acuerdo, me acuerdo perfecto, ¿cómo estás? ¿Qué me has dicho? Yo te
start for the immunology session, which I have the great pleasure to chair along with my friend and collaborator and researcher here at the Hellenic Pasteur Institute, Yannis Rabias. Yannis is head of the Quality Control Laboratory here at the Hellenic Pasteur Institute. I head the vaccine unit as well as the Laboratory of Medical Microbiology. And without any further ado, because we have quite a long list of speakers, maybe we should start with Eleni Toukaki.
I was here to present you first, not Eleni Toukaki.
You were doing the presentations. Okay, I'm sorry about this.
So, I will present you again. This is the fourth session of immunology. Our first chairperson is Dr. Yannis Rabias. He is a research staff scientist and head of the Quality Control Department at the Hellenic Pasteur Institute, a member of the Pasteur International Network. A chemist by training, he brings more than 20 years of experience in the pharmaceutical quality systems, including GMP and ISO certifications, as well as vaccine and sera testing. Dr. Rabias serves as a member of the European Pharmacopoeia Group of Experts for Human Vaccines and Sera, and his research interests include analytical methods, nanomedicine, and the development of novel quality control approaches. Our second chairperson, Dr. Dionysios Gouras, is a research director at the Hellenic Pasteur Institute, heading the Laboratory of Medical Microbiology. His research focuses on the mechanism of bacterial and viral pathogenesis, with a particular emphasis on the human gastric pathogen Helicobacter pylori, a WHO-certified carcinogen and major risk factor for gastric adenocarcinoma. Under his direction, the laboratory also serves as the Greece's national reference laboratory for Helicobacter infections, providing diagnosis and microbial resistance surveillance, virulence profiling, and genomic analysis of clinical isolates. So, thank you for being the chairs of this session.
Okay, thank you very much for the introductions. So we start with our first speaker, who is Eleni Toukaki from the lab of Konstantinos Lazaridis. So Eleni Toukaki studied molecular biology and genetics. She has a master's degree in molecular biomedicine, and she's a third-year PhD student performing her research at the immunology laboratory of Kostas Lazaridis. The title of her talk is Pharmacokinetic Improvement of an Antigen-Specific Therapy for Myasthenia Gravis, an investigation of its mechanism of action. Eleni, the floor is yours.
Thank you for the introduction. This is on?
No.
Now it's on? Oh, yeah. Thank you for the introduction and all of you for being here. Good evening, everyone. Today I'm going to present to you my research on recent efforts to develop an antigen-specific therapy for myasthenia gravis. So, myasthenia is an organ-specific autoimmune disease characterized by muscle strength reduction and fatigue of the skeletal muscles. Here in the picture we can see the neuromuscular junction, which is comprised by the presynaptic nerve and the postsynaptic muscle membrane, which is rich in acetylcholine receptor structures, where the neurotransmitter acetylcholine binds, thus causing muscle contraction. However, in myasthenia, there are autoantibodies that target components of the acetylcholine receptor complex, and are responsible for the inhibition of the nerve signal towards the muscle, leading to the pathogenicity of the disease. Most myasthenia gravis patients have autoantibodies against the muscle acetylcholine receptor, which is comprised by 5 homologous subunits, and most of these autoantibodies specifically target the extracellular domain of the alpha-1 subunit of the receptor. There are some treatments for myasthenia, but remain non-specific, mainly acting through generalized immunosuppression. and most exhibit a short-term effect with often adverse complications, variable success, and patient-to-patient variability. To date, no antigen-specific therapy is available. There have been some efforts from other teams to develop antigen-specific therapies that, in rodents that rely on the mucosal administration of acetylcholine receptor domains, but provided limited success, in part due to difficulties with the animal model used. For such experiments, the Lewis rat animal model is mostly used, and it can develop experimental autoimmune myasthenia after subcutaneous immunization with the whole receptor, leading to mild and chronic disease that can in part mimic the human condition. In our lab, we have developed such an EMG model by single immunization of Lewis rats with the extracellular domain of the alpha-1 subunit of the human receptor. We used then this improved rat model to develop an antigen-specific therapy for myasthenia gravis. And in some of the following slides, I will provide some more background on some of the lab's previous results concerning this endeavor. So, a typical course of our experiment follows therapeutic administration 7 days after immunization, and symptoms occur around day 35. What's important is that in our experimental timeline, the therapy is truly for a therapeutic purpose, not a preventive purpose. Our approach involves intravenous administration of the human alpha-1, and as we can see in the first graph, intravenous injection had a strong therapeutic effect compared to the intranasal route, which had shown positive results in the past, but also when compared with other routes, such as the intraperitoneal and the subcutaneous route. In the same study, it was also shown that intravenous treatment with alpha-1, 7 days after immunization, had a dose-dependent response with higher doses exhibiting a stronger therapeutic effect compared to smaller doses. And more specifically, administration of 100 micrograms of alpha-1 per dose had an optimal therapeutic effect as presented by the reduction in clinical scores, the increase in body weight, and impulse muscle transmission. We have also seen that administration of alpha-1 to either healthy or EMG rats has the same effect, indicating that the presence of autoantibodies against the protein does not affect the protein's pharmacokinetics. But what we also observe from this graph is that alpha-1 exhibits a short plasma half-life of around 15 minutes. And since our protein remains a limited time in the bloodstream, and due to the needs for large protein quantities overall, there is an urgent need to find ways to extend its survival and potentially optimize its therapeutic potency. For that reason, and as part of my PhD, we engineered alpha-1 polyethylglycol conjugates and alpha-1 incorporating nanoparticles. But for the purposes of this talk, we will only focus on the alpha-1 PEG conjugates. So, at first, we constructed those alpha-1 PEG constructs by using 2 different molecular weight PEGs, 1.2 kilodalton and 5 kilodalton PEG. And we performed electrophoresis to identify the pegylation percentage. These constructs were used in different PEG to protein ratios as well. And as we can see from these charts, all constructs appear to be homogeneous and exhibit an increase in molecular weight in correlation to the PEG bound. We concluded that 100 times of a molecular ratio of PEG to protein was optimal to apply for serum half-life studies. And in these serum half-life studies, we saw that both alpha-1 PEG constructs exhibit an increased half-life in the serum compared to the unmodified protein, which is rapidly degraded or removed. And even at the end of the observation period, the alpha-1 PEG constructs are still present, while the biodistribution in the organs remains relatively the same. Now, since the 5 kilodalton PEG created larger constructs and exhibited a significant increase compared to the other PEG, it was chosen for in vivo application. In this in vivo study, we compared multiple alpha-1 PEG doses, 100 micrograms, 20 micrograms, and 5 micrograms, and applied the same dosage of unmodified protein. As we can see in the graph, with a bright blue line, the 100 micrograms of unmodified protein had a strong therapeutic effect compared to the 20 micrograms and the 5 micrograms, which is expected according to our previous findings. But what's most important here is that the 20 micrograms of pegylated protein, represented with a bright red line over here, has a similar therapeutic effect with the 100 micrograms of unmodified protein, indicating that pegylation can allow us to lower the therapeutic dosage of alpha-1 to 1/5 of the initial dose. Another very important part of this current study is the elucidation of the mechanism of action behind the therapeutic effect of our protein. For that reason, we measured the proliferation of lymph nodes lymph node cells by thymidine incorporation assay, and splenosyne-derived B cells by alkaline phosphatase assay. And as we can see in this graph, cells isolated from treated animals present a reduced proliferative response in both organs and assays when cultured in the presence of alpha-1 compared to the untreated rats. Furthermore, we reported a significant increase in the Treg population in both, again, the cells derived from the spleen and the peripheral lymph nodes of treated rats versus untreated rats. This effect was accompanied by a statistically significant decrease in inflammatory cytokine release from cultured lymphocyte supernatants, again derived from the spleen and peripheral lymph nodes of treated rats versus untreated rats. And most importantly, we saw that effect on inflammatory cytokines such as interferon gamma in an early time point of day 30, as well as a later time point of day 70, further supporting the long-lasting effect of our therapeutic approach on a molecular level. So, to conclude, alpha-1 PEG conjugates have increased serum half-life and require less protein for the same therapeutic result in vivo, allowing us to apply 1/5 of the initial dose for the same effect. And as for the mechanism of action studies, we saw that therapeutic administration of alpha-1 shifts the inflammatory response towards a regulatory phenotype. In the future, and as part of my PhD, we aim to do further studies where we utilize the alpha-1 PEG to lower the number of total intravenous doses. We also plan to utilize polymers that incorporate alpha-1 and study their in vivo therapeutic effect. And finally, we will continue to examine the mechanism of action of the therapy by further studying the proliferation of lymphocytes, the molecular profile of lymphocyte subpopulations and their cytokines in response to treatment, but also in different time points throughout the course of the treatment. Okay, at this point, I would like to thank all my supervisors for their unlimited support, the members of my lab, the Department of Animal Models, Biotechnology Unit, and Flow Cytometry Unit of the Hellenic Paristeur Institute, and finally all of you for your attention. Thank you.
Very nice talk, Hellenic. The paper is open for questions. Can I have a quick one? So, what do you think is the explanation for having a much higher persistence of this
the pegylated
in the pegylated form in the bloodstream?
Yeah.
I looked at the liver levels, accumulation levels, and they seem the same, so.
Here?
Yeah.
Yeah.
Did you actually look at other organs like the lungs, and you're giving it not subcutaneous but intravenously?
Intravenously.
Right? So you should have also accumulation in the.
Okay. For this experiment with the pegs, we haven't checked other organs besides the ones that are mentioned. For the unmodified alpha-1 in general, there is a study that they have checked like throughout the heart tissue, the lung tissue, the brain tissue, everything. We just don't have the data over here. And why it does persist in the blood for longer? Well, it's injected intravenously at that point. And well, the PEG generally protects the protein, okay, so that it doesn't degrade as fast.
Reduce degradation.
Yes, it will degrade in a slower way than the other one. So that's why we have the difference with between the two PEGs. This is 5 kilodalton PEG, stays at about 40%. The other one is like at around 10%. There's a huge difference between them. So the overall size of the construct, which is shown over here as well, is bigger, so it degrades more slowly. And since it doesn't seem to accumulate in the liver, it's a good thing. At least it stays in circulation, gets inside the organs, gets filtered outside of the organs, stays in circulation for longer, it exhibits its therapeutic effect that way. Yes?
Actually, a chemical synthesis question. Is it the direction that you made with PEG, is it quite stable? Have you tried when you synthesize it and some months later, have you seen signs of instability?
Oh no, it's very, very stable. And yeah, I have checked my constructs in a later time point. And it doesn't degrade again. This protein is not released, and they're very stable even when you defreeze them and stuff like that. They're very good.
Perfect.
Okay, I have a couple of questions. How big is your protein?
How?
How big?
It's around 27 kilodalton.
Okay, why did you put such a small PEG? Is that sufficient to change the pharmacokinetics of the protein? Or there are models of PEG from, I don't know, 5, 10, 20, 40, 60 kilodalton. Depends on what you want.
Well, okay, at the beginning we started small, but it wasn't optimal. But from what I've read in the literature, is sometimes it's not optimal to choose like 40, maybe 20 kilodalton PEG, because then you may have signs of it won't degrade as easily. I mean, the concept is that eventually our protein will be released, and it will be possibly internalized by cells and exposed in a tolerogenic matter. If I fully disclose it, I won't have an effect.
Okay.
Thank you.
Well, perhaps the nanoparticles then maybe.
Well, the nanoparticles. Last year I had the talk about the nanoparticles. It's a work in progress.
If you're thinking of releasing the cell, how do you produce the protein?
We produce the protein in a Hellenic Paris model. It's excreted in the culture medium, and we isolate it from there.
Okay. Thanks.
Thank you. Thank you very much.
Okay. So we're going to the second speaker, Ioanna Moitou Theophani. She is from the Biomedical Research Foundation of Academy of Athens, the lab of Dr. Andreakou. She holds a master's degree in biotechnology from the Agricultural University of Athens, School of Applied Biology and Biotechnology. She's currently working as a research assistant at the Laboratory of Immunology of the Central Clinical Experimental Surgery and Translational Research of Biomedical Research Foundation of the Academy of Athens. her title talk is Deciphering the Biological Role of Immune Cell-Derived IFN Lambda. And you can, the stage is yours.
Hello everyone. Thank you for giving me the opportunity to be here today. And I'm going to present you my project titled Deciphering the Biological Role of Immune Cell-Derived Interferon Lambda. So let me start with some background information. So interferons are cytokines with major antiviral properties which are produced by various cell types in response to a viral infection. They are classified into three families, type 1, type 2, and type 3 interferons. In this study, we're going to focus mainly on type 3 interferons, which in mice include two members, interferon lambda 2 and interferon lambda 3. Type 1 and type 3 interferons have low sequence homology and they use distinct receptors. However, they share the same downstream signaling and they activate common interferon-stimulated genes. But it is still unclear whether these two systems can interact and whether one system can affect the production of the other one. In a previous work conducted in our lab, it was shown that interferon lambda is produced first in the respiratory tract to limit initial viral spread without activating inflammation. If the infection escapes this control, then type 1 interferons become the dominant mediators of the antiviral response. However, at the same time, they induce pro-inflammatory responses which cause tissue damage. According to that, the current dogma suggests the compartmentalization of type 1 and type 3 interferon responses, which means that interferon lambda, which is produced mainly by epithelial cells, is important for battling the infection at the anatomical barriers, while leukocyte-derived type 1 interferon comes up as a second line of defense when there is a systemic spread. However, from this project, we wanted to see whether immune cells can also produce interferon lambda in the context of a systemic viral infection, and also understand how this production is regulated. To monitor the production of interferon lambda on a per cell basis, we used an interferon lambda 2 EGFP reporter mouse, in which whenever interferon lambda 2 is expressed, EGFP signal can be detected. So using this reporter mouse, we want to see whether immune cells can produce interferon lambda. So to do so, we injected intravenously poly-IC to the reporter mouse. Poly-IC is a double-strand RNA synthetic analog, which is used to mimic viral infections. So we injected the mice with poly-IC, and 4 hours later, we checked different immune cell populations for their capacity to produce interferon lambda. We did that by flow cytometry by monitoring the EGFP signal. And this approach identified CDC1 dendritic cells as the main source of interferon lambda production by immune cells. And this comes in contrast to what we know until now, and shows that an interferon lambda can be produced by immune cells in the context of a systemic viral infection. Then we wanted to understand how this production is regulated and also investigate a potential interaction between type 1 and type 2 interferon systems. So to do so, we crossed the reporter mouse with mice that were lacking either type 1 interferon signaling or type 3 interferon signaling, or both. We injected again poly-IC to these mice, and we monitored the capacity of CDC1 cells to produce interferon lambda. And we saw something really interesting, that in the mice that were lacking type 1 interferon signaling, there was a strong reduction in the production of interferon lambda. So this comes in contrast to what we know from the current dogma, and shows that immune cell-derived interferon lambda production depends on type 1 interferon signaling, and it is proof of a unique interaction between those two systems. Then we wanted to identify the specific type 1 interferon that affects the production of interferon lambda by CDC1 cells. So to do so, we did RNA-seq analysis in CDC1 stimulated cells, and we saw that the EGFP-positive CDC1 cells, which are the ones that mainly produce interferon lambda 2 and interferon lambda 3, did not express most type 1 interferons, except from really low levels of interferon alpha 5 and a bit higher levels of interferon beta 1. This led us to hypothesize that maybe interferon lambda production by CDC1 cells depends specifically on interferon beta 1. To check our hypothesis, we used an in vitro cell culture of bone marrow dendritic cells. So in this protocol, we isolate bone marrow cells from the mice. We culture them for 7 days in the presence of FLT3 ligand, and on day 7, the cells are fully differentiated, and we continue with all the stimulations and the analysis. The protocol that we use is according to publication that shows that although the dendritic cells that we generate in vitro do not express the same markers as the in vivo ones, they're equivalent to them according to their function. And this is the gating strategy that we use to distinguish the different disease subsets that we generate in vitro. So to check our hypothesis, we sorted CDC1 cells. We treated them for 1 hour with interferon beta and then stimulated for 4 hours with poly-IC. And then we checked the production of interferon lambda by monitoring EGFP signal by flow cytometry. These are some representative plots from the conditions that we had, and we can see that the untreated cells did not produce any interferon lambda. And this shows that interferon lambda is not produced in tonic levels and needs a specific stimulation. Indeed, after poly-IC stimulation, we saw an induction in the production of interferon lambda, which was amplified upon interferon beta pretreatment. So this shows that interferon beta affects the production of interferon lambda by CDC1 cells, and also it is proof of a direct effect of interferon beta treatment on CDC1 cells. Having understood the important role of interferon beta in regulating interferon lambda production by CDC1 cells, we wanted to identify the DC subset that is the source of interferon beta. So to do that, we utilized an interferon beta YFP reporter mouse in which whenever interferon beta is produced, YFP signal can be detected. So we generated bone marrow disease from these mice, we stimulated with poly-IC, and then we monitored the production of interferon beta. And we can see for PDCs and CDC2 cells that there was no induction in the production of interferon beta upon poly-IC stimulation. However, for CDC1 cells, they expressed interferon beta even when they were unstimulated. And this shows the homeostatic levels of interferon beta production and comes in contrast to what we saw for interferon lambda that needs a specific stimulation. And we can see that upon poly-IC stimulation, there was an induction in the production of interferon beta by CDC1 cells. So then we isolated the YFP positive and the YFP negative cells and did qPCR for interferon beta and interferon lambda. And we saw that the YFP positive cells were the ones that mainly produce interferon beta. So this shows that the reporter mouse is functionally correctly. But what was more surprising was that the YFP positive cells, which are the ones that mainly produce interferon beta, were the same with the ones that were producing interferon lambda. So in summary, we know from the literature that in anatomical barriers, interferon lambda is produced first by epithelial cells to battle the infection. However, our data suggests that when there is a systemic spread, interferon beta needs to be produced first by CDC1 cells in order to shape and license these cells to produce interferon lambda. So I would like to thank the organizing committee for giving me the opportunity to be here today and present my data, as well as the team at the Biomedical Research Foundation of the Academy of Athens, where all of the experiments have been conducted. And I'm happy to take any questions.
Do you plan to perform any experiments with real transfections? With real infections, actually?
Yes. Right now, we want to, we have seen that interferon lambda is produced systemically, but we don't know what the role of this production is, of this interferon lambda that is produced systemically. So to investigate the role of interferon lambda systemically, we are planning to infect the mice with a systemic virus, such as Zika virus, and then monitor. We're also going to use mice that do not express interferon lambda and monitor what happens in the context of real systemic infection.
Any other question?
move on with Siherim from Institute Pasteur in Paris. Sehi is originally from South Korea and she's trained as a medical doctor in Norway. She's currently pursuing a PhD in infection immunology with interests in infectious diseases, global public health, and bioinformatics. Her research focuses on vaccine development against a type of E. coli, a major source of diarrhea in children and travelers in developing countries. Her talk, the title of her talk is Mapping Human Immune Responses to Enterotoxicogenic Escherichia coli in a Controlled Infection Model to Guide Vaccine Development. The floor is yours.
Hello, good afternoon, everyone. It's really nice meeting you. My name is Sehi, and today I'm going to talk about my PhD project, Human Immune Responses Against Enterotoxicogenic Escherichia coli. So I came into research because I was interested in global health, and my pathogen of interest became Enterotoxicogenic Escherichia coli, also called ETEC, and it's a major source of gastrointestinal infection, especially in children and travelers in lower and middle income countries. So per year, there are 70,000 deaths, and 25,000 of them being younger than 5 years old, and then it's causing 7.5 million diarrheal episodes in the world. And currently there is no vaccine for this yet, so that's why I wanted to do some research in this. So the aim of the study was to characterize the immune responses against this bacteria, and sub-aims was like define the baseline immunity against this bacteria, and then test some vaccine candidates ex vivo, so that we can define some vaccine efficacy of some of the vaccine candidates. So yesterday at the lecture, we actually learned that we are supposed to do no harm to humans, but this is actually quite the interesting model that we're using, which is called the controlled human infection model, which basically means that we recruited some volunteers, and they got infected with the bacteria with ETEC, and they were hospitalized for around 7 to 10 days. But we had a lot of security and safety measures, and this got permission from the ethical committee. So if you want to talk about this infection model, I'm happy to take some questions on that. So all in all, we recruited 30 volunteers. So we collected a lot of different types of samples from them. For example, from blood, we had the serum, plasma, and PBMCs. And we also collected stool samples and also saliva. From all these samples, my focus was actually systemic immunology. So I used plasma, serum samples, and PBMCs. From plasma samples, I try to look at the inflammatory proteins using O-link and mass spectrometry, and metabolites with mass spectrometry. And from the PBMCs, we used mass cytometry and spectroflow cytometry with over 30 markers per single cell. So I am summarizing the results from two of my studies. Of all 30 volunteers, there were some who developed diarrhea, and some who did not develop the diarrhea. So you can see here, there is the diarrhea group, and there is a non-diarrhea group. And here's the timeline after infection. So this is like the baseline, and most of the volunteers developed diarrhea before 48 hours after dosing session, and then it lasted for around 3 days. And then we collected lots of samples, and for the baseline immunology, for those who got diarrhea, we see actually some early innate immune responses with the innate cell populations coming up on day 1 to 3, with a decrease in mite cells, and also increase in different types of dendritic cells and also different types of natural killer cells. And when it comes to metabolites and inflammatory proteins, we see increase in CRP, C-reactive protein, total amyloid A, neopterin, and kidney-renin tryptophan ratio. And we see also decrease in vitamin B6. And as time goes by, here is a bit adaptive immunity kicking in. So we see actually T-cell immune responses and B cells as well. So when it comes to T-cell immunity, we see most of the TH17 type immune cells. First of all, with the effector memory cells, and its counter cell, which is the TH17 effector memory regulatory cells. And also we saw plasmablasts that are producing a lot of antibodies against this bacteria. And on day 10, we see TH17 central memory cells, which means that our body is trying to keep the memory of this bacterial infection. But in the non-diarrhea group, they did not have this kind of innate immune responses, but rather they got their adaptive immune responses a bit earlier in the phase, which may mean they may have seen similar bacteria before, or yeah, we are not really sure, but their adaptive immunity is a bit earlier on the phase. So, past year, so I've been analyzing data from Olink proteomics. So we used Olink target 96 inflammatory panel, so that we can see 96 inflammatory proteins from the plasma. And antigen-specific T cells, we used the antigen-induced antigen-induced marker assay, which means that we stimulated PBMCs with different types of antigens from ETEC, and then saw rises in antigen-specific markers, which is CD69, CD134, CD137, and CD154. So from these analysis, we found out that there are some early responses from day 1 to 0, and we see some very general classic inflammatory mediators, and then it actually included interferon gamma. And then we actually see a very early rise of interleukin 17A, C, CCL20, and CXCL chemokines. So even though cellular immunity comes up a bit later in the phase, we actually see this interleukin 17 cytokines earlier in the phase, so that's a new thing. And also in the later response, on days 7 to 10, we see persistent interleukin 17A and C expression. And all these general inflammatory mediators are sort of calmed down. We don't see them anymore. And we actually use PBMCs from day 10. And we see these cells that were stimulated with different types of ETEC antigens. So antigen-specific central memory T cells from day 10, they are particularly TH17 type, which is characterized by CD90, CD39, and CD161, and also ROR gamma T. Yes, and they were enhanced upon stimulation with, there are several ETEC antigens, right? And then these are some of the antigens that showed especially enhanced increase of these type of cells. And they're also vaccine candidates, so that's very nice. Yes, so to complement my systemic immune responses, we actually had some measurement of gut wall thickness from different regions of gastrointestinal tract. So this was measured by ultrasound by clinicians, and we actually see a bit thicker gut wall in ileum region, and then this is supposed to be having a lot of receptors for toxins from ETEC. So When this gut wall is thicker, it means there was edema, and then this is like a targeted region for ETEC. And we don't see this kind of tendency in other parts of gastrointestinal tract. And this has also a correlation with some of the inflammatory proteins, for example, kynurenine tryptophan ratio, and also C-reactive protein in plasma. So I told you about this model, and it was quite difficult to get permission from the ethical committee to have more than 30 volunteers. So statistical power has always been a question here. So we wanted to compensate our data from milieu interior cohort at Pasteur. So this is the data set from 1,000 healthy donors, and then we collected their whole blood, and it was stimulated in different settings. So we compared this data on stimulated heat-killed E. coli and purified E. coli-derived LPS. So 33 genes were stimulated with E. coli but not LPS, which means that, and these 33 genes were very much associated with the Th17 cell differentiation. So we could confirm that the non-LPS components of ETEC was driving Th17 differentiation. Yes, so to conclude, immunity against ETEC from innate to adaptive. In the early phase, we see quite broad and unspecific immune responses with NK cells, dendritic cells, and mite cells, and also lots of different inflammatory proteins. And this is also reflected in gut wall thickness changes. But in the later phase, we see very much of a Th17 targeted immune response. And these Th17 cells with the central memory, with an ability to have central memory, is especially enhanced by CS5, CS6, and CFA1. So yeah, so that was a short introduction to ETEC immunity. Thank you very much.
Well, thank you very much, Serhi, for this talk. Very interesting. So, if I may kick with the first question, did you, in your group of volunteers, right, the 30 volunteers, did you actually, first of all, what is their age stratification? Are they young people, older people?
Yes, so when we wanted to recruit these volunteers, we were actually targeting first-year medical students. So it's mostly women because, women are better at getting good grades in Norway. So medical studies are dominated by almost like 80% of women in Norway. So it's actually women in the early 20s, and also, so that is...
And do you have any indication about the profile of their microbiome, gut microbiome? I mean,
yes, so that was actually a thing we really wanted to try. But from the previous studies, there are some challenge studies from the US, for example, and they have looked at microbiome from the feces, but it was very heterogeneous, and it was quite difficult to confirm with a very low number of subjects. But it could have been a very good idea. But we kind of saw that E. coli with some E. coli increases, of course, and there are some protective genus. But we would like to try it if there is a funding for it.
Questions? From the audience?
Thank you. Thank you for your nice talk, and also congratulations for your work. My first question has already been covered in part from Dr. Zgouras, but would be again in a more broad aspect. which were your inclusive criteria for the enrolled volunteers, and if there was any specific excluding criteria?
Yes, that's a very good question, and thank you. When these volunteers were recruited, it was crucial that they have never been to, they haven't been to e-tech endemic countries over the past 2 years. And then there are other criteria that we have gone through, for example, general clinical history, and some gastrointestinal diseases, for example, inflammatory bowel diseases, and they have been eating well, and they are functioning well in terms of gut health. So yes, that was sort of excluding criteria that they haven't epidemiologically infected with e-tech, and also some other kinds of diseases in gastrointestinal.
Okay, thank you. There is time for another short question.
I was wondering how important is it in the cases of the children that are dying because of this disease? Is it mainly because of the disease itself, or because they maybe have another parasite infection that is ongoing at the same time, and they are kind of what's the cause of death in the end?
Yes, so the cause of death is actually correlates very much with socioeconomic situation, because cause of death is actually dehydration and also malnutrition. So actually treatment for this bacteria is not antibiotics. You just need to hydrate yourself well and eat well. But in these countries, it's a little bit difficult to provide them with this kind of resources. So that is the main reason why the mortality is high for this kind of bacteria. And there is also quite a lot of co-infection with other gastrointestinal pathogens for this bacteria. So yes, yeah.
Okay, I have another question. It's quite short. You mentioned that you had gut wall thickness changes in a short phase. Have you checked if this thickness was reversible at later phase?
So when it comes to gut wall thickness, so they come to a baseline level around that after day 7-ish. Yeah. So it's a temporary change. The reason why the gut wall is thickened is that they absorbed water, and then it's become edematous. And then when the diarrhea phase is over, infection phase is over, then it kind of goes to the baseline level.
Okay, thank you very much.
Okay, thank you.
Thank
you.
you.
Very much. Okay, let's go to the next speaker. The next speaker is Tom Roblin. He works in the Institute Pasteur Paris. He's a PhD student there, focusing on the development of diagnostic tests for viral infection within the Diagnostic Test Innovation and Development Core Facility, a group from 30 rows. His research combines molecular biology, biochemistry, and de novo protein design to advance diagnostic innovation. He holds an engineering degree in biotechnology and biochemistry from Lyon and gained experience in biomedical engineering at the City of the University of Hong Kong. His title talk is Real-Time Population Immunity Profiling to Enhance Epidemic Preparedness, Insight from High Throughput Serology of SARS-CoV-2, Influenza and RSV. The floor is yours.
Okay, thank you for the kind of introduction. So today I'm going to talk about SeroWatch, which is a study we did in our diagnostic platform in Institut Pasteur in the LIPS program of the EU. So the goal of the study was to do a seroprevalence snapshot, so a human profile of the infection in Ile-de-France, so in Paris region at a given time. So for the context of the study, so you have your population, and there are several viruses. So we focus on respiratory viral infection, so SARS-CoV-2, influenza, and RSV. And some of them are getting infected, but how can we measure, how can we be sure how many of them are infected by that? Because it's very important for public health policies. So there's different reporting systems. There's like passive reporting systems where there's notification from hospital emergency department or laboratory notifications, mortality data. And there's also active surveillance through doctors with the patients. But we can ask ourselves, are we really seeing everything? Because those methods of reporting are relying on symptomatic cases, on healthcare seeking behavior, and we can ask ourselves if we are not underestimating the incidence, particularly for the asymptomatic cases and the mildly symptomatic cases. So the big question of this study was, okay, who is infected really, how many of them, and also who is not protected, because so that we can adapt the public health policies. So we designed the SEROWatch study. So when we did it, we focused on the pandemic preparedness view, and we were, how can we do a snapshot of the immune profile of the population in a very short window time, in case there is a new pandemic, for example. So we got 3,000 samples from leftover diagnostic blood draws that represent people from 0 years old to 101 years old, with equity between male and female, and they are coming from the Paris region, Ile-de-France. So there is 8 departments in it, and with different density, which is very important because we know that density is a factor of infection and epidemiology. And all of those samples were taken during 12 consecutive days, so that we can have a very precise snapshot at a given time in a very precise window. It's not even 2 weeks. And so what we did is we measure the IgG, the IgA, and the IgM level against the nucleoprotein of the 3 virus, SARS-CoV-2, influenza, and RSV. And also we measure the IgG avidity, so that we can know for everyone how many immunoglobulins they have, and also for IgG, how good are they in recognizing the nucleoprotein. And we did it with the LUISA technology, which stands for luciferasing immunosorbent assay, but I will present it just after. So it took us 8 days to do everything, with 6,000 tests per day, and for each sample we did 15 tests, and it was only €3 for the 15 tests, €3 per samples. Okay, so now let's dive a bit into the engineering part of the study. So first we have to know what is a VHH, or an antibody. So we do, as a human, we have heavy chain and light chains in our immunoglobulins, but camelids have also heavy chain only antibodies. And we use the variable part of these heavy chain only antibodies to have a specific probe to recognize our immunoglobulins. So we infected acute alpaga with IgG, IgM, and IgA, and we got the specific VHH. Then we expressed it in E. coli as a fusion protein with a luciferase. So the luciferase is a protein that is doing light, a very nice blue light, and so that we can do the LUISA test, which is basically an ELISA sandwich test, but with the bioluminescence, we can do the test faster, cheaper, and it's also more sensitive. Okay, so about the results, first we were looking about the IgG, so that we can answer the question, who has been infected in the past? So those are the results. In red you have for the SARS-CoV-2, in blue influenza, and in pink RSV. First, if we look at the distribution around the 3,000 samples, we see that for influenza and RSV it's very compact, whereas for SARS-CoV-2 it's very diverse in the response in the population. And this is maybe due because of its new emergence in the population, because it's only been 5 years that the pandemic started. And we use a threshold to separate positives from negatives, and we see a lot of IgG seronegative for SARS-CoV-2, which was okay, but also we saw few IgG seronegative for influenza and RSV, which was very uncommon because normally everyone has already been infected by the flu or by RSV in his lifetime. So who are they? So actually there is no difference in sex. We have male and female in both IgG seronegatives. And about the age, we see that we have youngers, which is, it does make sense because they didn't have maybe the time to build a proper immune response that can persist in the serum. But we have also 5 adults, which is very a question we ask ourselves. Are they lacking a immune response, for example, because those persons can be at risk for a new infection? And also, when we were looking about their IgM and IgA response, 7 of them were negative, seronegative for all of the isotypes, which is problematic because there is either a lack of immune response and a lack of immune protection. But let's go back to the IgG. After we were looking at who are the low responder in the population, and if you take random distribution, you're supposed to have 1.6% who are the low responder in the 3 viruses, but we see 4.2% of the data set were low responder in each viruses, which is higher than what was expected. Those persons, there is a U-shaped distribution with age, so it's mostly at the younger and the older person, but it's actually concerning every age group. And when we looked about the avidity of IgG, so the strength of the IgG, we kind of have the same results. It's because we saw that the more IgG you have, the better they are. When we segmented by age, we obtained this. And what we see is that we have IgG increasing with age until a certain point, and then it plateaus for the rest of the age group. For influenza, it was about 40 years old, but for RSV, it was 30 years old. And this is because of the chronic reinfection throughout life until a point where there is a saturation of IgG in the serum. And for SARS-CoV-2, there is no age dependency. We haven't seen that, and this is because it's very recent. Okay, now let's look for IgM and IgA, because this is very interesting because we can know with them who has been infected recently. And yeah, so this is our data. We got a very high seronegative component with almost a lot of persons, and then a few seropositive individuals, which was what we were expecting. And actually, our segmentation is working very well because in black here you have a negative serum dilution, and here in color you have different serum dilution of positive, and we see a clear impact of the serum dilution on the IgM or IgA response. So from our data, we found that between 25.9% to 29.5% of the person had detectable IgM or IgA against at least 1 viruses. But when we took the data from the French active surveillance system, they had only 13.1% who has been infected in the past year for influenza-like illness or acute respiratory illness. So it's actually, we actually detected twice more persons who has been infected because they have positive in IgM and IgM and IgA, and we know that those isotypes are fading after the infection has been clear. Also, when we were looking about who is IgM or IgA seropositive for different viruses, we see that the odds ratio to be seropositive to more than 1 viruses is significatively high, and so there is an increased risk of co-positivity if you are already infected by one of the respiratory viruses. And when we were looking about age, there is 2 interesting things. First, we will look about the scatter plot, so the little dots, and there is the age growing, and we see that, so those are only seropositive people, when people are older, they tend to have a very higher AGM or AGA response than the younger groups. And this might be, our hypothesis is maybe it's because of the senescence of the aging of the immune response. And now, when we look about the gray line is how many of persons are seropositive in this specific age group, and for every AGM or AGA in the 3 viruses except AGM for SARS-CoV-2, we see an increase of the seroprevalence between the age groups. So older adults are more susceptible to be AGM and AGA seropositives. Okay, so with all of that, what is the take-home message? First, in a general insight, we observed AGG level saturation after 40 years for influenza and 30 years for RSV because of chronic reinfection. We found highest AGM and AGA seropositive for older adults. Our technique, the EURISA, is very adapted for the high throughput study with a low cost because we were able to do 3,000 samples in 8 days. And in this specific insight about the E difference at this given time, we actually didn't find any immunodepressed patients. At least they had been positive to at least one isotype and one virus, which was a bit surprising, actually. 4.2% were about concerning individuals, and for us, those are the persons at risk that public policies have to take into account to better protect them and prepare the seasonal epidemics. 25.9% were at least AGM or AGA seropositive to at least one viruses, and we found significant co-positivity between the viruses. We also were looking about the department, like the region, because as I told you, like density is a factor in infection, but we found no department differences, and we think it's because of the movement of the population through school, through work, and so it makes it less important for the density as an impact. And finally, we can say that the large multi-simplex serology we did can easily determine the immune profile population and can be adapted to another study. Actually, we did it for vaccine coverage if people were having enough IgG through vaccination in difference, and it's working very well. Okay, so efkaristos a tutti. Yeah, I want to thank everyone who helped us in our study and also the financial support. So Institut Pasteur, Horizon Europe from the EU, Région Île-de-France, BPI France, and also STABA for paying for the trip to allow me to present my talk here. And thank you for listening to me.
Any questions?
Thank you for a wonderful talk. Congratulations to your work. So my question is that did you try to identify the differential serological profile for different strains of the virus?
So we used the nucleoprotein. So it was like very general. For example, like the nucleoprotein of the influenza was the same for each strain. So we don't have difference in like the virulence. Is that what you ask?
Different strands of the same virus?
No. We use only one nucleoprotein for each viruses.
Thank you for the presentation. I have a question about, specifically for the IgGs, how can you differentiate IgGs from infection or from vaccination campaigns? Because especially influenza and COVID, many people have been vaccinated.
So that's one of the limits of the study. So for RSV and COVID, there is no problem because the nucleoprotein is not in the vaccine. But for influenza, the nucleoprotein is in the vaccine. So, like, we know that with the French data that the 65 years old plus age group are like targeting with influenza vaccine. So it's normal so that they have higher IgG against the nucleoprotein. Yeah, that's one of the bias of the study.
Okay, thanks.
Thank you.
Thank you.
Thanks a lot for a very good talk. Okay, let's go to the next talk. Okay. So the next talk is Remigijus Valocha. He's working in the Institute of Pasteur Paris. He's a final year PhD candidate in cell biology in the Institute of Pasteur Paris, focusing on intestinal biology. He has a degree in biotechnology in Italy and collaborating with the World Health Organization on influenza virus surveillance. He has a double master's degree in molecular biology and genetics between Paris and Rome. That's impressive. And during his PhD, he combined organoids and organ-on-chip models to explore the human gut and study host microbiota interactions in healthy and disease conditions. And his talk today is impact of mechanical forces and microbiota metabolites on colon homeostasis and intestinal infections. The floor is yours.
Thank you so much. So I will start my presentation talking about biological model systems. So as you might know already that we have several models that we can use in the lab trying to answer our question. We can have something that is much more close to spectral tractability, so 2D cell culture. We can reach model organisms where we have much more physiological relevance. But actually, what we are doing in the lab is trying to have organism organ on chip. We have, in order that like kind of in the middle of the rest of the models, because we can have quite good reproducibility of the human physiology, but at the same time we are not using animal models, and we have structures that are much more complex than the 2D cell cultures or like cell lines. So actually, to study infection, as I said, we can use organoids or organ-on-chip. We have several pro and cons in both cases. So with the organoids, we can have a 3D topology, liquid-liquid interface, we can have cellular diversity, we have a closed lumen, so this perhaps to have infection can be much more complicated because we have to microinject inside the organoids the pathogen. And we don't have any mechanical simulation. To the other hand, we have organ-on-chip where we can still have 3D topology. We can choose our interface. We can have liquid-liquid interface or air interface. Cell diversity, we have an open lumen. So this is much more similar to what we have in our organs in the human body. So it's easy to infect, and we can have mechanical simulation. So this is just to show that microenvironment is fundamental. We are always studying infections on our cells in the lab, but we are not counting about the complex in which the cells are actually, the mechanical forces that are impacting, and the interfaces in which the organ are. In our lab, we are focused on the intestine. So if you might know, the human gut is quite complex organ. It's like a 3D dynamic and multi-layer organ. We have the epithelium layers, which is made of epithelial cells. We have much more stem cells and prayer cells in the bottom of the crypt. And going up, we have much more specialized cells, such as colonocytes or enterocytes, goblet cells, enteroendocrine cells. Below this one, we have immune cells that are ready to fight any infection that we can have. And above this one, we have the mucus, which is another layer protective, but also linked for like several functions that the gut have. And here we can find the microbiota with specific metabolites that are essential for the human life, let's say. The human intestine is not only involved in the absorption of nutrients, but it's actually like a real barrier. So this is why it's always in contact with pathogens. This is why we have the immune cells downstairs, and the pathogens are always present in the lumen of the gut. To make everything much more complex, we have also two different mechanical forces. The fluid flow, which is created, for example, by the passage of the food, and the peristaltic motion, which is a static stretching constantly applied. And both of them are essential for the function of the human gut. So the aim of my PhD project is understanding of human chronic bioorganization, the impact of gut mechanical forces and microbiota metabolites, such as short-chain fatty acid, on the colon tissue, And we know that the microbiota is related on our lifestyle. So if we have a good lifestyle, so we don't smoke, we don't eat too fat and stuff like that, we might have a good microbiota which is able to produce several metabolites, essential not only for the to have like a healthy intestine, but it's also important for, has also benefit potential effect on different organs of the human body. And the third aim is the impact and the consequences of SARS-CoV-2 infection and inflammation. As you might know, ACE2 is the main receptor, tends to ACE2 actually, the SARS-CoV-2 can enter inside the cells. We know that ACE2 is well expressed in the human lungs, but actually also in the human gut we have huge expression of ACE2. So this perhaps is why first COVID-19 patients experimented a lot of gastrointestinal symptoms such as diarrhea and vomiting. So the experimental plan is starting from human colonic tissue taken from colonic surgeries. We isolate the crypts, so then we isolate the stem cells. We generate human colonic organoids, and then we maturate them in two different devices. The first is the organ-on-chip, trying to have a mechanical simulation. We have polarized cells, and we have a 3D topology. And then we have the filter or transfers. It's just to mimic a static condition where we do not have any mechanical simulation. We still have polarized cells, but we have a 2D topology. And after this, we want to add the short-chain fatty acid for 4 days, and then SARS-CoV-2 for 48 hours. We're using in the lab the emulate organ-on-chip. This is just to show you how the organ-on-chip is looking like. So it is like this little chip of like maybe 2 centimeters. It's a microfluidic chip made of PDMS, so we have 2 separated channels. We can put 2 separate like cell types. And the 2 channels are separated by these porous membranes, so we can still have communication between the 2 cell types. And then we have this specific reservoir where we have the liquid, the media. There are specific pumps that are like pumping the liquid inside the channels. And then close to the channels, we have this vacuum room that can have like negative pressure so they can make the stretch. And this is how the stretch look on our carton chip. So about the mechanical forces and the impact on the colonic tissue, this is how my 2 devices are looking. So in the top view, you can see how the mechanical forces are helping to have a 3D topology on the human gut, while in the transfer, where we do not have any mechanical simulation, actually the whole tissue is really flat. Here perhaps you can appreciate a little better with this video. You can quite see that here in pink you have the cell acid, and what we see in general is that we have an increased cell differentiation, which is going towards the colon. What's really interesting for us was also to try to have this increase of cell differentiation, but interestingly, we are losing stem cells and peripheral cells, but we don't lose the cluster of those ones. So the number of crypts we have in our tissue is still the same, it's just that we have less peripheral cells inside, because the mechanical forces and in combination with the short-chain fatty acid are improving the cell specialization. So we wanted to analyze a little bit further this, so we try with bulk RNA-seq, trying to see what are the gene expression changes inside of our models, and we see that I have a lot of genes that are actually up or down regulated upon SCFA. We're talking about more than 600 genes upregulated upon SCFA, and more or less like 500 genes are downregulated with SCFA. And inside those, we find a lot of stuff linked to metabolism, and in particular to pathways that are like linked to cancer initiation and progression. So we can have the JEC-step linked with interleukin-6, the TNF signaling via NF-kappa-B. We have also the epithelial mesenchymal transition, and also KRAS signaling. At this point, we try to analyze with the public data that we found. We wanted to compare what we have in our gene expression with those public data. Of course, we are comparing something in vitro and something in vivo. In this case, we are comparing colon mucosa from healthy people, from Crohn's disease non-inflamed patients, and from Crohn's disease inflamed patients. So you can clearly see that there is like a distinction between the two, but the tendency is that our non-treated samples are much more close to the inflamed one in Crohn's disease, while the ones treated with the SCFA are much more close to the healthy people. So at this point, I wanted to see how it is going with the infection in our model, to see if we can have the same inflammation that we can reach in the COVID-19 patients, and actually how the mechanical forces and the microbiota metabolites are responding to these specific cues. So first of all, we have infection. So you can really see that in green we have the SARS-CoV-2, which is affecting the cells and disrupting totally the shape of the cells. And we have also like a really good expression of ACE2 here in red. With the electron microscopy, you can clearly see that the cells are like, the microvilli are like totally affected on infected cells, and at the same time here the virus is actually budding out from the apical part of the cells, disrupting the microvilli organization. So we were wondering what is going on with our chip when we treat them before with short-chain fatty acid. So we saw in both cases, in the treated samples, that we have a reduction of the SARS-CoV-2 infection, and the replicability of the virus also reduced by plaque assay. We were wondering why, and analyzing the gene ACE2, we saw that before infection, we have a reduction of its gene expression with SCFA, not only on a gene expression level, but also by FACS on a protein, so on the surface of the cell level. I wanted to say to you more what is going on with the single cell RNA-seq, and we found Dumbo actually. You can see that our shape is quite similar to Dumbo. At least I see a Dumbo. I don't know you can tell me later. So in general, like, we have all of our conditions. We found once again more or less what we saw already by qPCR before. So we have several clusters of colonocytes. We have stem cell, TA, so transamplifying peripheral cells, goblet cells. But what was really interesting for us is that in particular, so in general, all the clusters were going in the same way that we saw already with the qPCR. So we have an increase of colonocyte when we add the SCFA. We have a reduction of the peripheral cells, but what was really interesting for us was this cluster, the cluster 7, which is actually a colonocyte cluster and is the only one colonocyte reduced upon SCFA, which was weird for us. And actually analyzing, we saw that this cluster is the one who is expressing the most ACE2. We are doing other studies about this, trying to link the fact that we have an overexpression of ACE2 because those cells are actually inflamed cells. So that's why with the SCFA we have the reduction, but we're still working on it, so I'm not showing you anything. But going to the infection part, we can clearly see that we have two clusters that are appearing with infection. And those actually have two different behaviors. We have the cluster 8, which actually is reduced upon SCFA, and the cluster 10 will actually stays the same. And if we want to analyze and to be further this cluster, in the cluster 8 we have all the SARS-CoV-2 related genes, so all the envelope spike and the different ORF that we can find. We have a lot of inflammatory genes. But actually the cluster 10 is totally different. We cannot find any trace of the virus. And we actually see a lot of, with this gene ontology, a lot of pathway activated linked to the reply, to an answer, an immunologic answer to the virus. So you can see there are like several cellular like response of virus, regulation of interferon, innate immunologic response, NF-kappa B activation, so several of these. And we find a lot of antiviral genes in this specific cluster. Moving even further, we wanted to see what is going on with this specific inflammatory panel, is like another kind of bulk RNA-seq. And we see once again the same behavior that we saw with the single cell RNA-seq. So we clearly see that we have in our infected samples by SARS-CoV-2 an increase in all of those inflammatory genes. But actually, when we treat them with SFA, we have a reduction of all of them. They're really able to turn off, and it's kind of firefighters with like a house on fire. But then we have also the second group of cells where actually are all antiviral genes, And what we can see is that even if we treat them with SCFA, they're just modulated, but they're not turned off. So we can actually think that, once again, like firefighters, they're not only trying to turn off the fire, but also to save what they can save in our model. And now we are concluding some ELISA tests, trying to see that we do not have only gene expression actually, but it's also like going in the same direction in our protein level. So we just have like for the moment EL8 and MCP1, and we can clearly see that with the SCFA we're going down about the production in both cases for MCP1 and EL8. So just to conclude, I want to show you that organ-on-chip are able to recapitulate all cues present in the human intestine. Mechanical forces are fundamental for the colonic barrier development and organization, so we can have peripheral cells, stem cells, but still specialized cells. The SCFA's absence is inducing an inflamed phenotype in the gut, which is what we see similarly in IBD patients. SARS-CoV-2 can infect intestinal cells, has a strong impact of tissue organization and inflammation, as Dumbo. Microbiota metabolites, SCFA, are key for tissue homeostasis by affecting gene expression, but at the same time they are able to reduce SARS-CoV-2 invasion and inflammation, decrease inflammatory gene expression, and increasing the antiviral defense at the same time. So, like a firefighter dog. Thank you so much.
Thank you, Remy, for this very exciting talk.
Sorry, I forgot just to say thank you to my lab.
If I could just put the
you
first
the
question.
Yeah.
Yes. So organoids from gut tissue, like stomach or intestine, are quite challenging, aren't they?
Definitely, yes.
At least if you compare them to other organs that are more sterile. So I'm always very aware that you could have artifacts because you have to use an awful lot of antibiotics to start with to isolate your stem cells and grow them.
Yeah.
So do you have an idea about this and how antibiotics can affect your results, or have you seen a certain effect of that?
So to be honest, we haven't really checked it because our protocol is linking a lot about the chelation. So when we have, before starting to use organoids for our experiments, we have several passages to be sure that we don't have any bacteria or any other stuff that are like still in our cells. So we haven't checked like before and after, to be honest, but in our culture we do not have any staining of bacteria of like debris, let's say from from the patients. And we keep them in culture and everything, and we test regularly for like mycology tests and whatever, and they're always negative. So for the moment, we haven't seen this any impact about that.
Mimika, then Spiros.
Thank you very much. Very interesting work and methodology as well. I'm out of the microbiology field, but I have a more general question. Given the significance of short chain fatty acids, are there any efforts toward clinical trials towards increasing the levels of these metabolites, or any drugs or any?
So yeah, for sure there are, and in general we know that probiotics are already like available in commerce, so they can have beneficial effect in general. But the point is that it's always like a sort of prophylaxis, let's say. So we do have already like our microbiota, which is quite good. The point is just to taking care about it. So I mean, I'm the worst one because I'm smoking. I'm eating always McDonald's, so voila, that's it. But I should be here to say yes, so we should take care of our microbiota too. There are several trials here where they're trying to use these as like therapy. Sometimes it works, sometimes not, because of course it's always depending on the lifestyle of the person. It can really be variable between person to person. So for the moment in our organ on chip, for example, we do have several patients from which we are like using organoids, and we do see some differences. So in general, like we can see the tendency is the same, but of course the differences, let's say, between the gene expression of like the ELISA test, stuff like that, they can like be really variable. So I cannot answer you more about this. I don't really know really about any specific trial that I'm doing at the moment.
I was thinking of a more specific drug or something.
Yeah, no. For the moment, at least what I know, it's no. Maybe I will do it. Maybe we can do it. I don't know. Yeah, I don't know. Sorry.
You write in your last sentence there, SARS-CoV-2 invasion. What is the difference between invasion and infection?
I mean, with invasion, we just want to specify the way that he's entering. Since we are like also interested to see at ACE2 level and stuff like that, with like conducting other experiments in parallel. So with invasion, we really want to see the uptake of the virus. So of course, we can say infection is always infection. Invasion and infection can be correlated. But with infection, we can also have all the address of the process inside the cell. So in our case, we specify invasion because we are really looking at the first step of the infection, the way that SARS-CoV-2 is entering. So with the SCFA, we have less ACE2 expressed, so you have less uptake of the virus, so the invasion is reduced.
Okay, thank you.
Can I just use the privilege of the chairman to ask a final question? In equivalent work that is being done with bacteria, okay, there seems to be a tropism where bacteria target LGR5 positive cells, so stem cells. So do you see anything like that with viruses or with
Yeah, so actually it was quite complicated for us to, the cluster 8 and 10, they're like the infected ones, it was really complicated for us to identify especially the 8, since there is expressing mostly viral genes, but we found some markers for colonocytes. So we are quite sure that there are colonocytes. And overall, ACE2 is much more expressing colonocytes compared to the other cells that we have in the gut, so 99% there are colonocytes. So we can see this tropism. Then, as you know, the human gut is changing these cells every 2 days. It's really dynamic, so we cannot be sure like 100%, but this is what we see from our results.
Indeed, thank you very much.
Thank you.
So, we move on to the final speaker, which is Evangelias Vestopoulou from the University of Siena. Evangelia is a molecular biologist and geneticist. She's a Greek student in the University of Siena at the laboratory of my really dear friend and collaborator, Professor Mario Milko-Delios. He's quite a renowned immunologist, Mario. So Evangelia is working on Helicobacter pylori and T-cell responses in autoimmune gastritis and Helicobacter pylori infection, and she's working in a very immunologically versed lab, but she came in our lab for the next 6 months to learn a little bit of microbiology and how to isolate the bacteria and characterize them, so do the work from the bacteria sort of side, having completed one part of her work from immunology. Evangelia's title is T-cell responses in autoimmune gastritis and Helicobacter pylori infection. Evangelia, the floor is yours.
Thank you so much. So I am Evangelia, good afternoon, and today I will present my work on T-cell responses in autoimmune gastritis and Helicobacter pylori, a study which is from the University of Siena with my professor Mario Milko-Delios. Autoimmune gastritis is a chronic inflammatory disease where the immune system destroys gastric parenchymal cells. The main autoantigens, HK-ATPase, which produce gastric acid in the stomach, and the intrinsic factor, which is crucial for vitamin B12 absorption and is associated with acid-producing cells. As shown in the immunological pathway figure, antigen-presenting cells present gastric self-antigen to CD4 T cells. Instead of remaining tolerant, some become autoreactive and polarized, mainly into Th1, producing interferon gamma, and some in Th17, producing interferon 17, that drive inflammation and tissue damage. This condition leads to vitamin B12, pernicious anemia, which is a condition where the body can't make enough healthy red blood cells, and eventually gastric atrophy, which is the loss of acidic tissue in the stomach. On the other hand, Helicobacter pylori infection is a persistent gastric inflammation, which is usually obtained in early childhood between 3 to 5 years old, and if it's left untreated, it becomes a chronic infection. Helicobacter pylori infection is the etiological cause of chronic active gastritis and peptic ulcers, and it's the major risk factor for the development of gastric cancer. It triggers both innate and adaptive immune responses, which are responsible for higher T-cell invasion of the lamina propria and neutrophilic infiltration. As illustrated here, dendritic cells present bacterial antigens to CD4 cells, leading to Th1 and Th17 responses. In some patients, we also observe Th2 responses, which produce interleukin-4,
which may support bacterial persistence and immune tolerance. Despite strong immune response, the infection persists, so there are strong immune evasion mechanisms. But in real life, the problem is that it's really difficult to discrete these two gastritis in the clinic. So the aim of our study is to address how autoimmune gastritis relates to Helicobacter pylori infection, which is an infection-driven immune condition. Secondly, to identify distinct T-cell profiles induction associated with these two conditions. And thirdly, to characterize putative Helicobacter pylori virulence factors which act as a trigger or as amplifier of autoimmune responses. So our hypothesis is that co-occurrence of autoimmune gastritis and Helicobacter pylori infection broadens T-cell specificity, reshaping gastric immunity and possibly contributing to autoimmunity or even cancer. To answer all these questions, I have analyzed antral and fundic gastric biopsies and divide the patients in 3 groups with the autoimmune gastritis in the absence and in the presence of Helicobacter pylori, and the third group with Helicobacter pylori only. Firstly, we detect the IgG, the Helicobacter pylori IgG in the plasma. Then from blood samples, we isolated PBMCs, which are used as antigen-presenting cells. And from gastric biopsies, we expand T cells in interleukin-2, which helps them to expand the in vivo activated T cells. We then performed proliferation assays with HK-ATPase, intrinsic factor, and Helicobacter pylori lysate. And finally, cytokine production was analyzed by ELISA, focusing on interferon gamma, IL-17, and IL-4. Here we see the levels of antigens on each group. First, we have the first group with autoimmune gastritis only. We can see production. Wait a minute. Here, we can see production of HK-ATPase and intrinsic factors. Then we have the patients with autoimmune gastritis and Helicobacter pylori, and there is proliferation in both antigens, HK-ATPase and intrinsic factor, and also to Helicobacter pylori. And here is the last group with Helicobacter pylori only. We can see only the proliferative responses only to Helicobacter pylori. Here, even if we did the measurements on the antigens, both of them, we didn't observe any expression. Moving on the cytokine profile, on the first group, we see their cytokine profile showed production of interferon gamma and IL-17. On the second group, with Helicobacter pylori patients with autoimmune gastritis, we can see production of interferon gamma and IL-17 again, but not IL-4. And on the last group, with Helicobacter pylori infected patients without autoimmune gastritis, we can see production of interferon gamma, IL-17, and surprisingly, there is also production of IL-4. To summarize all this, in patients with autoimmune gastritis with no Helicobacter infection, gastric T cells specific for HK-ATPase and intrinsic factor produce both interferon gamma and IL-17, but not IL-4. In Helicobacter pylori infected patients with autoimmune gastritis, gastric T cells specific for HK-ATPase, intrinsic factor, and Helicobacter pylori produce both interferon gamma and IL-17 again, but not IL-4. And on the last group, Helicobacter pylori infected only patients with no autoimmune gastritis, gastric T cells specific for Helicobacter pylori produce interferon gamma, IL-17, and surprisingly, IL-4. This Th2 response may reflect bacterial strategies to manipulate the immune system, promoting persistence rather than clearance. And for the next period, I will be on the medical microbiology lab, and I plan to isolate respective clinical Helicobacter pylori strains and characterize individual virulence factor profile with respect to presence and expression of the CAG-A, VAG-A, and BAB-A genes, which are all contributing to persistence and disease severity, and to associate bacterial virulence factor profiles to the respective immunophenotypes of gastritis in pediatric and adult symptomatic patients. I would like to thank my group in Siena and my new group here that I can't wait to work with, and all the hospitals that collaborate with us for this. And thank you all for your attention.
Okay, thank you very much, Evangelia. Questions, please. Spiro, ευχαριστώ.
I've reached the conclusion that the two disorders actually do not interact with each other, or they do not affect one, the progress of the other. Is that correct? Do you reach the same conclusion?
We are trying to figure out if the co-occurrence triggers the immunity in both of them. This is what we are trying to see. And also we will have more patients in the next year, so we are hoping that our results are more accurate next year.
Well, okay, fine. In the interest of time, thank you very much, Evangelia. We should move on. And it's my... so, do I continue here? Okay. Fine, so it's my great privilege, and we're very happy to have here with us Sotiris Misailidis from Paris. So I met Sotiris last year in the annual meeting of the Pasteur Network down in Brazil, where I realized that there was a Greek having spent a good part of his life working at the Fiocruz Institute and Biomanquinhos, which is quite a famous place institute in Brazil. And now Sotiris is in Paris. So Sotiris is head of the Vaccine Innovation Development of the Institut Pasteur in Paris. So he's managing the vaccine portfolio for the Institut Pasteur and leading the innovation development of vaccine projects from basic research to clinical development, spin-off facilities and industrial partnerships for vaccine-related projects. So he joined the Pasteur Institute in 2024, having previously, as I said, been working for 7 at least years as Director of Technological Development and Innovation at the Research, Development and Innovation of Vaccines, Biologics and Diagnostics at Biomanquinhos, the Institute of Technology for Immunobiologic at the Fiocruz in Brazil. Sotiris is a biologist with an MSc and doctorate in chemistry from the University of York and postdoctoral studies in pharmaceutical sciences in Nottingham. And then he moved to Cambridge in the UK. He's been an academic member of the staff in Open University in UK at the Universities of Lisbon, visiting France, and also the two basic big universities in Rio de Janeiro, the Federal and the State University. And he has been the founder and the director of two smaller companies that I'm all to listen to one day, Zoia and Naxos DNA Services. He brings more than 30 years of experience in applied research focused on the development of vaccines, and I'm really looking forward to listening to his talk. Thank you very much for being with us, Sotiri.
Thank you very much. Thank you for the invitation. It's a pleasure to be here. I've been here a couple of years ago on a different setting. So Sotiri is just restless, I guess. So I was in one place, I was here, I went to England, I went to Brazil, now I'm in France. And I've been to academia, to industry, sort of startup companies. And now I'm working on the vaccine development at Paris. And yesterday I changed my talk because after I looked at the presentation, I decided I would mess with it from the last moment. So please excuse me if it doesn't make sense. Okay, here is Edward Jenner doing his first vaccination on smallpox at 1796, and about 100 years later, Pasteur did the first vaccination on the anti-rabies vaccine. And apart from that, Pasteur was a great innovator. He did vaccine, but he produced wine, beer, yes. So, and Pasteur Institute was a great success of a startup a long time ago. You can say that. So, looking at the presentation, there were a lot of people presenting basic science, a lot of people presenting interesting molecules, concepts that could be developed into future therapies, small molecules, vaccines, diagnostics. So I, innovate maybe for some of you the way forward. And there are many good reasons why you could. Maybe you want to save the world, maybe you want to make money, maybe you want to see what you're doing becoming a product. And in part is what my work at Pasteur Paris is nowadays. So a lot of inventions that you do in the lab remain unexploited because there is insufficient validation, because there is insufficient data to mature this project and take it into an industrial partner for further development. And a lot of time there is intellectual property missing, there are supporting evidence. So an invention is not an asset. And a lot of the times I started there, the first time I got my first antibody or my first antigen, I thought I had a product. And I didn't, I still don't have one. So an invention is not an asset. You need several years of work from invention with high potential to become applied research towards a solid patent, and then it has to pass what we call the valley of death. And there it needs maturation and acceleration to go into either licensing or a startup creation, depending on your target. And well, this is the valley of death, is where basic scientific research, specifically on drug discovery, vaccine discovery, focused at the university stage, stops because it doesn't know how to cross on the development side and reach clinical trials. And we try to do that at Pasteur. We have a team dedicated specifically for getting the research discoveries of about 2,500 scientists into developing an invention, patenting it, and then help it through the acceleration and maturation phase to be ready either to be exploited by the researchers when one has a desire to become an inventor and has its own startup, or licensing it out. and about a third of the income of the Institut Pasteur was coming out of licensing fees. So we are good at that, and and we are also creating on average 1.5 startups a year, which means some years we have 2, 3 startups, others 1. So the last 20 years we have 1.5 on average per year. Again, we have the different offices for the detection, for the patents and invention office, the development and innovation office where I work, and then the tech transfer. Pasteur doesn't have production, and I will come to that later, but Pasteur in Paris doesn't have production. So we only have 2 ways out. Either we license to the industry, or we have a startup creation. In my job before at Biomanguinhos, we had production, so we were taking products from early stage all the way to producing them, to registering them, producing them, and distributing for the Brazilian public health market. So a lot of the projects I started there actually went to the market and were licensed or were produced and distributed in the local market. At Pasteur we don't do that, so we need to work with partners for that part of the development. I said about the startups, and we are all experts with experience on having taken products from the bench to the market, and that helps and is there to help you guys develop your products. So I saw various things that I thought very interesting, and I'll be here to talk to anybody who wants. So you may have great ideas, concepts, innovative pathways, novel mechanism of action, or in vitro data for target activity or cellular activity, but you may not have reference compounds, you don't know the specificity, the selectivity, the plasmatic stability, or have some in vivo data, but still miss reference compounds, comparison with other products, what's your market. Part of the maturation acceleration step includes building a target product profile, a TPP, identify your deliverables, have a flow chart, supporting studies, and they're different. I will talk a little bit briefly about small molecules and diagnostics, only a couple of slides each. I will talk a lot about vaccines, just because I like them more, or I work on them nowadays. So you need to have a development plan and a financial plan. That differs a little bit from the academic way, because once you commit to developing something, you can't follow every possible way that is interesting scientifically, but it takes you off the development path. So once you decide, you sort of stick with it, but with go, no go. If it doesn't go, it doesn't go. So for small molecules, you need other kinds of de-risking steps. So there were a few posters on small molecules. Having a small molecule that has an activity doesn't make it already a druggable product. Because you have to think of other things like absorption, distribution, metabolism, and excretion, the half-life of the product, so the pharmacokinetics, the pharmacodynamics. Usually your molecule is not your first, is not what will go to the market, so you have a hit to lead, and then you optimize the lead, and then you have a candidate selection. That can even work for biological molecules. Sometimes we develop antibodies, and the first antibody we generate, we get out of a mouse, is not good enough. So you need to improve it with mutagenesis, you need to increase the affinity, or you need to conjugate, or you need to do any number of things before you have a lead compound or a candidate. This is a development plan, for example, for an antibody. It may need humanization if it's from an animal model, if it's from a mouse or a rabbit, but It may not. Nowadays, there are human antibodies directly. You have the pharmacology. Then you have to do toxicity studies, and talking about vaccines is the same thing. You need to show that it's not toxic. And then you have to be able to produce it under GMP conditions. You have to have a stable clone that produces it at high yield to be able to be financially viable when you go to the market. And then after that, you go to clinical development. Yeah, that's right. Diagnostic is also another very important part, and you may have either an antigen or an antibody that you can use as a therapeutic. An antibody can be a therapeutic, but an antibody on a diagnostic test can be used for antigen detection. SARS-CoV-2, for example, had the first thing they did was to have the antigen of SARS-CoV-2 to detect antibodies, and then when they had antibodies, were the antibodies on the chip to detect the antigen. So anything can be used on a diagnostic, and without diagnostic, medicine is blind. We need to know what we treat, and sometimes many things have similar symptoms, so diagnostic is crucial. And the diagnostic development is different from the other things, from the biologics and the vaccines. It's something that doesn't go into the human being, so it's faster. You don't need clinical trials. What you do need, first of all, some of the things are mainly academia on the diagnostic world, especially the research on early biomarkers, new antigens or antibody detection. And then you have a part that is mainly companies, but it's a blend between the two. And then you have the part that is industrialized. Some companies, obviously, from the diagnostic industry, do the whole part. But usually when you start in the academia or in Pasteur Institute, you work mostly on the earlier part, and what you can help is to do that translation to the later. And here you need to know what your user need is, what your intended use, why is this diagnostic necessary, and what's your target product profile again. And your analytical performance, because it has to pass the regulatory factors for sensitivity, specificity, accuracy. A diagnostic that is 60% accurate is not a diagnostic. You can flip a coin. And also the clinical performance of that, the ease of use, and so on. Now in vaccines, well, you may want to develop vaccines instead. To develop vaccines, you need to consider a number of things, like the epidemiological impact, determined by the disease and its strength. Vaccines for something that already exists, a therapy, or that affects half a dozen people in a particular place of the world, is not usually going to become a vaccine that somebody will invest millions on the development and will not have a market afterwards. So the existence of alternatives, whether for treating or for prevention, the cost benefit, and in the case of orphan diseases, economic issue is not or should not be the dominating decision. The technological, scientific knowledge of the disease, the analysis of the complexity of the selected topic, the gaps in the vaccine development, who is doing what and why, what you have in terms of laboratory for preclinical studies, for GMP standards, the production of the vaccine. Can you produce it here? Do you have the facilities? Do you need to find partners? Do you have the money to contract? Regulatory experts. It's a crucial part, and if you develop vaccines, it's good to have that from early on, because it guides your development, and sometimes when you don't, you have to go back a lot of steps to think of all the things that were necessary to have been done. financing sources, and what is your organizational and administrative capacity? There are many types of vaccines. There can be inactivated or attenuated vaccines. There were the earlier types. You had the virus and you inactivated or attenuated. Then it can be based on the type. Is it a viral vaccine? Is it a bacterial? Is it a parasite? Nucleic acid, recombinant protein, and is it a combination? Is it a conjugated vaccine? Are you conjugating it with a protein so that you elicit a better immune response, or you use an adjuvant? And what's your aim? Is it prophylactic? Is it therapeutic vaccine? Is it tolerogenic vaccine? We heard earlier about tolerogenic molecule. Is it liquid, suspension, lyophilized, injectable, oral, nasal spray? And that induces also different types of antibodies. So a nasal vaccine induces IgA, but the other ones don't. And what's your presentation? And it takes a long time to develop a vaccine. It takes 10, 15 years, and not all of them make it to the market. And that depends on regulatory factors, biological factors, corporate factors, financial. So you start from target discovery and proof of concept, where you identify your target. Is it an antigen? Is it an attenuated molecule? The modification, activation, your constructs. Then you go to the preclinical development, where you test that in animals. And you do toxicology study as well to show, first of all, that there's no toxic in animals. Then you need to show that you have to have the definition of your analytical methodology, how stable it is, and produce your first batch. So after your GMP batch, you have a folder that you submit for clinical trial development. And then it's clinical development phase 1, phase 2, phase 3. Phase 1, you only usually show that it's safe and immunogenic, but then you have to show, you have to produce in bigger lots, and you have to show that it's efficient as a vaccine, and then you do an efficacy study usually on the phase 3 with a target population, big numbers of people, and so there is a development chain from the proof of mechanism and proof of principle with experimental development, the clinical studies, and then the registration. And you start with low knowledge, high risk, and low cost. In the beginning, it costs a few tens of thousands, perhaps, and then you go to millions, hundreds of millions in the clinical development. But then, as you go to progress through the clinical development, your risk goes down. So a lot of companies will buy a product when you've already done tox studies, but at risk and at a lower price. If you've gone to clinic, then the price increases and the risk goes down. If you have phase 2 data, then the chance that it would fail on phase 3 are minimal, and so your product is a safe bet, and a lot of companies will enter and buy it at that stage. It depends. So in each of them, I think I will, for the sake of time, I will pass the details of that, but each of them has requirements and it has outputs. So you have your target product profile already early on. You decide what you want to do. Then you have quality target profile on the proof of concept. You start thinking about a material tree, what you will need. And then finally, you have your clinical trial reports, and then you do your batch production, process validation, documentation, and the regulatory authorities' submission and approval. And it depends very much on how much it will take you to do that. At COVID, we did it at 250 something days, and it was record, and everybody put forces for that to be possible, because if If you needed 10 years to do the development, then nobody will have a vaccine. But for other vaccines, it's taken many years and it has not gone anywhere. Others after 60 years, for example, polio, we have vaccines, but we still develop new ones. For SARS and MERS, we never got a vaccine, but that's because the cases dropped. So we had started vaccine development, which subsequently was used for SARS-CoV-2, actually, the knowledge we gained with that, but we never managed to get a vaccine because the cases dropped and there was no way to do a clinical trial, there was no way of testing. I'll show you a bit about that with a Zika case as well. So traditionally you do step by step. You complete your first step, then you go to the next, then you do one animal model, then you go on non-human primates, then you do your tox studies, then you go to clinical, 1, 2, 3, and so on. And similarly, you start producing at phase 1, and then you do large-scale production only after registration. We know your product is ready. At COVID, everything happened, but everything happened in parallel. So you wouldn't wait for one thing to happen to do the next one. People started optimizing already at phase 1. They started large-scale manufacturing in the middle of phase 2 trials because they needed to have the product ready as soon as the results were ready, even at risk. And that's why a lot of governments bought 5 different vaccines, and they thought if one of them works okay, we use it. If the other fails, we throw away what we bought. It doesn't matter. The important thing is to be able to vaccinate the people. We came almost back to where we were. Unfortunately, I had hoped that we would be somewhere in the middle, but we're not. We're almost to where we were. As money dropped and as the emergencies disappeared, we still do things only almost in series. But so different types of vaccines and the nucleic acids, inactivated viral vector, subunit. That was only for, well, in the case it was COVID, but it can be for any viral vaccine. But at the same time, there are polysaccharide vaccines for bacteria that express polysaccharides in their surface. And some examples are pneumococcal, meningococcal, Shigella, Cinetobacter, and so on. And many of them, the polysaccharide is conjugated to a toxin, like tetanus toxoid or diphtheria toxoid, and that increases the immunogenicity. It gives better antibody response. Some of them are based on synthetic peptides, and here's an example of a protein for dengue that is small peptides on a gold nanoparticle, but there are also others on LMPs, on scaffolds where the peptides are expressed on these parts. So it depends how you want to present it. Short peptides by themselves are not immunogenic, so they don't make a good vaccine, and they very rapidly degrade it as well. So you need some way to protect them and to present. And this is a short story of a vaccine that started in 1937. It was in France. It was transferred to Biomanguinhos in Brazil in '76, some 40 years later. And then it's still in production there. It's an egg-based vaccine. It's produced in embryonated eggs. They're infected, and then the egg with embryo is removed and is used to produce the vaccine. And that same technology is also used for MMR vaccine, the measles, mumps, and rubella. We were producing that there, and even all this time, 90 years later, there are still studies about this vaccine, and there are still studies for new yellow fever vaccine development. This is a vaccine that is used in more than 75 countries, and Biomanguinhos is the biggest producer. Institut Pasteur Dakar is one of the producers. So two of the members of the Pasteur network are producing this vaccine nowadays. So it depends also what you want to achieve. Do you want a preventive vaccine? Do you want to prevent infection? We didn't have, for example, for SARS-CoV-2, we didn't have any vaccine that prevented infection. We were trying to prevent mostly severe disease, but if possible, disease. And then there's also, well, you may want to prevent death. It's sufficient enough. Or prevent transmission. And that depends how you tailor your vaccine, where you want to aim it. There comes the target product profile, and that defines goes and no-goes. And it usually says what's desired, what you would like in an ideal world to have, and what's the acceptable. So yes, we would like for the COVID vaccine to inhibit transmission and to be sterile immunity and so on and so forth. But if it avoided hospitalization, it was okay. Similar thing was with the Zika challenge, and I took the Zika just because we've heard too much about SARS-CoV-2 in recent years. Zika doesn't have a vaccine to date because Zika cases dropped. So when in 2016 there was a Zika epidemic, everybody started doing Zika vaccines, and so did we. And we had to decide which platform, what type of vaccine, which animal model, what was best to use, would be cross-protection with other flavivirus vaccines, would be antibody enhancement like was with dengue, what happens? And WHO published a TPP saying that they wanted it ideal for pregnant women to avoid microcephaly. Because 70–80% of the cases of the other individuals were were asymptomatic. So the important thing was that a vaccine that could be used in pregnant women. But failing that, they said any vaccine that could be used in the age of 9 to 14 would be okay, because then it would be, you could vaccinate girls early on to avoid possible effects. So we started with 4 different products. One was internal ours, one was collaboration with Protein Science, one was collaboration with another institute, and one was with the University of Texas. One of the problems, for example, for Pasteur Institute with SARS-CoV-2 was that they, if I'm allowed to say that, they put all their eggs in one basket. They were the fastest public institution to get a vaccine to phase 1, not competing with companies, but within academia and research institutes, they got a vaccine to phase 1 very quickly. But they had only one vaccine, and when the phase 1 failed, they had no other candidates, so there was a stop. So to avoid that, we had started with 4 different ones. We tested the 4 different platforms. One was inactivated, aiming at pregnant women, but knowing that inactivated had less immunogenicity. One was live attenuated by mutations, and another one was using the yellow fever vaccine, because we produced yellow fever, we knew everything about yellow fever, so we could manipulate it. It was a flavivirus, So we could take out the yellow fever proteins, the PRM and the envelope, and substitute them with a Zika to have a viral vector vaccine. This is another construct of the same methodology. And then other companies started Zika vaccines. Recently, Moderna had a Zika vaccine that they stopped the development. The Institute Pasteur produced lentivirus Zika, measles-based Zika, yellow fever Zika, Oxford produced an adenovirus Zika, and all of them stopped the development. Because the moment the Zika epidemic went down, there was no interest, financial, for the companies, and also there was no sufficient number of targets to do an efficacy study. And so the Zika vaccine development stopped, but we remain at the point where we are unprotected if or when Zika returns, because at the time of the epidemic, there was sufficient immunity because of people were infected to have a drop in cases, but as the new generations are coming, that immunity will not be there anymore. So that brings me to a point of something that CEPIC was trying to do with a 100-day vaccine development. They had a proposal of creating a bank of vaccines where they've been at least to phase 1. So you have vaccines that are not ready, but you have shown that they are safe and immunogenic. And then if everybody would accept to put them there, in that library, in that bank of vaccines, then when cases appeared, we could quickly draw them out to develop. It's not going forward as they had hoped, because not many people were happy to develop and put their vaccines in that library. And that brings me to the Pasteur Network. And we've heard yesterday a presentation about the network, so that saves me some of the time. But it's a great network for biomedical research, public health, training, career development, and innovation and technological expertise. I will add to that another initiative that we've been working on. But one important thing is that the Pasteur Network is present where the epidemics have been. So if you look at the map, wherever you have recent outbreaks, there is locations of the Pasteur Institute. So that makes for a great environment to have epidemiologic studies, to follow where the new vaccines appear, the new viruses, the new infections appear, the new outbreaks, and also ideally to have development and production. And here we have the various research and training activities for public health challenges. But what we started recently doing, and the Institute Pasteur of Athens and the Institute Pasteur Hellenic, rather, and the Institute Pasteur of Paris are actively participating, is the vaccine R&D and manufacturing initiative. So the idea there was to look, okay, we have sufficient research capacity, but within the network, there are 7 or 8 manufacturers as well. So it would make for an ideal environment where we could start from early discovery, from antigen discovery or an antibody, and take it all the way to production within the network without having to, ideally in a collaborative and cheaper way that can help us do the whole development chain that I showed earlier in a shorter amount of time. So these are the institutes. Some of them, for example, Dakar, Morocco, Fiocruz, Algeria, Tunis, Hellenic Pasteur have production capacity, Iran as well. Others have clinical trial capacity, others have early research and good research services that can offer to the network, preclinical studies. So ideally we create an environment where we can take a project that is of interest and we can bring it all the way to conclusion and to production. And if we could do that with the participation or the collaboration of different members and perhaps share the production, so share the IP, share the production, that would create an environment where we could develop, because let's face it, none of us independently develops projects all the way to the end. Like I said, at Pasteur we license it out and we hope that the Sanofi or whoever of life will develop it to a product, or we span out. But we cannot take it all the way to a conclusion, and even the ones that can, like Fiocruz, they've done very successfully for diagnostics, that the necessity is bigger and the requirements are less, but they haven't got a vaccine or a biologic that have taken to the market by themselves. So as a collaborative, we think we can. And within Pasteur, we've established a Center for Vaccinology and Immunotherapy to bring together all the researchers and the facilities that work with vaccines. And that can be a part of that great collaborative. So we have the research, the platforms, product development, and the clinical development up to the point where we can. And some of the different targets that we work, HIV, Shigella, Lassa fever, malaria, chikungunya, the plague, dengue, and many others. We have some 15 or 20 different products in our portfolio that may be of interest. And the other thing that aims to do a very similar thing, but at a European level, is the European Vaccine Hub. That brings, it's funded by HERA, by the European community, and it brought the best vaccine centers in Europe to make sure that Europe is independent and make sure that Europe can produce products. If you ask me, I will say that we have better chance with the Pasteur network than with that. Even though perhaps they have better groups and perhaps they have more money, but they have greater competition between them and they have bigger egos. And it makes a difference if it's going to be the German vaccine or the French vaccine or the Italian vaccine that will be developed at the end. So the idea, when we put it on paper, it was perfect because Italy would lead the preclinical, Italy would lead the early development, Pasteur would lead the preclinical in Paris, Belgium would lead the clinical trial, and Germany would lead the production. But you can't tell the German guy that is developing a vaccine that he can't take his because that part is the Italians. And you can't tell that to the Pasteurists used in France and anybody else. So the dynamics may be more difficult than they are when we discuss that at Pasteur network level. But in theory, the objectives are to make Europe independent and capable of taking a product from early development to production. And the pillars are exactly that: discovery of new vaccines and antibodies, preclinical studies, clinical studies, manufacturing, and regulatory. And they're split, they're led by different groups. And we hope that the other problem with that is only interested about vaccines that are affecting Europe. So anything that may affect Asia, Africa, Latin America, we don't care. That's their problem. At Pasteur Network, we have 35 members in all continents, so anything may be of interest. And that brings me to the suggestions for regional self-sufficiency, especially for non-high value commercial products. Everybody wants an influenza vaccine, or H5N1 or H1N1. Sanofi has influenza vaccines, but other members want to have, and so on. But for non-high value commercial products, partnerships and joint networks are critical as a development strategy. And you have, depending on the production challenges, working in collaboration and using the better skills that each one can bring to that without duplicating efforts and creating a supply chain between the institutes is critical for that vaccine development. And finally, the introduction of new health products must consider the public demands with a broad perspective, increasing and aiming for equity, social responsibility in other countries, especially now that in some parts of the world there is what I heard in a talk of an orange elephant in the room, who thinks that vaccines are not necessary, and a lot of research at NIH, for example, is dropping off. We have a responsibility for looking at equity and social responsibility in other countries and bring solutions perhaps from our side. So for a health vaccine ecosystem, and that's my last slide, we need R&D, we need strong R&D, we need manufacturing, and after COVID everybody was looking at manufacturing, and we need to put a lot of manufacturing plants in Africa and Latin America. Every country has to have a manufacturing plant. Great, okay, perhaps, but if you don't have R&D, there's nothing that goes into that manufacturing plant, because then you still depend on Pfizer, on GSK, or Moderna to do a tech transfer to you for you to produce. And if they say they don't do a tech transfer, then you produce nothing. Or if they say that you do a fill and finish for them and you send it to where they want, as it happened with many companies in Africa, they produced in Africa, well, they did the fill and finish at least, but they sent it to Europe or the US. They didn't use it in Africa. So if you don't have R&D, you don't have the chain, you don't have necessarily what to produce. You need to have a procurement and pricing system. The country has to guarantee that we'll buy it from you if you produce. For example, there were cases in South Africa, one example, that the company produced the vaccine, but the government didn't buy it from them because they bought it cheaper from India, I don't know. So the company spent millions and then they dumped their own production. And you have to have distribution and logistics, supply chain. Supply chain is crucial. For example, in Brazil, we started producing a COVID vaccine 6 months in the pandemic, but we needed material from India. We needed a buffer. So it didn't matter that we were producing the vaccine. Without the buffer, we couldn't send it out to anybody. So you need to have a supply chain. And finally, you have user acceptance. Somebody will have to accept to put their arm out and take the vaccine. Otherwise, we develop for nothing. So it's from one stage to the other. It needs to be a consistent supply to achieve your goal. I think that's it from me. Thank you very much, and I'm happy to get any questions.
Well, thank you very, very much, Sotiri, for this very nice talk. Very eye-opening, I would say, to most of us who are scientists, who do the basic research, and we don't realize how many steps, really, and some of them are very crucial, it takes to actually do the leap from the laboratory to the manufacturing level of things. And then things get really difficult and ugly, and it's a different world out there. So I don't want to monopolize. Questions, please. Start with Pelli. Wait for the microphone.
Thank you so much for this informative presentation. So you talked a lot about human vaccines against human viruses. I would like to ask if there is a market out there, or in your vast experience, about vaccines that target animals that spread these viruses, so for anthropozoonosis, and how to stop transmission.
No, definitely. First of all, there is the veterinary market, and there is an enormous market. In Brazil, the cattle were a much bigger number than the humans, and the farmers were prepared to pay bigger value for a vaccine than for a human vaccine. So the market is vast. And then you have One Health. The concept that things pass from the animal and infect humans, and in this case, anything, well, if you want to prevent a disease that can affect human, vaccinating the animal may be definitely one solution. And there is the veterinary market for companion animals as well, which is also big, and there the prices are also much higher. So definitely a lot of development of vaccines for that market.
Thank you. I have a question concerning the SARS-CoV-2 vaccine. What were the factors which contributed to its speedy development within 6 months? I mean, as you told us, it takes usually 10 to 15 years before a vaccine is out in the market.
It was money, and then it was scientific collaboration. A lot of groups opened their data. So, for example, we developed in 40 days the first diagnostic in Brazil, molecular diagnostic based by PCR, because there was a Berlin and there was a CDC protocol that they published. They gave out the sequences so we could develop our own PCR test. Usually we developed a molecular biology test in a couple of years. But as everybody else shared data, we could develop one in 40 days. The other thing is what I said about doing things in parallel. I don't know, non-human primate studies cost a lot, so you won't run non-human primate studies before you have mouse studies. But during COVID, nobody cared if it costed a lot, because there was funding for research. So you could run the two at the same time if you wanted. And you could start scaling up production at the same time, even though you didn't know if it would work at all, because you had money to do that. And companies put a lot of money, but actually they didn't, because they got it from governments that already bought vaccines that were not even in development yet. All the funding organizations like Gates, CEPI, Gavi, all of them finance projects in new technologies, so everything worked very fast. If you do that one thing after the other, well, we wouldn't have a vaccine for COVID, which saved a lot of lives, because it would take 10 years and it would be, everybody would have had COVID by then and it wouldn't matter.
Thank you.
Thank you, and a very interesting talk. I wanted to jump off to the example of the timeline for the Zika vaccine. Do you find a way? Could we find a way for vaccines such as those that fall off in development to perhaps come back? Is there any way for as a preventative measure, let's say, to avoid something in the future? Is there a way in the market to make that work somehow?
Yes, there is. The problem is that the companies were not interested anymore, but in fact, you can take it to phase 1 easily enough, because phase 1 is not looking at infection or efficacy, it's looking at safety and immunogenicity. So you can show that it's safe, and you can show that it's immunogenic, it raises antibodies, or if it's a T-cell vaccine, it raises a T-cell response, and you can see that. There was a presentation on chimp studies this afternoon, so you can have chimp studies, and there has been a chimp study in the US for Zika. So you can infect the person, you can vaccinate and then do a controlled infection study, and that helps to go a bit further. It's almost into phase 2, because you can test doses and you can design your vaccine. It's never going to be an efficacy unless you have the cases. But if you have it, you have done a phase 1 and you have done a chimp where possible. You won't do a chimp for Ebola because there was no medicine, and if the vaccine doesn't work, the person dies. But it was not the case with Zika. It's not the case with malaria. There are anti-malarial drugs. So when we test the new malaria vaccines, we do chimp studies. You have a controlled infection, and after so many days, if the vaccine hasn't worked, or there are individuals who haven't responded, then you treat with anti-malarials and it's okay. But that helps going to almost a phase 2 of the development. And if then occurs an epidemic, you can roll it out as a phase 3, already protecting people. Which was what happened with Ebola, actually. The Ebola vaccine was almost ready. Well, it had phase 1, and when there was the next crisis, they did a phase 2, 3 at the same time. Thank you. There is another number.
Thank you very much for the interesting talk. I have a question regarding the vaccines that they target multiple diseases, like how are they developed?
Okay, some of them are developed individually and they're bundled together. So the MMR, you have a measles vaccine and a mumps vaccine and a rubella vaccine. And now you have also varicella. And then you put them together at the end. You produce one, you do quality control, you produce the other, you do quality control, the third one, the fourth one, and then you put them together. The pneumococcal, there are now 21 valent from Merck and 20 valent from Pfizer, and it's the same thing. You produce 20 different vaccines and you join them. There is another effort to make multivalent vaccines where you look, for example, there's a project on flavivirus where you look for common things in all the flavivirus, and you can try to use that for an immunization, or there's a T-cell vaccine that is being tested, but it claims to be pan-flavi, because then one vaccine would protect from yellow fever, dengue, Zika, West Nile.
And the ones that they combine, do they need to do further quality tests?
They do the final one, yes. So you do quality control on each one, plus the final product. And there is a great need for combined vaccines. If you ask for producers or parents, everybody prefers that we have less vaccines because children get vaccinated 20 different times. And some vaccines have 3 or 4 doses. So if you have vaccines with less number of doses, or if you can bundle more things together, it's obviously better accepted by the public, and it has a good effect. Please.
Thank you for the presentation, and I was interested in a little bit about the institutional things. So you talked about the European vaccine ecosystem, and I feel like it's very difficult because there are so many different entities that are involved. For example, they have all very different interests: pharmaceutical companies, academia, and maybe the government. And European government may be a bit interested in, for example, adulthood vaccine programs. For example, they're more into, for example, HIV vaccination development. So I am wondering if you have any strategy to for priority settings for different pathogens for vaccine development. And can I ask? Yeah. And also if you have any different indicators for different pathogens. Yeah.
Everybody has lists. And there is a WHO list, and there is the CEPI list, and there is the and EVAs, the European Vaccine Hub, has their own list. You can't avoid it, and usually you will see that things like coronavirus, influenza, RSV are in the list because they are airborne. And they have the greatest possibility for pandemic crisis. And then you have flavies, and you have sort of mosquito, tick-borne, vector-borne diseases. I don't know, in Europe there is Lyme disease, and in the North America, but in the north of Africa, this is not a problem, but they have more leishmania, they have rabies still, that a lot of the European countries don't. So then they start differing. WHO list looks at world, obviously, and CEPI. The AVH looks at Europe and pandemic preparedness, because pandemic affects Europe too. We're not exempt.
If there are any more questions, then we should. There is one more? One. Excellent.
Look, I had 60 slides and I passed some of them so quickly.
It's okay.
To do it in 45 minutes.
Thank you for the presentation. I wanted to ask about this bank that you said, bank on vaccines, that you will create until, like, the idea is to go until phase 1, no?
Yes.
Do you think that this is actually possible, and which are the factors that could determine which kind of vaccines could go on, like, will be economically okay to go until this phase?
So this was a PATH and CEPI led project to follow on their 100-day mission, because there is a famous curve that shows that up to 100 days the SARS-CoV-2 was going like that, and at 100 days did a curve and went up exponentially. So they want to cut it at that 100-day mark. We haven't got the capacity to develop a vaccine in 100 days. We did it in 250 something, nearly a year. And that because we had data from SARS-1 and MERS. If we didn't, if it was totally agnostic, it would take us longer. So the idea was to be able to have something for 100 days. But that would imply that nobody would do it for profit. And that's the problem. That's why no company agreed to participate in that. I was invited to participate because I was at Fiocruz, which is a public non-for-profit institution, and there were a few other members. There was a meeting in India that we all went and we discussed and everything, but we needed to get the vaccines that we're developing and submit them to that bank so that anybody could use them at the moment of crisis at the place of the world where they were needed. So if I developed a vaccine for Zika in Brazil and I took it to phase 1, I would give it freely, and if there was a crisis in Indonesia, they could take that vaccine and do a phase 2, 3 there. The problem with that is that not everybody wants to put their vaccine in the bank. You know, there's the money for the development, and so at the end, it's or it didn't happen, it's not going to happen, or it happens very slowly.
It also seems to be, however, Sotiri, also a national directive element here. There are countries that have vaccines quite high in their directives for public health. Brazil is one of them. India is one of them. So India is the biggest producer of vaccines in the world. It's not the US, it's not Europe, it's India. So maybe we should also consider some change in the way we are thinking about public health. Vaccines are preventative measures, but they're not therapeutic. You use them to prevent the big crisis. So you have to play a little bit with, I think, with the concept in people's minds. As vaccines have been vindicated a lot through the COVID-19 crisis. I mean, most of the people in here, we are in the biomedical profession, so most of us have been vaccinated, and most of us probably have a very good idea about vaccines, but I don't know if this is the same way that people outside of this room feel about vaccines. And I'm saying that also as a member of the National Vaccination Committee, as you know. I mean, you know, there's a whole different attitude about vaccines outside this room.
So it's very different. Like Brazil, like you said, is an example because the vaccine producers are public and the Ministry of Health funds them to produce the vaccines and guarantees to buy the vaccine from them. And the population, the first, at least the first vaccination for COVID, we had 98% of the population vaccinated, and it dropped to 80 for the second, and to 75 or 72 for the third vaccine. But the first one, we had 98%, which was second to Finland or something like that. It's something that is offered for free. All the vaccinations are offered for free, and people consider it as their right. They have the right to be vaccinated. The other problem is that a lot of the diseases that vaccines aim to eradicate have been eradicated for many years, so we don't have cases of children suffering from them, and people forget, or the current generation does not remember how it is to have certain diseases, or measles, or yes, and so they think that perhaps it's not necessary until it becomes.
Well, I think we should put this conversation to an end. Well, I see the president there from the conference.
Thank you very much for being here.
Welcome.
Just an announcement. Now we have a short coffee break, and then there's the bioinformatics session and another plenary talk about artificial intelligence, which was also very interesting. Tomorrow we have a problem. The metro will be open from 9 to 5, and the buses from 9 to 9 there will be working. For the morning sessions that some of you asked to, that they may be delayed, We will start a bit later, and also we will start with the tapas that they're living nearby and they can start. So if anyone has a problem, they can contact us. Okay, let's go for a coffee break.
Okay, everyone, so we will start the fifth oral session of the Congress on Bioinformatics and Computational Biology. So this time we will be hosted by Dr. Timokratis Karamitros, Senior Researcher at the Hellenic Pasteur Institute in Bioinformatics and Applied Genomics Unit, who's advancing on viral genomics, host-pathogen interaction and precision medicine, and as well as, I hope I pronounced it well, Dr. Nicholas Vakilis, Group Leader at the Hellenic Pasteur Institute, with an evolutionary genomist whose research explores de novo gene birth, orphan genes, and the origin of genetic novelty. So,
good evening, everyone. It's my pleasure to welcome you to this session of Bioinformatics and Computational Biology. First of all, we would like to thank the organizing committee for their hard work and their efforts. Having experienced the whole process myself a few years ago, I know how much energy and effort it takes. So the results that we see today are really impressive. Well done, guys. And thank you. Without further delays, let's start this session with our first speaker, which is Ekaterini Paleokrassa. Katerina holds a BSc in Computer Science and Biomedical Informatics from the University of Thessaly and an MSc in Applications of Biology and Medicine from the National Kapodisimo University of Athens. She is currently working as a research assistant in Dr. Dimas' group on the FAST Bio project at Alexander Fleming Research Institute Center, which investigates the molecular impact of periodic animal product dietary restriction in humans. The title of the presentation is Short-term animal product dietary restriction alters metabolic profiles and modulates immune function. Katerina, the floor is yours.
Good afternoon, everybody, and bonsoir à tous. I'll present my work entitled Short-term animal product dietary restriction alters metabolic profiles and modulates immune function. To begin with, dietary intake influences all physiological processes, including metabolism and immune system regulation. Diet in industrialized countries, frequently referred to as the Western diet, is rich in red meat and highly processed foods, while being low in vegetables, fruits, and fish, and has been shown to contribute to the incidence of chronic inflammatory disorders, including obesity, metabolic syndrome, type 1 and type 2 diabetes, allergies, autoimmune disorders, and cancer. Dietary restriction is defined as a reduction in nutrient intake without causing malnutrition and has been shown to lead to many beneficial effects in model organisms and humans. Dietary restriction enhances resistance and immune responses, improves metabolism, and in model organisms also promotes longevity. In humans, dietary restriction has been shown to reduce pro-inflammatory markers and affect biological pathways that influence the risk of age-related diseases. Dietary restriction may have other beneficial effects on human health. However, we don't know there are also negative effects. Dietary restriction studies are difficult to conduct in humans. For this reason, we leverage a unique human model, individuals who practice animal product restriction for religious reasons, and we aim to investigate the effects of this dietary pattern on systemic metabolism and immune cells. So we established the FAST Bio project, which explores the molecular impact of periodic animal product restriction in humans. We study a unique subgroup of healthy individuals from Thessaloniki who voluntarily alternate between omnivory and animal product restriction for religious reasons according to the Greek Orthodox Church. These individuals abstain from animal products, including meat, fish, dairy products, and eggs, for 180 to 200 days annually, in a temporary structured manner for at least 10 years. Animal product restriction is practiced during 4 extended periods throughout the year, as you can see in the calendar, along with all Wednesdays and Fridays of each week. And for this group, we use abbreviation PR from periodically restricted. In parallel, we study a group of individuals who follow the diet of the general population as control group,
And for this group, we used the abbreviation NR from non-restricted. The groups were sampled in autumn, when both groups were omnivorous, and in early spring, when PR individuals had abstained for 3 to 4 weeks before Easter. FastBio is a multi-omic study, and in this presentation, I'll show you the biomarker blankout findings, along with immune cell phenotypes. Here are the results for biomarker a complete blood count traits following animal product restriction. We observed a striking decrease in blood lipids, improvements in markers of liver and renal function, reductions in BMI and diastolic blood pressure, changes in immune cell population and traits associated with red blood cells, and also a striking decrease in normal range CRP levels. In the NR group, we observed decreased levels of gamma-GT creatinine and diastolic blood pressure, and we also observed changes in immune cells similar to those in the peer group, likely due to seasonal effects. Overall, following animal product restriction, most changes are in a direction suggesting positive effects on health. However, the increased levels of alkaline phosphatase may reflect negative effects on bone health and liver function. To explore differences in immune cells in more detail, we drew blood from a subset of participants and we isolated PBMCs. To explore the impact of dietary restriction on the abundance of immune cell populations, we performed flow cytometry to identify various immune cell subsets. To explore effects of dietary restriction on innate immunity, we performed a 6-hour simulation experiment. And to explore effects of dietary restriction on adaptive immunity, we performed a 48-hour simulation experiment, which I'll describe later. Here I'm showing you the results of the first experiment, the ex vivo. You see the distribution of immune cell types measured through flow cytometry in both dietary groups. Each participant is represented by 2 columns. The first column shows the distribution of immune cells at time point 1, and the second column shows the distribution of cells at time point 2. We observed high inter-individual variability between participants and relative stability of cell distributions across time points. Although there are high levels of stability across time points upon dietary restriction, we observed decreased frequencies for CD8 memory T cells, CD56 NK cells, and non-classical monocytes. In this diagram, each dot represents a different immune cell subset. Blue dots represent a decrease at time point 2, and the size of dots represent p-values. In the NR group, there were no statistically significant changes. In the second experiment, to explore the impact of dietary restriction on the magnitude of cytokine response to innate immune challenges, we performed a 6-hour simulation experiment where PBMCs were plated with a bacterial ligand LPS and a viral ligand R848, and we quantified 18 cytokines from the supernatant of PBMCs. In the peer group, we calculated the magnitude of cytokine response to stimulation at time point 1, light pink, and at time point 2, darker pink. We compared responses between time points and found a diet-associated trend for interleukin 12. In the NR group, we performed the same analysis, and we observed that at time point 2, PBMCs from both groups showed greater response to stimulation for pro-inflammatory cytokines, interleukin 8, MCP1, and TNF-alpha, likely due to seasonal effects. We observed the same pattern for pro-inflammatory cytokines, interleukin 8, MCP1, and TNF-alpha in the simulation experiment with R848. And in the third experiment, to explore the impact of dietary restriction on the magnitude of cytokine response to adaptive immune challenge, we performed a 48-hour simulation experiment where PBMCs were plated with PHA, which induces T-cell proliferation, and we quantified 13 cytokines from the supernatant of PBMCs. We calculated the magnitude of cytokine response to stimulation as in the 6-hour simulation experiment, and we observed a diet-associated strong response for the anti-inflammatory cytokine interleukin 10 not observed in the NR group, indicating enhanced regulatory potential to counteract inflammation following animal product restriction. Conversely, in the NR group, we observed a dampened interleukin-17 response at time point 2 compared to time point 1, while cells, PBMCs from PR individuals, in PBMCs from PR individuals, the ability to mount an interleukin-17 response at time point 2 remain unchanged. Our study of healthy individuals revealed widespread systemic metabolic changes associated with dietary restriction, including reductions in levels of total and LDL cholesterol, improvements in markers of liver and renal function, a striking decrease in normal range CRP levels, and reductions in platelet counts, which are in a direction suggesting beneficial effects on health. Furthermore, the reduced levels of CRP and platelet counts, along with decreased frequencies of non-classical monocytes, which is the most pro-inflammatory and senescent monocyte subset, suggests a tempering effect on inflammation. In line with this observation, we have shown in proteomics on the same individuals that animal product restriction leads to decreased levels of key inflammation-associated proteins. And in addition to reduced frequencies of non-classical monocytes, we observed a robust response for the anti-inflammatory interleukin-10, indicating enhanced regulatory potential to counteract inflammation following animal product restriction. For CD8 memory T cells and CD56 NK cells, recent studies in mice and humans show that these cells migrate from blood into the bone marrow to survive under conditions of caloric restriction. In our study, we cannot exclude that these cells may be undergoing apoptosis, but it's possible that similar to caloric restriction, these cells may be relocating to metabolically safer microenvironments. Lastly, our study revealed seasonal effects on the immune system, including immune cells and innate pro-inflammatory cytokines. And although there is growing evidence of seasonal variation in immune system function and for diseases including cardiovascular disease and autoimmune conditions, this field remains largely understudied. Collectively, short-term animal product restriction mostly improves systemic metabolic and immune health markers. However, this dietary pattern may have negative effects on human health, such as on bone health. I want to thank, first of all, Dr. Antigoni Dimas for our great collaboration on this project, Dr. Michalis Verikokakis, the rest members of the lab, our collaborators from the University of California, FastBio participants and the staff of the Inter-Balkan Hospital, and of course all of you for your time and your attention.
Thank you, Katerina, for your talk. Amazing. Are there any questions for the audience?
Thank you for the very nice presentation. You mentioned seasonal effects, so I wonder, like, when you took the blood samples from the two groups, did you check whether some of those individuals may have had the flu? They may have, and you maybe could control for that, because that could affect your results.
Yes, all the participants at both samplings, they did not have any acute disease or a moderate illness, with or without illness, and all participants with these illnesses or a moderate illness with fever or not were excluded from the study. So all the participants were healthy according to the self-reports.
Thank you for your talk. I'm wondering if I understood correctly, you said that the people in the dietary restriction group were there because of religion. They chose to have this lifestyle choice. Are there maybe other lifestyle choices connected with this? Maybe they protect their health more exercise, more abstinence from harmful habits that could explain those results and not only dietary restriction?
We didn't know that. For our individuals, we know that they are strongly motivated from their religious belief, and they usually adhere to this dietary pattern from childhood, and they are growing up in families that adhere to this dietary pattern lifelong. So I don't know for other effects.
So if I may, actually, they are not eating very well, despite the fact that they are restricting their diet due to this particular, let's say, religious reason. And also they are not very physically active. It's a very well random, actually, populational size from the Greek population. They are not like vegans, for example, who might have a very specific overall lifestyle and different from the other Greeks.
Thank you, Alex.
To ask you something, Katerina. You presented mostly beneficial impacts from this dietary. I was wondering that about the anti-inflammatory components of the blood cells, which were increased, right? The anti-inflammatory cells were increased, but this came alongside with the increase in interleukin, for example. Have you thought about the possibility of this change in the profile of the metabolic profile is a direct effect from the dietary or a side effect from the general change of the metabolic profile of the persons?
Yeah, some effects are from the animal product restriction we study, and some other effects may be from other factors such as seasonal variation, as I discussed before.
So we cannot be sure if these changes are directly linked with the change of the dietary, or is a side effect, let's say. I mean, the general improvement in health and...
we study the effects that are stemming from the depletion of specific food stuffs here, which are almost all the sources of animal protein, except for mollusks and shellfish, which are permitted for these individuals to eat. And we study these effects.
Okay, thank you so much. If there are no further questions, we can proceed. Thank you.
So for the next talk, we're sticking with BSRC Alexander Fleming and dietary restriction. And so next up we have Alexander Osing-Stras, who's going to talk to us about the proteogenomic effects of dietary restriction.
Okay, so hello everyone. Thank you for the introduction and the opportunity to present here my work today. So my talk is entitled Dietary Responsive Proteogenomic Effects Following Short-Term Restriction of Animal Products in Humans. Mainly I'm working with human proteomic and genomic data, and my PhD thesis actually focuses on two complementary, let's say, research goals. The first one is to identify variants in the form of SNPs that affect protein levels, and these are called protein quantitative trait loci, or PQTLs. And the second goal is to explore whether these genetic effects on proteins might be modulated by the environment or the diet in this specific example. So what motivates this work is the recent advances in protein-wide association studies, where the goal is to measure thousands of proteins in blood circulation through targeted or untargeted methods. And then these proteins can be associated with individual level genetic variation to identify, let's say, the genetic determinants of these proteins. And ideally, this information can be combined with GWAS signals to explore the biological mechanism of complex traits and diseases by linking proteins to specific, again, phenotypes. So just to paint the picture around these studies, recently in the UK Biobank, one of the largest biobanks in the world, there was a study that identified genetic determinants for more than 2,000 plasma proteins in 35,000 individuals. Now, despite the huge success of this study, the authors also stated that we are reaching a plateau regarding the genetic regulation of these proteins when we are considering only, let's say, a single protein measurement for each individual, because we are getting a snapshot of the protein. However, I think it's fair to say that proteins would be under continuous adaptations due to environmental changes, and one particular example of this environment is diet, which constitutes a modifier risk factor for many diseases. Now, dietary restriction in particular, which of course has many forms, has been linked to various health outcomes, mostly beneficial, by inducing key protein and metabolic changes. However, the question is whether all individuals would benefit from a particular type of dietary restriction. So, to address this question, actually the FastBio provides a unique opportunity. Katerina, the previous speaker, has already presented you the current study. Let me remind you that we are investigating a group of individuals called PR group, periodically restricted individuals, who alternate between omnivory and restriction of animal products. Now, these individuals actually have been profiled at 2 time points, of course during omnivory and after 3 to 4 weeks of continuous restriction. And in parallel, we have a control group profiled at the same time points of continuously omnivorous participants. So, as for the data that we have quantified more than 1,000 plasma proteins at each time point for each dietary group, and we associate these levels of proteins to 5.1 million SNPs, genotyped and imputed autosomal SNPs, in order to identify which proteins are under genetic regulation, and this is actually the first goal of the study. So we did that, and we identified that 36% of these tested proteins have been associated with at least one PQTL. Again, this is a genetic variant that influences protein levels. So here you can see a summary table with the context. This is the time point and dietary group combination in which we perform the genetic association analysis. We have the number of proteins that are under genetic regulation, and of course the number of PQTLs that affect these particular proteins. So the next very crucial step was to evaluate the robustness of these findings. So we compared these results with the UK Biobank, and we managed to replicate close to 82% of these genetic signals. However, the question again remains, which PQTLs are actually triggered upon dietary restriction? So to formally address this question, we jointly analyzed the results from all contexts using a meta-analysis framework, and this actually revealed 9 cases where the genetic variants, the effect of the genetic variants actually depends on the environment or the context. So here you can see the effect estimates for these 9 cases at each time point and for each dietary group. So for example, here in the PR group, we have the same variant that has no effect during omnivory. However, upon restriction, we have a gained genetic regulatory effect. In addition to the genetic response, let's call them genetic responses that are due to diet shown here in red, we have other cases, a single case of a shared genetic response on a particular protein, shared, between dietary groups, and other cases where the genetic responses appear to be unique to the control group. And I will discuss a few of these in the next few slides. So just to showcase a very interesting finding, we have identified a diet-responsive genetic effect on a protein called LBR, which is essential for endogenous cholesterol biosynthesis. And as you can hear, as you can see here, this gene actually targets cholesterol production through two different, very complicated, for me at least, to understand pathways. And also we have found that this particular protein is required for cell viability under cholesterol starvation conditions. This actually aligns with our findings from our recent publication, which shows that this particular dietary restriction actually results in a striking decrease of plasma cholesterol levels. And here you can see the downregulation of various cholesterol metabolites only upon dietary restriction, and in no other case. So to explore the biological relevance of this finding or biological interpretation, we used other blood biomarkers that we have within our cohort in order to validate this genetic effect. So using total cholesterol levels, we identified that the same genetic response driven by dietary restriction that drives LBR levels also extends to total cholesterol levels in the same direction. So here a plausible explanation is that the carriers of this particular allele, this variant, could increase the levels of LBR upon dietary restriction in order to counteract the cholesterol scarcity that they are facing. As for the translatability of this finding, we combined actually our results with GWAS data on obesity using a method called colocalization, and we saw within this overlapping region that the same variant that drives LBR levels also can affect obesity risk. Again, this is evident only upon dietary restriction and in no other case. So, as I said before, not all dietary genetic responses are due to diet. There are several cases, 4 cases actually, where the genetic response is unique to the control group, to the continuously omnivorous individuals, and these responses are related to proteins that are interconnected through the same protein-protein interaction network, which is enriched to apoptosis and immune-related pathways. Now, given that various immune parameters appear to have a seasonal fluctuation throughout the year, we expect that this signal could be due to seasonal effects that are being masked from this particular dietary restriction. Therefore, they are not evident in the periodically restricted group. So, just to wrap this up and give you an overview of our results, we have identified or mapped hundreds of PQTLs with high replication rates. Most of these genetic effects on proteins appear to have a shared, let's say, effect across contexts. However, there are cases, again, where the effect of the genetic variant on the protein depends on the context. For example, PR-specific genetic responses are likely driven by nutrient intake, and here I showed you one example between LBR and cholesterol intake, while shared and control-specific responses are related to apoptosis and immune functions. We also demonstrated that given the right environment, we can link proteins to specific health traits that would have been missed, for example, if we consider a single protein measurement or a single context. Overall, our findings suggest that diet effectiveness can be mediated by genetics. Therefore, investigating gene by diet interactions or in general gene by environment interactions would help us prioritize targets for disease prevention or even personalized medicine. And with that, I would like to end my talk. Thank of course my supervisor, my colleagues, and all of you for your attention. Thank you.
Thank you, Alexander. That was fantastic. Questions?
Thank you very much for your work. Congratulations. I want to ask you, what is the genetic determinant that drives LBR expression?
Yeah. Actually, we have found several independent genetic signals which are mapped relatively close to the TSS of this gene of LBR, about 300 kilobases or so. However, we prioritize only one genetic variant that can have a diet responsive effect. If we look in larger biobanks, there are some other PQTLs that can drive this particular protein, irrespective of the diet.
When you say drives expression, you mean when it is there, we have increased expression, and when it is absent, we have lower levels of expression. This is what you mean?
Yes, yes.
And then can you hypothesize on the mechanism through which diet restriction changes or modulates the genetic effect?
I mean, I'm not sure about the mechanism. For example, I would expect if lands on a transcription factor binding site, maybe it could change the overall expression. But also, if it targets a nutrient sensing pathway, for example, it can then, let's say, affect the overall abundance of this particular protein. But mechanistically, no, I'm not sure how is this possible.
Could be an effect directly on the gene or indirectly through the regulation of the transcription factor?
Yeah, I mean, this is a fair point. We also have gene expression data, and currently I'm working on this. And if I have an overlap that the same variant that drives gene expression levels of LBR also affects the protein level, I imagine that it influences directly the gene and not the protein per se. But I'm not sure at this stage. I have to check.
Okay, more questions? Perhaps a few more minutes? No? I have a quick question. So you mentioned the transcriptomics data. Have you started looking at these in correlation with the protein measurements?
Not at this stage, but I'm planning to check whether exactly this is the point, to check the overlap between AQTLs and PQTLs, I mean genetic variants that influence gene expression, whether this effect also translates to proteins. In the literature, the overlap is between 30 to 40%, is not quite high. Maybe due to post-translational modifications, I'm not sure, but
it's buffering also.
I mean, I have no degree in biology. I'm a statistician, but I believe that.
Okay. And any other question? No, a quick one. How prevalent is this SNP in the DR cohort?
Yes, it's about, if I recall correctly, 30%. It's common. It's not like
30%.
Yeah.
And in the general population,
it's about the same, because all of these variants actually come from SNP arrays, right, which actually target common SNPs. And we also filter out all the variants that have a minor allele frequency below 5%. But I believe for this particular example, it's like 30%, 28 or 30, I'm not sure.
Okay.
And in European, it's fairly stable. It's not like you have, let's say, large differences or disparities between the allele frequencies.
Okay. Thank you. So if there's, yeah, I think other comments.
Our next speaker is Christina Emanuel, again from the Biomedical Sciences Research Center Alexander Fleming. Christina holds an MSc in Bioinformatics, where she focused on machine learning techniques and analysis. She is currently a second-year PhD student investigating the impact of structured dietary pattern on the composition and function of the gut microbiome. The title of the presentation is Rapid Remodeling of the Human Gut Microbiome in Response to Short-Term Animal Product Restriction.
Thank you for the introduction. Hello, everyone. I see that you're pretty familiar with the Fast Bio project already, but please allow me to add another aspect of our research, and this is what happens with the gut microbiome of these participants upon short-term animal product dietary restriction. And you may ask, why do we care about the microbiome in the first place? And that is because we have come to realize that it has a very important role in human health. Through fermentation of proteins and complex carbohydrates, it produces vitamins, amino acids, short-chain fatty acids, and metabolites with a broad range of effects, such as providing energy, regulating homeostasis, regulating inflammation, and participating in signaling. And through all these functions, it contributes to risk for diseases such as obesity, diabetes, cardiovascular disease, neurodegenerative conditions, or even cancer. And we know that diet is a major factor that affects gut microbiome composition. So you're pretty familiar with our study design as well, but let me briefly remind you that we study a subgroup of the Greek population that voluntarily practices animal product restriction. We call these people periodically restricted, or PR, because they alternate between omnivory and abstinence from animal products, and we have profiled them after an extended period of omnivory and after 3 to 4 weeks of restriction. And in parallel, we have profiled the continuously omnivorous control group, which we call non-restricted, or NR. So we collected fecal samples from all these participants. We isolated the microbial DNA, and we performed 16S rRNA sequencing in order to identify the bacteria in the samples up to the genus level. And here is where the bioinformatics analysis comes in, and I'm moving on straight to some results. So first of all, I calculated some alpha diversity metrics, and here I present richness and evenness. With the red color, you see the peer group in the 2 time points, and with the gray color, the control group. And what does this show us? That the number of unique taxa decreases upon restriction, whereas no changes happen in the control group. However, the distribution of bacterial abundances is not affected. The community remains even. That means that no particular taxon starts to dominate. And in general, I have to say that decreased alpha diversity in the literature has been linked to some diseases of affluence. However, in our case, although the value decreased, it still remains in the normal range. And we actually suggest this response could indicate an adaptive response of the microbiome toward functional optimization under the consumption of a less diverse diet. And it could actually indicate the depletion of taxa that are no longer relevant for the host, and mostly rare taxa are affected. And the question that arises is, okay, but which are these bacteria that are affected by restriction? So I performed a differential abundance analysis in the peer group from time point 1 to time point 2, and in the control group. With the blue color, we see bacteria that decrease in abundance, and with yellow color, those that increase in abundance. And we see only one finding in the control group, but almost a third of the community is affected upon restriction, 49 genera, and the majority decrease upon restriction. Among the decreased genera, we found genera that in the literature are typically associated with meat or dairy consumption, such as Ruminococcus or Collinsella. And notably, we identify a group called lactic acid bacteria that decreases. And the major source of these bacteria are fermented dairy products, which are not consumed under restriction. On the opposite direction, we see some increased genera that are commonly linked to plant-based diets, such as Butyricicoccus or Lachnospira. And we also identify some novel associations of dietary intake with bacterial abundance. Notably, the abundance of some of these bacteria has been linked in the literature with some diseases. For example, Negativibacillus and Merdibacter, which decrease upon restriction, have been linked to inflammatory bowel disease, non-alcoholic fatty liver disease, and more. And most findings were in this direction, in a positive, suggesting a positive effect on health. But we also had a few findings in the opposite way, for example, an increase in megamonas. We should note that when interpreting such findings from the literature, we should be cautious because in most cases it's not clear if the increased abundance of the bacteria is a cause or a consequence of the disease state. And also, many times, findings from the literature are contradictory. And these bacteria participate in various metabolic processes, so I used a tool called PyCRAS2 to predict metabolic pathways that these bacteria participate in. And again, I performed a differential abundance analysis on the pathway level. Again, no findings in the control group, but 9% of the pathways were affected in the PR group. Our most significant finding was the downregulation of the mevalonate pathway and geranyl diphosphate biosynthesis pathway, and these are fundamental for cholesterol synthesis. Notably, these pathways are primarily expressed by lactic acid bacteria, which we mentioned decrease upon restriction. And we also see decreases in pathways involved in peptidoglycan biosynthesis, which is a crucial component of the bacterial cell wall, and in purine degradation pathways. Whereas on the opposite direction, we saw increases in pathways involved in the biosynthesis of essential nutrients, vitamin B2 and tryptophan. Our body cannot synthesize these nutrients by itself, so we rely on dietary intake mostly through animal product consumption. So we suggest that this is a compensatory response of the microbiome, that it senses that these nutrients are missing and tries to compensate for that. So broadly, we characterize microbiome metabolic responses as passive and compensatory. And then another question arises, what do all these changes mean for health? So we took all the differentially abundant bacteria, the ones that change their abundance upon restriction, and we tried to see how they correlate with blood biomarkers of health that we have measured. And indeed, we identified a lot of correlations, but many showed mixed effects. However, there was a clear signal regarding biomarkers of renal function. This means that all restriction-driven microbiome changes correlated with decreased levels of these biomarkers, which suggest a positive direction on a positive effect on renal health. And then we asked a final question, which is how does the gut microbiome correlate with plasma metabolites and proteins that we have also measured in these participants? So I used a tool called Diablo to perform multi-omics integration with the aim of understanding the association between biological variables and ultimately, hopefully, facilitate the discovery of mechanistic links. And indeed, I identified 4 major bacterial metabolite and protein clusters. We see a cluster of Negativibacillus, which is, if you remember, was our most significant finding, decreasing upon restriction, and it was linked with molecules with also the most pronounced changes upon restriction, such as the proteins FGF21 and HAF-CR1 and IDL particles. And these are molecules related to energy homeostasis, lipid and glucose metabolism, renal generation, and the immune system. So we suggest a positive contribution of Negativibacillus to these processes, and it is actually a recently discovered genus. Then we see a cluster of Coltemanella with molecules related to NAD biosynthesis, oxidative stress response, and inflammation. A cluster of Ticerella with molecules related to cholesterol metabolism and cardiovascular health, and a cluster of Bifidobacterium with molecules linked to inflammation, especially in the context of cardiovascular health. So overall, we suggest that these clusters capture systemic effects of dietary restriction. To sum up, we have shown that the response to dietary restriction is rapid but also likely transient. We have shown that dietary restriction decreases microbial diversity, decreases bacterial abundance, and downregulates predicted metabolic pathways. We saw metabolic responses that we characterized broadly as passive or compensatory, and we revealed a characteristic response to sustained nutrient availability. And while we recognize the difficulty in establishing causal relationships between the microbiome and health, we suggest some diet-responsive taxa that are possibly linked to health, and we bring them forward as candidates for further investigation. Regarding health, we saw some diet-associated shifts that were linked to improved biomarkers of renal function. And finally, we identified some links between bacteria and host plasma biology that, as we said, likely capture systemic effects of restriction and suggests candidate mechanisms of host microbiome crosstalk. So this is an ongoing project, and we are currently running some shotgun metagenomic sequencing experiments with the aim of achieving higher taxonomical and functional resolution. What does this mean? It will allow us to go to the species level, not just the genus level, which will give us a much better image of the functional capacity of the community. And it will also allow us to identify other kingdoms other than bacteria. And we want to focus specifically on viruses because those can contribute to disease pathogenesis, and certain types are being investigated as novel therapeutics, for example, bacteriophages as alternatives to antibiotics. And we listened to some very interesting talks on viruses yesterday and today as well. We have performed some pilot experiments with very promising results so far. This is just an example, a plot showing the composition of one of the samples. And ultimately, another goal is to leveraging additional existing omics data on host biology and integrate them with the microbiome data in order to better understand the host microbiome interactions and their impact on health and disease. So with that, I would like to thank everyone who has participated in this project, and of course, thank you for your attention.
Thank you, Christina. Amazing work. Any questions from the audience? Professor Ευθυμιόπουλος.
Συγχωρείτε που κάνω τόσο πολλές ερωτήσεις. Θα ξεκινήσω πρώτα με ένα σχόλιο όμως. Μου άρεσε ιδιαίτερα η συσχέτιση που κάνατε μεταξύ συγκεκριμένων taxa or genera with συγκεκριμένες καταστάσεις υγείας ή παθολογίας. Αυτό θεωρώ ότι μπορεί να έχει στο μέλλον πολύ σημαντικές επιπτώσεις στη σχετική έρευνα και στην μελέτη του πώς το μικροβίωμα επηρεάζει την υγεία. Είδατε, μιλάω ελληνικά.
Yeah, we should switch to English because we have
you showed data that the diet affects the population of certain taxa and certain genera. A decrease, actually, in the number. Yes. Is the decrease due to the death of these members of this genera and taxa? And if death is involved, what is the trigger of death?
Mm-hmm. So, yeah, we saw it through the alpha diversity metrics, where we saw the decrease in richness. This means that the total number of taxa decreased. And yes, since the bacteria actually feed on what we eat, some may be specialized in feeding themselves with meat or animal product components. So when they do not receive this through our diet, they probably die or get eliminated.
Get eliminated is very, very interesting, at least.
Yeah.
And very important to check whether this really happens, because this indicates a very active decision from the body to do something, which is really will be a breakthrough if you can prove this. And a question that does not really come from your talk or the previous talk, but it's something that came up in my mind is, do we have changes in the gut microbiome during the day? Are there any data in the literature?
Yes, actually. The microbiome is very active throughout the day. It follows a circadian rhythm as well. It has seasonal effects, but also daily effects. So for our study, these samples were delivered at the same time during both time points in the morning. But overall, yeah, it follows circadian rhythm.
Any other questions? Please.
Hi, thank you for your talk. It was really interesting. I suppose this is for you and also the previous presentations. Sorry if it already got asked, but so I suppose ultimately one of the things of this sort of work is to tell people, well, I don't know, influence us on our own diets and stuff. Do you feel like because I think it was 3 weeks on the
3 to 4 weeks of restriction. Yeah.
Do you feel like that's sufficient? And if you were to look at people who had stayed on for a vegetarian or a vegan diet for longer, do you feel like you'd see different results? And do you think that's, I don't know, do you think 3 weeks is sufficient?
So there have been some studies, extreme fasting conditions like drinking only water, for example, where you see differences after 7 days or maybe less in the microbiome. And yes, 3 to 4 weeks are definitely enough to see changes in the microbiome. And your other question was if, sorry?
If any other diets would be better, like vegan or vegetarian? Like longer periods of diets?
I suppose it's just, like if 3 weeks was sufficient, but also if you did it for longer, because people nowadays, I don't know, you've got this like intermittent diet, but also people nowadays also do vegetarian and veganism for extended periods of time. Whether you would expect to see, I feel like you would, but like very different results if you had looked at people for a longer period. Do you know what I mean?
Yes, I would expect some different results. For example, in our case, we saw an increase in vitamin B2 biosynthesis, but we didn't see an increase of B12 biosynthesis, which is something that we know is missing from the vegan diets. And this is because vitamin B12 can be stored inside the body for a longer period of time, whereas vitamin B2 does not. So, for example, this is one of the changes I would expect to see when someone follows a prolonged dietary restriction.
Hello, thank you for your presentation. I'd like to ask something that has to do with the unique microbiome of every person. Is it true that even with the ideal diet or the ideal probiotics, we can change due to a point the plurality of the microbiome? That's what I wanted to ask.
Yes, that is also true. We know that host genetics affect the microbiome. This is one of the things I want to explore in the next steps that I mentioned. Your lifestyle affects it, and also the microbiome becomes pretty stable during the first 3 years of life. And of course, you can alter it as we age, but yeah, there is a plateau.
Okay, thank you. It has to do with the genetic code as well.
Yeah, the genetics and the early life.
Thank you so much.
Thank you.
I have 2 questions. The first goes to you, Christina. How do you reconcile the apparent reduction of the bacterial diversity to the general shift to a more healthy condition in the people participating in the study? I mean, in general, the reduction by diversity is linked to a more negative, let's say. Can you comment on this?
Yes. So let me show the slide. Okay. So as we said, the value here of richness decreases upon restriction, but it still remains in the normal range. So there are some ranges, and we say that if it drops below that, it's linked to diseases of affluence. But for us, it remains in the normal range. And we believe that having more bacteria does not always mean the optimal state. You want to have bacteria that are optimized for your lifestyle and your body. So the fact that you lose some under a less diverse diet is not necessarily harmful for the body. It might mean that it shifts, the microbiome shifts in order to achieve functional optimization.
I was wondering if the time point that you choose is critical. I mean, you take the some during the shift, and the body might adapt later and recreate the richness, but with different genera. I mean, you maybe have during the change.
Yes, I understand.
It might be interesting to look at a few more time points later on.
Yeah, that is a nice point, yeah. Maybe bacteria that are involved in plant digestion, maybe they increase later on, yeah.
And the second question, a really quick one, because we're running late. It goes to the whole group of you guys. How do you comment on the sampling procedures that are involved in microbiome studies? Because lots of groups, I mean, even here in Pasteur Institute, we work with microbiomes. We have seen that sampling techniques and procedures and standard procedures are hugely linked to the results. Do you follow a specific protocol? Have you optimized the protocol?
Yeah, we actually spent, I think, more than a year optimizing our protocols from the recruitment phase and all the lab work. It took more than a year. We also had some collaborators in England, and they happened to test their protocol on their own. So we were doing experiments at the same time, separately, and we came to the same conclusions. So we believe that we have achieved an optimized protocol, and we want to try to publish this.
Great. Thank you so much.
Thank you.
Okay, so next up, the change is not going to be about dietary restriction. So it's the turn of Emilio Stasios, who's a PhD student here at the Hellenic Pasteur Institute. He's going to talk to us about the evolution of novel genes in yeast.
Okay, thank you for the introduction. Thanks for the organizers for letting me use a pointer and talk about cool stuff. So, fast bio. No, I'm joking. Actually, we're talking about evolution and genomics. Specifically, we're talking about protein and gene novelty. So let me make a small introduction about what gene novelty is, so everyone can be on the same page. And I'm going to use this tree as an example, this toy tree. So as we can see, in the ancestral species, there are 3 genes, blue, black, and red. These genes exist in all of the daughter species, but on the second species, there's an extra 4th green gene. And this is what we call a novel gene. Basically, novel genes are genes that exist only in a very, very narrow taxonomic group, like species, and does not exist anywhere else. Neither they have any similarity. And why do we care about them? Well, among other things, novel genes are linked to very cool species-specific traits, like the antifreeze protein in the Arctic cod that enables them to live in freezing cold waters without dying. And how do they originate? Novel genes can emerge by extreme sequence divergence. So let's say we have a speciation event, and the ortholog gene sequences of the daughter species acquire so many mutations as time progresses that they reach to a point where they have diverged so much that any similarity cannot be detected between them, thus appearing novel. A second mechanism that novel genes can occur through is the novel gene emergence. In the novel gene emergence, a non-genic region of the genome acquires genic-like characteristics like an open reading frame and regular expression, and somehow transitions to genehood, thus becoming a gene. These genes are entirely novel and do not share any real similarity with any existing genes. This by itself makes these genes very interesting biologically and evolutionary, because they have a high possibility of encoding proteins that they have new functions and new structures. But what makes them even more interesting is that even though the novel genes have been discovered 20 years now, and we are sure they exist, basically everywhere we look, and we have the quality of the data and tools to search for them, but most of the things about them we are not really sure, meaning that from how they emerge to how they acquire a function to how they get fixed in the population, most we have is indications. Also, most of the works that have been done on de novo genes are on a small scale, focusing on a species or two, and basically focuses on a few examples to analyze somewhat further. On the project that I'm going to be talking to you about, we undertook the task of analyzing de novo gene emergence at a higher level, at a subphylum level, specifically the budding yeast subphylum. And why the budding yeast? Well, because some species that belong to this subphylum are as diverse as humans are with roundworms. And also, the quality of the datasets are pretty high. Here you can see the species tree of the dataset we used, which is comprised of 332 genomes. So in the dataset we used, the genes were already clustered in gene families by OrthoMCL. We applied a few corrections on these gene families because we want them to be as precise as possible for the de novo gene identification. And for the identification process itself, we did a two-step filtering process. Basically, we excluded all of the gene families that had similarity outside of the Saccharomycotina. And second, the genes we were left with, we required them to have a conserved synthetic region in at least 5 closely related species that did not have any genes or ORFs in them. So after these steps, we identified a robust gene set of 5,600 and something de novo genes. And here you can see them. You can see their distribution on the species tree. As you can see, most of the species do have at least one de novo gene. As I said in the beginning, the novel genes can occur through de novo gene emergence and sequence divergence. With a similar pipeline as we identified de novo genes, we were able to identify 8,000 sequence divergent genes. And we asked ourselves, how similar are these genes, these two different gene sets? The thought process is that one gene set, the sequence divergent genes, have their origins in coding sequences, while the de novo genes have their origins in non-coding sequences. So we thought, okay, this should reflect on their sequence or structure. So we calculated a bunch of properties, and 2 of them showed large differences. The first one was length. Divergent genes are much longer than de novo genes. As you can see here on the left, we compared de novo and sequence divergent genes on a genus level, so we can exclude any taxonomical biases. As you can see, in most genera, the difference is very visible. And when we put them all together, as well, you can see that divergent genes are much longer. The other property that showed difference was disorderedness. The divergent genes were more disordered than de novo genes. And this actually agrees with the previous analysis that showed that disordered proteins evolve faster and have a higher possibility to accommodate sequence variability. As you can see, again, in all genera, divergent genes are more disordered. So, okay, we identified the de novo gene set, we compared them to sequence divergent, but are they really genes? Do they have a function? One way to find out is to measure selection. Now, selection can be measured in various ways. We chose to use the DMDS ratio, which is the number of non-synonymous to synonymous substitutions. The thought process behind it is that if changes on a gene sequence affect the fitness of the population in a negative or a positive way, these genes should have a function. Now, for the ratio, if the ratio is under 1, it means that there is negative selection or purifying selection. If the ratio is over 1, it means that there is positive selection. And if the ratio is equal to 1, it means that it's evolving naturally, no selection at all. So for the gene families that we were able to calculate this ratio, we saw that in their majority, as you can see here on the histogram, that they had a ratio of under 1, so purifying selection, thus providing us with proof that these should encode functional proteins. Now, something very interesting about de novo being new is also their structure, and structure can also gives us indication about function. So what we did was we translated those genes in peptides and sent them to our collaborators in the Vanderbilt University, Dr. David Ringer and Dr. Adonis Rokas, where they had an ESM fold pipeline set up for us, and they used it to make a structural prediction about de novo genes. And as you can see here, here you can see the confidence of these structures. As you can see here, most of the predicted structures have a confidence of lower than 0.8, meaning that we are not really confident in these structures. But still, there are some structures that we are pretty confident about them. And we said, okay, we have the structures, maybe they have structural homologs. Maybe they look like a protein that already exists. So we used FoldSeq to search for structural homologs. And we found that de novo genes have novel structures, like this A helix. It's a very simple structure. But also, what's more interesting is that some structures were not so novel, like this protein right here, which matched for which we found a homolog in E. coli. It's called protein INA-A, something like that. Now, I'm reminding you that these two sequences do not share, these two proteins, their genes, do not have a similarity on the sequence level, but on the structural level, I think we can all agree that they're pretty similar. So this might be a case of evolution reaching to the same solution from two entirely different paths, meaning there might be a possibility of convergent evolution. Of course, we cannot really exclude a case of a horizontal gene transfer from E. coli to yeast. So to sum it all up, we showed, it's already been shown, but I think on the scale we did it, we showed that de novo gene emergence is an important evolutionary force. We were able to identify a set of 6,000 genes, and for some of them, we showed that they should be functional. And also we made prediction and found out cases of convergent evolution and novel structure. Now, I want to thank all of my lab mates in the egg, as we like to call it, our collaborators in the computational genomics group in Fleming, Roga's lab in Vanderbilt, and of course, our funding agents, and all of you that were here today. Thank you.
Thanks a lot. Thanks a lot for the amazing and the fun talk. What I perhaps missed is in the first step when you check for the protein families, these are from computational based genome. Yeah. So why you didn't try to search from the RNA-seq of yeast? Either
I don't think we have so many RNA-seq for all of the tree there. I'm not sure the data exists.
But even though for the proteins that you say that are de novo proteins,
yeah.
will be this a clue? Either are
we actually have checked RNA-seq and we see that they are expressed, at least some of them, I'm not sure about the number.
And also one more question. If they have these, if these genes transcribe to RNA, this doesn't mean that they have a functional role.
No.
No.
It could be a case of pervasive transcription. We know that most of the genome is being leakily transcribed. If this is a word, I mean, yeah, it could be cases of leaky transcription.
Thanks a lot.
There is a question back there.
Hello, and as has been said, amazing presentation. What I didn't quite catch during your speech was de novo genes arise from non-coding sequences.
Exactly.
Do they stay non-coding after the emergence of it, or do they start coding?
That's the whole case. I mean, transitioning from non-genic to genic. That's the main idea.
Oh, okay. Thank you.
Can you listen to me? Maybe not just like this. Yeah. Okay. Yeah.
If there are no further questions, oh, there's okay, one right there. To the left of you.
So great talk. You mentioned that you used a symphony rule to select the de novo genes. Do you think that you may lose some of them because of some rearrangements in the genome?
Yeah, for
and
sure.
What do you do to may, also include them in your analysis?
We are trying to be as strict as possible, so we do not include them. Basically, we cannot be 100% sure that they are de novo. We do not exclude. We do not include them in the de novo gene set. There's a question.
Hello, thanks for the talk. It was really cool. I just have one question because it was not clear for me in your groups of de novo genes and genes that arise because of mutation. In which of those groups do you include genes that appear because of duplications of genes, and then those pseudogenes accumulate mutations are in fact then new genes?
So duplicated genes should be excluded, except if they have the duplicates have diverged so much that they do not share any similarity. So duplicated genes are excluded.
And those ones are included in are not.
Yeah, they're not. Yeah, in neither of neither of the gene sets. Yeah.
Okay. Thank you.
You said that duplicated genes could be excluded, but they can only be excluded if they are starting to be like non-coding genes afterwards, like through generation of the genes, because otherwise you could have like a positive selection on them, like with either with negative and positive selection, you will have you could trace the mutant like frequency.
Yeah.
And so you could not really exclude that then, like.
So
if this duplicated gene would be like pseudogene, then this could be considered.
When we talk about duplicated genes, are we talking about paralogous genes or genes that exist in different species?
No, paralogous genes.
So, yeah, I think we've excluded the paralogous genes now. Why? It's because we want to be as strict as possible. So, for the paralogous genes, we are not sure about their synteny, right? If they exist in two regions of the genome, I'm not sure how much we can look for the synthetic data.
Okay, just last question.
Yeah.
You said that you could not exclude horizontal gene transfer, but did you try, for example, to compare the different de novo genes, like GC percentage content, percentage of GC, to, for example, E. coli GC percentage? Because it's one of the criteria, for example, for
actually, we had some cases of horizontal gene. I mean, some cases of that we think that there are horizontal gene transfers. We did an extra similarity search in prokaryotic databases, and we actually found that a few hundred cases of small but significant similarity. So yeah, I just did not include them, so I think
Okay, yes? Yeah, please. Well, no, but if it's quick.
Yeah, I really wonder, that's why. So thank you, Milis, for the great talk. Congratulations for you mentioned that the horizontal gene transfer between E. coli and yeast?
I mean, this is a case of, we believe that it's not a horizontal gene transfer, but I mentioned that it might be, yeah.
Yeah, because we know that this usually happens between prokaryotes.
Yeah, exactly.
And the only rare occasions is when we have endosymbiosis. I mean, you have a prokaryote living within a eukaryote.
Yeah.
So, do you have evidences that we have endosymbiosis of E. coli in the
I think there have been some works that showed that the horizontal gene transfer between yeast and prokaryote is possible even without endosymbiosis, but I'm not sure on that. Okay. So yeah, we don't have it's just speculation, we're not sure. So.
Okay, thank you.
Perfect. Thank you, Milios.
So, next talk is from Dimitrios Tremoulis. Dimitrios is a PhD candidate in the Bioinformatics and Applied Genomics unit here in the Hellenic Paris Pasteur Institute, in our group. His research focuses on cancer and viral genomics, with particular interest in transposable elements, expression in cancer, and the molecular mechanisms of cellular senescence. Dimitrios' talk is entitled as Single-Cell Transcriptomics Reveals Immune Senescence as a Key Determinant of Melanoma Immunotherapy Response.
Thank you for the introduction. Thank you all for being here. Let's begin with a brief introduction. Immunotherapy is not effective in all cancer patients, and there is an urgent need to elucidate the differences between responders to the therapy, responders to the therapy and no responders. The unresponsiveness to the therapy can be attributed partly to dysfunctional immune cell states such as cellular senescence. Thus, the aim of our study was to validate a senescence gene signature called Senevic, which has been previously developed in this project, and also elucidate the role of immune cell senescence in the response to immune checkpoint inhibitors in melanoma. In this slide, we can see the public dataset that we utilized in this study. First, for the in vivo signature validation, we used the public single-cell RNA-seq dataset from mice of different age groups. Then, in the in vitro signature validation, we used a public single-cell RNA-seq dataset of human fibroblasts in two experimental settings. In the first setting, the fibroblasts were treated with different senescence inducers, namely irradiation, replicative stress, and etoposide. In the second time course experiment, the fibroblasts were treated with etoposide for 10 days. Then, after the validation of our signature, we used it on a melanoma cohort, which was also a public single-cell RNA-seq dataset from melanoma patients after immunotherapy. The cohort had 48 patients, 11 of whom responded, and 37 did not respond to the therapy. This is an overview of the workflows we employed. First, for the in vivo signature validation in mice, we performed data preprocessing, quality control, signature scoring, and then we compared the senescence scores between young and old mice. Next, we tested whether there's a correlation of senescence and age. In the in vitro signature validation, we also performed data preprocessing and quality control. Then we integrated the datasets, we performed signature scoring, and then we compared the senescence scores between the different senescence inducers, irradiation, replicative senescence, and ETO. Lastly, we also tested for a correlation of senescence and time under etoposide exposure. Next, for the melanoma cohort, again, data preprocessing, quality control. Then we performed automatic cell annotation, integration, signature scoring, and we compared the senescence scores between responder and not responder patients across different cell types. Now let's focus on the in vivo signature validation in mice. The data preprocessing step included the data retrieval and the transformation of the counts. Then for the quality control, we discarded all the genes that based on the number of their expressed genes and the housekeeping gene expression. We also removed the mitochondrial genes and decontaminated the counts. Then we performed signature scoring utilizing Senevic, the senescent gene signature, and other signatures to calculate a pathway score. Senevic is a 100-gene senescence signature which consists of both up and down regulated genes. In order to calculate a signature enrichment score for each particular cell, we calculated a score based on the Mann-Whitney U statistic. For each cell, we ranked the genes based on their expression, and the enrichment score was high if the upregulated genes of the signature clustered on the top of the list, and the downregulated genes of the signature clustered on the bottom of the list. Conversely, the enrichment score is low if these genes are randomly placed in the gene list. On the right, we can see a UMAP representation of the cells of the mouse dataset. The closer two cells are together, the more similar there are in their gene expression. The color of each cell is the enrichment score, in this case, their senescence. For example, this cluster has highly senescent genes. The next step, after the calculation of the senescence score, was to compare the senescence between young and old mice in various tissues. We found that old mice had higher senescence scores in various tissues, and we also found an increase in senescence score over the age of the mouse. Next, let's talk about the in vitro signature validation with fibroblasts. After the data preprocessing and quality control steps, we performed integration in order to remove the batch effects that are due to the sample, and also we performed signature scoring. On the graph on the bottom, we can see that cells from the same day under ETO exposure cluster well together, and they also form a path that closely resembles the time under etoposide treatment. On the right, we can see the Senevic senescence enrichment scores, along with the scores of the two most prominently senescence signatures, Sen-Friedman and Sen-Mayo. We can see that Senevic, along with Sen-Friedman, have a high dynamic range, since in day 0, the senescence scores are very low, and on day 10, they're very high compared to Sen-Mayo. Next, we tested whether the enrichment scores are different between the different induction settings, the ETO treatment, the irradiation, and the control. The control and the replicative stress. We found that all signatures exhibited higher scores in all methods compared to the control, irrespective of the senescence induction method, which shows the robustness of our signature scoring method. Then, we also have to point out that the enrichment score difference between the control and the senescent settings was higher in the Senevic signature compared to the other signatures, which shows that it is highly specific. Then, on the time series senescence induction setting, we found a correlation of the senescence scores across time using segmented linear regression. This also shows the robustness of our scoring method, and again, Senevic showed a bigger difference in the senescence scores of day 0 and day 1 compared to the other signatures. Now, after validating the senescence scoring method and the signature, we analyzed the melanoma cohort. Again, after the data preprocessing and quality control steps, we performed automatic cell annotation based on a reference dataset of 300,000 immune cells. We can see in this UMAP representation that the cells of the same cell type cluster together. For example, these are B cells, these are plasma cells, etc. Next, we performed integration. The aim of the step was the correction of clustering patch effect due to the different studies of origin of the datasets. The algorithm we employed was the fast mutual nearest neighbor correction algorithm. And here on these UMAP representations, we can see before and after the integration steps. Before the integration, we can see that the cells mainly clustered due to their study of origin. However, after the integration, we can see that the clustering is more uniform and it is based on their gene expression. Now, next, we performed signature scoring using Senevic, the senescence signature, a T-cell exhaustion signature, and a T-cell energy signature. This resulted to what we can say that is the most important finding of the study. We compared the no responders and the responders, and we found that no responders exhibited the highest senescence score in multiple immune cell types, namely CD8 cells, NK cells, CD4 T cells, and B cells. Interestingly, there was a specific cluster in CD4 and CD8 positive T cells, which consisted mainly of no responder cells and had a low senescence score, a high T-cell exhaustion score, and a low T-cell energy score. This shows that the unresponsiveness to immunotherapy is a multifaceted phenomenon driven by senescence and other pathways. The majority of this bioinformatic analysis was conducted here in the Pasteur Institute. However, these results have also been validated by our collaborators in the molecular carcinogenesis group in the Athens Medical School under the supervision of Professor Gorgoulis, and in the flow cytometry unit in the Athens Biology department under the supervision of Professor Ciciloni. In detail, in an in vitro experimental setting of primary skin fibroblasts, they performed immunocytochemical staining and FISH and found that 75-year-old donors exert replicative senescence. Then they isolated RNA from these cells and implemented the Senevic signature in RNA-seq data and found a progressive enrichment of the signature upon replicative senescence and age. Overall, these findings highlight the potency of the Senevic signature in detecting senescence. Then they assessed the senescence of an ex vivo clinical material from melanoma patients treated with immunotherapy. They detected and quantified GLF16 positive PBMC populations via fax and also stained tissue biopsies with GLF16 and immune cell markers. They confirmed that indeed NR patients exhibit a significantly higher senescence compared to responding patients. In conclusion, Senevic senescence signature accurately detects senescence in various settings. The cellular senescence of the immune cell compartment within the tumor microenvironment is a potent determinant of the response to immunotherapy. Strategies for accurately eliminating or reversing immune cell senescence are promising approaches to improve the outcome of immunotherapy. And I would also like to note that currently there are undergoing experiments in our lab for the detection of transposable element expression in cancer tissues using our 10x sequencing platform. I would like to thank Senior Researcher Timokratis Karamitros for his very valuable guidance and all the members of our lab for their continued support. I would also like to thank our collaborators for the molecular carcinogenesis group for their great collaboration. And I would also like to say that the manuscript for this work is available on bioRxiv and will be published soon. Thank you for your time.
Thank you very much, Andres. Are there any questions from the audience? Professor Efthymiopoulos.
Can you go back to your one or two slides? Let's see.
Yes.
Okay.
Sorry.
You say here that strategies for accurately eliminating or reversing immune cell senescence. Do you want to mention one or two?
Yes, this is a work that has been extensively done by Professor Gorgoulis' lab, but they're actively working in biomarkers which can show the senescent cells both in a live setting and or fixed tissues, as well as on senolytics, which is a class of drugs which is very promising for various cancers.
Any other questions? Here one, please.
Hello, thank you for the presentation. And I was just wondering, this CINVIC algorithm, can it be used for other types of markers, for example, not just senescence, but for example, proliferation or yes, activation, for example, because I'm having trouble with defining how activation markers are working. But yeah,
yes. Thank you for your question. The senescence signature has 100 genes, but since senescence is a very complicated phenomenon, these genes can be subclustered, let's say, to the activation genes, the proliferation genes, the SASP genes, which are the secretory molecules. So for sure, and we also tested some algorithms with subsets of the signature to verify some things. But as it is mainly for senescence detection. I'm sure that if someone looks more closely to the genes, you can make subgroups. But yeah. Thank you.
Other questions? No? Dimitri, thanks a lot.
Okay, so now we move on to the final speaker of this session, which is Maria Busali. And she's going to talk to us about, so she's a PhD student at the Hellenic Pasteur Institute, the molecular virology group for Dr. Karamitros. And she's going to talk to us about HBV integrations, occurring early in post-infection rewire metabolic and oncogenic pathways in hepatocytes. Thank you.
Good afternoon. Thank you for the introduction. Today I will present you our work on hepatitis B virus integrations that was performed at the Unit of Bioinformatics and Applied Genomics under the supervision of Dr. Timokratis Karamitros. Hepatitis B virus is a hepatotropic virus, prototype member of the Hepnoviridae family, that emerged 10 million years ago and is responsible for 1.1 million deaths annually. Although a preventive vaccine exists since the early 80s, functional cure strategies are still under development, and they have to overcome two main obstacles that arise mainly from the biology of the virus itself. First, the cccDNA, that is a replication-competent mean chromosome form of the viral DNA, and HBV integrations into the human genome, that is the topic of this presentation. HBV integrations are detected in the majority of HBV-related hepatocellular carcinoma cases, as well as in chronic and acute infection cases. They have a series of consequences that contribute to viral persistence and cancer progression, though their dynamics, both in number and genomic distribution, over the course of infection, as well as the integration hotspots, and the key physiological functions disrupted in hepatocytes have not yet been explored, and the aim of this study was to elucidate them. So what did we do? We strategically designed a methodological workflow that is based on state-of-the-art cell line models, long-read target enrichment sequencing, and dedicated bioinformatics workflows. A few words on the experimental setup. Mouse Passage primary human hepatocytes were derived from human liver chimeric mice. The cells were infected with HBV obtained from the Hep D19 producing cells. And an infection time course was performed for 1, 2, 3, 7, and 14 days post-infection in biological triplicates per time point and technical triplicates per sample. To enrich for HBV integrations into these samples, we developed a dedicated wet lab protocol that is compatible with Oxford nanopore sequencing. In summary, DNA was restriction digested and adapters were ligated to the sticky ends. Then, 2 rounds of nested PCR were performed and the PCR products were sequenced on the Oxford nanopore platform. Of course, the protocol had been previously optimized and validated using multiple negative and positive controls, as well as plasmids that mimic the HBV integrations and were in-house designed. Our in-house developed target enrichment protocol has also its very own accompanying computational tools. After the sequencing, the raw nanopore data are base called using the super accuracy base calling model of the Dorado tool. The goal here is, the goal in this process that is performed via GPU computing in NVIDIA's CUDA environment is to convert the raw nanopore signals enclosed in the POD5 files into DNA sequences into a FASTQ format, thus using neural network approaches for accurate base calling. Then, a bioinformatics suite developed and optimized in-house is applied. In brief, the raw reads are pre-processed, then human genomic loci with HBV integrations are identified, and on them annotation, functional enrichment analysis, and statistical analysis are performed. A few words about our bioinformatics suite, that, by the way, has been wrapped as an R package and will be available through Bioconductor as a standalone tool. The key concept here is to go from the base call data to the identification of human genomic loci with HBV integrations. And then these human genomic loci to be suitably annotated in order to identify hotspots of HBV integration into the human genome and reveal key functional pathways affected by HBV integrations. Moving on to the results, HBV integrations were detected as early as 1 day post-infection and reached their peak at 3 days post-infection. Poisson regression confirmed this peak, showing that the rate of HBV integrations at 3 days post-infections was 2.82 times higher than in 1 day post-infection in a statistically significant manner. Regarding their distribution, HBV integrations were found to preferentially occur within regulatory elements and CPG islands, as well as within protein coding regions, as determined by chi-square testing against their expected frequencies in the human reference gene. Moving on, functional enrichment analysis of the disrupted human genomic loci revealed that genes involved in metabolic and tumor-promoting pathways were currently affected by HBV integrations. Specifically, 56 genes linked to metabolic pathways and 20 genes linked to tumor-promoting pathways were disrupted. Within metabolism, we observed recurrent targeting of 17 lipid metabolism-associated genes, 10 glucose metabolism-associated genes, and 5 nucleotide metabolism genes. In the enrichment bar plots, where the bars are colored based on the p-values obtained from the functional enrichment analysis, nucleotide metabolism stands out. In particular, the functional terms cyclic and cyclic purine nucleotide metabolic processes were significantly enriched and were suggested as preferred targets for HBV integrations. To conclude, our developed long PCR-based target enrichment protocol combined with the log-grid sequencing and the optimized bioinformatics suite offers a valuable strategy for further exploration of HBV integrations and their implications. The observation that HBV integrations occur early, peak early, persist, and are predominantly localized in protein coding regions, suggest they could act as a stable viral reservoir, which poses challenges for functional cure strategies and highlights the need to consider them in HBV elimination approaches. And finally, the recurrent disruption of metabolism-associated genes suggests that HBV integrations may rewire hepatocytes to a cancer-like state and require functional validation. Closing, I would like to thank Nostos Foundation and the Hellenic Paris Pasteur Institute for supporting me through my PhD, all of our collaborators, as such a multidisciplinary work couldn't have been done by one lab only. Specifically, I would like to thank, sorry for that, Professor Michailidis and PhD candidate George Dangas from the Emory University, and Dr. Urane Georgopoulou, head of the Molecular Virology Lab of the Hellenic Pasteur Institute, and also Dr. Georgia Papadopoulou from the same lab. Thank you. Sorry. And finally, from this step, given also that I am defending my PhD soon, I would like to thank my supervisor and mentor, Dr. Karamitros, and all my dearest colleagues for all those amazing years. Thank you.
Wonderful questions.
Thanks a lot for the amazing talk and all the great work. Perhaps I missed out in your presentation. Are there any specific signatures in the genetic, in the genes that the HBV go inside them?
Like hotspots of integrations, right?
Yes, or something like.
It depends. Which actually in the liver biopsy samples, the TERT gene is considered a hotspot of integration. Though the last couple of years, this tends to not be found that much in liver biopsy samples. Though we are studying the hotspots of integration in our analysis, because the result I showed you today are one part of what we have done in HBV integrations, because we have used 4 different cell line models and animal models. And we're searching for HBV integrations, we are hotspots, and we are trying to make a map on how and why HBV integrations prefer to occur in specific regions of the human genome.
Thanks a lot.
Great talk. I would like to ask how to check the integration of HPV inside the transposable elements.
Yes, we have lots of projects about HBV integrations in the lab. In these cell lines I showed you today, there is not preference for integration in repetitive elements, though also we have a very big cohort of HBV integrations, and there it seems that repeatable elements are a hotspot for integration. Though also in the bibliography there are contradicting opinions on that, because also when you're talking about NGS, repetitive regions are a big problem to sequence, and maybe, yes, but in here we don't observe that. Sorry for the...
Thank you.
Yeah.
More questions? Otherwise, I have a quick one. So I don't know anything about how HBV integrates in real life infections. These are all from cell lines, right?
These are cells from humanized mice.
Yes.
Mice with human livers, and
Yes.
we obtain the cells from the liver of the mice.
But the infection happens on the animal.
No, it's de novo infection outside. Okay. We isolate the hepatocytes and then we infect.
But how often does HPV integrate in actual human genomes? Like if you get infected with HPV, how often does that happen? Do we know?
In the hepatocellular carcinoma cases, it has been found from the bibliography that HPV integrates in almost 85% of the cases as well, and this is almost the case for chronic hepatitis B, but in acute we have much less integrations.
So now it's a driver
regarding the timing though, we don't have this knowledge because, as you may know, liver biopsies are very not used these days because elastography and other more modern approaches are used. So we don't know what's going on in humans, in liver biopsy samples.
Okay. More questions, but in the interest of time, we should stop there, right? And thank all the speakers of the session again. I think now we take a really short comfort. No? No, we need to take at least a 2-minute break. Yeah. Okay, a very quick comfort break. No one leave the room. Okay. That sounded like a threat. It wasn't.
Διάλειμμα, Βασίλη, μόνο.
Of course, they cannot think. We program them, we make them. If you leave a computer on the desk, it cannot think. If we put an AI algorithm inside the computer, it cannot do anything. If you don't provide the data and you don't execute and you don't know what you're looking for, it doesn't mean anything. So, some of the examples that he had in his paper, examples of questions that the interrogator would ask to the two curtains is, please write me a sonnet, and he was expecting two answers, and his goal is to identify who is human, who is the machine, okay? So the answer was, count me out. I never could write poetry. That is something that a human being could answer. That is something that a machine answered. And one of the most famous questions that he asked is, add this number to this number, 34,957 to 7764. Wait 30 seconds and give me the answer. That's what he asked, okay? And the answer was this. Do you play chess? Yes, and so on. So, this number, add this number to this number, and his system, his algorithm, came up with this answer, okay? Right, so this answer is wrong. The right answer, if you add those two numbers, is 7 to 1, not 6 to 1. But he had programmed in his pipeline a defect, because his aim is not to give you the right mathematical answer, is to confuse you who is human and who is the machine. And if you have gotten this answer from a curtain, you would have believed that must be human behind that curtain who made a mistake in the addition. Okay, this is what AI is about, is thinking like humans, even with the defects and the flaws that we have in our minds. So he deliberately introduced a mistake so he could confuse the interrogator. So keep that in mind every time you think, I'm going to apply AI pipelines in my data because I want to be successful in my PhD thesis, okay? There might be flaws and defects because the AI pipelines follow mistakes that a human being would have made, okay? So this is his answer. It's too meaningless, of course, and the only real solution in doing proper science is knowledge, is know what you're doing. There's no magic AI that will solve your problems. This is his paper, 1950. I have a PDF copy, if you like. You can send me an email and I can send it to you. It's very interesting. And this is The Imitation Game. There's a very nice movie on Netflix. So, what we are doing with AI, we are interested in putting everything together, but doing the right annotations and the right links in the data that we have. So, data is the actual data. So, for example, you can ask a clinician, a physician, what is the blood pressure of your patient? And he will say, I don't know, 14 something or 20 something. That, in its own right, it has a meaning, but it may not have the meaning that we think if you don't check the metadata. So if this guy ate something salty 30 minutes ago, of course, this is just noise to your data. And by doing the metadata analysis, you can save yourself from putting together all those big data sets and actually shooting yourself on the foot, because you are actually putting in there a lot of noise. And the idea of attributing, identifying through the metadata analysis should be excluded from your analysis is very important nowadays in having a very good, well-defined, clear data set. And this is
the timeline of data accumulation. So this is the phase of no data, and this is a very blissful and fantastic phase. I advise you to stay here as long as you can. And then there is the phase where you have limited data, and then you have sufficient data, but there is no criteria and no function and nobody to tell you when you have enough data. If you have 10 people, 10 patients, or 50 patients, or 150 patients, if you send it to a good standard scientific journal, there will be that one reviewer who you think knows you and personally hates you, who will say, come on, you can do 5 more, you can do 10 more, it doesn't matter. Then you do 5 more, and it's, come on, now you did 5, and do another 20. There is no definition of how much data I should have to make a conclusion. And if I publish a paper with 100 patients, and I do everything right, I do all my homework properly, then when I reach 300 patients, I bet, and you understand that something will change to my original paper. When I ask ChatGPT or something to go and look on the World Wide Web and find some information, it will pick up my erroneous paper, my outdated paper, that will have the same statistical significance as the new paper. And then you see there is this artifact of calculation and availability of information on the internet. And there is this other problem, which not many people talk about it, and I should tell you a little bit about it. When those large language models became public, which is not far away, I mean, it's a year, maybe a year and a half when it started, this ChatGPT hype. The information that was available on the web, doesn't matter how good or bad it was, but it was human-made. It could be rubbish, but it was rubbish from people who did rubbish work. And there was other people who did excellent work, but it was human, okay? Now, a lot of people are using ChatGPT for the last year. They have been using ChatGPT to write papers, as we know. Of course, there are too many papers now. I mean, if you quote, certainly here, you will get about 70 papers on PubMed right now, okay? And somehow they went through peer review, somehow the editor didn't notice, somehow the author didn't decide to retract it. It's there. So that is AI made. And slowly, it was 100% to 0, human ChatGPT. Now, I'm saying a number so I can make an example. It's 98% to 2% that is being reproduced by those systems. If we keep on like this, after many years it will be 70-30, and after many years it will be 1 to 99. And then the human input will be statistically non-significant to the information that is available out there. It will bring so much noise to the scientific standards that it will, if it reaches a point and beyond, it cannot be reversed. So we should be super careful when we decide to use those systems. It could be beneficial from an energy conservation point of view, because otherwise you would have spent 2 weeks to write a proper essay. But in the long run, it's killing your opportunities to get a proper job in the future. So think about it. We should be very, very reluctant and very, very careful on how we use. I'm not saying don't use it, use it. But be extra careful when you're producing scientific work. So we need to come up with some definitions and find solutions to these problems. And here is an example. This is the Eiffel Tower. There is one more in Filatra. And you know it comes with architectural plans, and those people who were putting it together, they knew which beam and which piece of iron goes where, because that thing, the architectural plan, was made before the Eiffel Tower. So someone sat down, drew it, designed it, and then they built it, okay? And because we have those sketches, we can replicate the Eiffel Tower on a computer. And this is not a real picture of the Eiffel Tower, this is an AI-generated, a computer graphics generated picture of the photo of the Eiffel Tower using Autodesk. The same with the Empire State Building. Again, there are these architectural plans and everything, everybody knows where it's office, a window, and WC, and elevator, and cable should go. So it works as a building. And then we can use CAD, and based on the architectural plans, we can recreate the same building and make some very nice computer graphics representation of the real thing. The problem is that we don't know yet how to make a computer replica of that thing, because we don't have the architectural plans. And this is why we're all here. This is research, this is basic research. Basic research is trying to come up with architectural plans, is trying to understand the mechanism, the functions, the molecules, the philosophy behind life. And even though we have those, and we can make another Eiffel Tower if someone comes up with the funds, the money, we can make an exact replica of the real Eiffel Tower anywhere you like in the world, okay? But how can you program this? And this is a big problem. But if we break it into little problems, there might be a solution. And the solution that is nowadays gaining speed in AI is tokenization. I don't know if you're familiar with the term NFTs. Tokenization, συμβολοποίηση in Greek, ένδειξη, is to take a big picture and break it down in little but manageable parts that you can tackle, you can address. Tokenization is the process of creating a digital representation of a real thing. And you can do, nowadays, we do that in identifying the fingerprint. So if you're trying to unlock your phone and you put your finger like this, it just needs a part of your finger. It doesn't need your whole fingerprint. When you try to unlock it with your face, it needs, I think, 15 different points out of more than 2,000 points that it records every time. So if you put glasses, hat, something, it will still unlock your phone because it needs 15 different points out of more than 2,000. And this is due to forensics. Forensics, if you need to identify your fingerprint, they're looking for 15 different identities, and then they define that this fingerprint belongs to this person. And the market size now in AI is driven by tokenization. And to make it simpler for us to understand is that this is the full sentence. This is like 100% of the data that we have. We are moving to LA. This is 100% of that sentence. But in some cases, moving to LA is sufficient. Maybe that person is not interested who is moving to LA. He just needs to know where you're going, where everybody's going. Okay? And some other people are just interested that you just get the hell out of his site and just move anywhere you like. I don't care. Go to LA, I don't care. Move. And then you understand that if that is the full data, the 100% of the data set that you have, asking specific questions do not require 100% of the data. So even if you don't have the whole population of the globe, 8 billion people, to analyze all the transcriptomic data, because only then you would have the full data of the people who are alive today. That doesn't mean that you have the full data of the evolution that came about, so that gene behaves like that. Okay, so even if we could sequence everybody, you would still have just a tiny fraction of the real biology that is behind methylation that you're trying to understand why it's taking place or not. And those are the token labels, the BERT algorithms. So you have the input, and then you break it down in different portions, and then you make labels, different classes. And when you come up with a specific class that answers the phenotype, you lock the root. This is what you're after. You're not interested in the rest of it. And there are many different ways to do that. In general, the biomedical domain is very rich in words, and the jargon is, and the language that we use is above average. So we get better descriptions. And there are many ways to do that. So you have one data set, another data set, and then you can compare the two data sets together, or you can have a reference data set that you compare all the data that you have together. But the main thing is that you try to make a computer think like a human, as we said before. And of course, everybody's investing in life sciences. More money are being invested day by day in life sciences, big pharma companies, governments. Every time I say this, I wish that one day I will say this, I will say the same thing about this government, this country. But anyway, we're optimistic. I said that once more in another audience, and there was a fantastic old man who said, but they never invested, so they don't follow the trend. They never cared about research. Anyway, the problem is that the new medical entities that we have are getting fewer and fewer, basically because the easy ones have been discovered. Maybe because the legislation and the laws and the ethics now, in order to analyze, to go in vivo and do some experiments in animals or humans, has become harder and harder, and that discourages a lot of people. So in the older days, it was easier to go in human trials. Now it's harder. You need more money, you need extra work, you need a lot of things, and maybe that discourages some people. And of course, then there is the data issue, because when you have some sequences in one-dimensional data, it's not that difficult, but when you start to increase the dimensions and go to 3D and 4D and many different dimensions, and in science right now we are dealing with problems that have more than 450 different dimensions, physicochemical properties of a specific interaction between a protein and a ligand, or a clinical phenotype. There was a clinical trial for dementia that had 75,000 dimensions analyzed at the same time, because there were too many sub-phenotypes in dementia, and there were 75,000. So you can understand that there is no way for a human being, a person who sits in front of an Excel file, to do the analysis without using an AI system. And this is why we need data fusion and matrices and expert algorithms that bring these things together, because as I said before, it's very hard to come up with a candidate molecule, because all those things, it has to be safe, it has to be soluble, it has to be potent, and of course, it takes a lot of time to come up with a good compound. When you set your target, so you know that I'm starting my PhD in developing antiviral agents against a helicase of that virus, you stand less than 1 in a million to hit the market. So when you know your target, chances are less than 1 in a million that your compound that you will design in the future will hit the market. And the reason is simply because you have to validate your target, which takes a couple of years, then to identify the lead, optimize the lead, drug dosage interface, preclinical, and all those things will take more than 10 years and will cost to pharma industries more than $1 billion to go here. And you understand that if you are a young researcher starting your PhD, or you just got a position somewhere and you go to your boss and you say, look, man, I need 15 years, $1 billion, and for less than 1 in a million chances, it will not go well, okay? When you try to promote yourself, okay? And this is why we need new strategies and new modern pipelines to be applied in our science. So one thing that we did was to combine evolution with structural biology, and now I'm coming to the actual thing. A couple of examples that we have been developing in the lab. And the reason that we did this is that we know that structure is more conserved than sequence in nature. So the structure of a protein is more conserved than the sequence of the protein. It's easier for the sequence to change than the structure. And it's very simple. If the structure changes, usually it brings, I mean, there is the potential of something better, of course. This is why we're here. We started as single-cell organisms 4 billion years ago. But usually it's a non-survival or defect. Okay, so structure is more conserved. And we thought, why don't we, instead of comparing sequences, compare structure? And we started on nuclear receptors about 10 years ago. We started with a data set of 84,000 different nuclear receptors, entries, sequences of nuclear receptors from more than 300 organisms. And because we have so much information, when we see that a specific set of amino acids is conserved, and remember that we have 84,000 sequences aligned, so if this is 100% conserved here, it is there for a reason. We believe that if that thing is removed, something bad will happen to that protein or this organism. So simple. And then we go to the protein and we see where those couple of days, the proline and aspartic acid, where do they sit? Here. So if we change them on a computer and we model the system, then anything that will come up different than the wild type could be potentially be defective. And by doing that, we started to try to elucidate a couple of clinical cases that we had. And about it's about 9 years, 10 years now, we introduced a new syndrome, which we called Crusoe syndrome. Of course, it's Professor Crusoe who was leading and still leading this research. He still is one of my mentors, and we worked a lot on some cases that he had in the pediatric clinic, and we tried to elucidate, to understand the mechanism. So you have a mutation here, a histidine is replaced by an arginine, which has a longer side chain, picks up different interactions, but the most important is that the torsion angles now are defined, so that protein cannot move as a hinge anymore, it's blocked. And this explained the phenotype. And then we did it again with another case, and at some point it got boring because he had too many cases and we were doing the same thing for too long. So we stopped doing it. Other people are doing that now. We started to question ourselves, how can we learn from that? We did about 10, 15 different mutations. How can we reduce the dimensionality of the problem? How can we, because we have a very specific, very well-defined clinical phenotype, and now we knew sort of the mechanism. It has to do with the Cushing syndrome, and the resistance to glucocorticoids and the glucocorticoid receptor. So everything was there. Now we knew more or less the mechanistics of the protein. And we started to think if we can use that scenario as the scaffold on which we could be able to build our own algorithms that can be applied to different things. And we have to think differently. So what we did is we took all the proteins that we had and we tried to apply as many physicochemical properties definitions as we could back then. So you have hydrophobicity, polarity, charges, electrostatics, whatever. And then we took the proteins, we vectorized the proteins, we got rid of the proteins, and we kept the vectors. And we started to think if the vectors could lead us in finding similar vectors in other proteins. And by doing that, we developed our own algorithms that take the proteins from a 3D problem and converts them in a 2D problem. It's a similar thing that you would do with a globe, the Earth, when you want to make a map of the Earth that you want to put on your wall. Of course, the globe is not flat. Okay? And it takes a little bit of mathematics to go from a 3D object to 2D object. So we developed our own programs, our own algorithms, and then we discovered that when you make a 2D representation of a 3D thing, at a specific pose, at a specific structure, and then you subject that thing to a change, as proteins do, they're not rigid, they change shape, you can come up with something like that. So when we do molecular dynamics here, this thing changes. So if you come up with many molecular dynamics based on crystallography that are more reliable than sequence because we're talking about structure, and you come up with those maps, then you have too many of those maps, and then you can find the relation of each dot to the real thing, and then you can do molecular dynamics on the 2 dimensions, which saves about 2000% of the computational time, and you can do more calculations using the computers that you have in your lab. And there are many different ways to do that, and when you do that, then you can do a fast Fourier transformation, and you can go from 2D to 1D, and then take the histograms, the signals, and compare the signals, and those signals are proteins. So we started with that, and then we started to try to find scenarios, epigenetic scenarios, genetic scenarios, on which we could apply what we have already developed. And we started to focus on how cells communicate to each other. One thing I should say is that I don't have the grasp of time, and there is nobody here, so I'm going to be talking forever. And, it's like Forrest Gump. Someone has to put a sign and stop me. But to me, it's fine.
Where are these guys? What happened to them?
They got bored and they committed suicide or something. Anyway, so what we did is you have a parent cell. You know, most of us know what exosomes are. They're little balloons, membranes that travel from one cell to another. I wish it was that simple. It's not just like that. They start from one cell at some point, then they go into another cell, then they come out from that other cell a little bit changed, and then they go to another cell, and then they change a little bit, and it's like the Odyssey. It's like Odysseus, who went to different islands and changed his personality a little bit, but it took 20 years to reach the final version of him, because he was slightly modified due to experience and exposure. Okay? So we wanted to understand the information that goes from one cell to another, not just the molecules. It's not just defining the cargo of exosomes, like having a big list of proteins. We have worked closely with many collaborators who've done wonderful work on our samples, proteomics and metabolomics of Vangelis Ogikas, Martina, Pantelis, Hadjis, wonderful people, excellent work. But they gave us lists of 3,000 proteins. What am I going to do with 3,000 proteins? Too many proteins. I need to find something meaningful in those, in this data. And it's not just how many proteins you have. I really had long conversations with those scientists, with those collaborators. Said, please, can you please do the proteomic analysis and give me 2 proteins? Please. It's already too many proteins if you're trying to elucidate something, if you're trying to answer a hypothesis. 3,000 proteins, how can you put them together? So some of the interesting results that we found in exosomes is that we found some cytochrome, CYP genes, which was interesting because these are exosomes from breast milk, mother to infant, to newborn. And we didn't know why would you have those P40, 50 genes in milk exosomes. And then there were too many inflammatory genes. Too many, TRIM32, IFIT1, TBK1, and now he really got bored of me, so he said I should put a stop. Okay, I don't have a look. I am in slide 130 and I have 60 more. At least I'm honest. But I go through them quickly, okay?
Let's say that you have 5 minutes.
I have 5 minutes, okay. So, one of the most prevalent thing that came out from the transcriptomics analysis and the proteomics analysis was STING. STING is the stimulator of interferon genes, and it was always there in differentially expressed in high concentrations in exosomes of breast milk from more than 170 different mothers. And we were curious, why would you have the stimulator that this thing kickstarts the immune system basically? Why is it there? So we started to go into that thing and try to understand how it works. I'm not going to spend time, I only have 15 minutes, as you said. And inflammatory stuff, we know this thing, and then we started to model the protein and see how it behaves in the 3-dimensional world. There are some X-ray files, PDB files of that protein, but we wanted to see how it moves, so how flexible it is. So we subjected it to molecular dynamic simulations, and then we came up with our own neural networks. First thing that we did is we designed some machine learning approaches where, based on the torsion angles and sequence, and using support vector machines and neural networks, we attribute the function of a sequence of a protein. So we understand the protein without reading the paper, basically. We try to understand that protein, why it was built by nature, blindly. And then we went into neural networks, deep learning, and we developed our own spatial segmentation tools, as you will see later on defining space. And the best way to define space, conformational space, molecular space, was by using the Minecraft algorithm. And we took the protein and we represented it. You're familiar with Minecraft, okay? And the big thing behind the Minecraft algorithm is that a lot of people are playing on the same stage, the game, Minecraft, but that thing doesn't send back and forth an actual movie or an image of what is happening there. It compresses the image that you see and the actions of everybody into a function in mathematics, and it sends through your router, through your modem, the mathematics. and the mathematics is being recreated on your tablet or your phone or your laptop because they want to save the bandwidth. It's not like streaming a YouTube movie. They segment what you see, what happens on your screen, into functions. And we thought, let's do that to proteins. Let's take proteins and compress them into functions. What functions? What function will come up? What is the mathematics that can describe that? In order to do that, we started to work with voxels, 3D space, and we annotated the proteins with many different physicochemical properties. So you can have electrostatics, blue for positive, red for negative, white neutral, hydrophobics, and so on and so forth. And then we started to look for patterns. There are very good and very efficient algorithms that we don't need to reinvent the wheel that can help you look for patterns in anything. And then there is a triangle of 3 red cubes, and we know the x, y, z of those cubes, and more than 450 dimensions. And then, just an example, there are a couple of blue ones there. But we are interested in all patterns, in all proteins, and the associations between those patterns. And we analyzed 226,000 proteins from protein data banks using that approach. And then we got rid of the proteins and the cubes, and we kept the patterns. And we realized, as you will see later, that because we know so much about the associations, we identified that there are similar patterns in proteins that there is no clear biological explanation. They have similar structure, but you would never, as a biologist, guess that that protein behaves or has the same structure as your protein, the one that you're studying in your PhD. And then we started to think the Greek way, Poniroulika, and we started to teach our algorithm to learn, to take each cube and learn from the cube next door. So if you have a red cube there, and there is a white one there, and there is a blue one there, learn. And we have 226,000 of those representations, and we did that by randomly removing cubes and asking the computer to put back the right cube based on the context. So when the computer managed to achieve more than 90% accuracy in putting back the right cube, we gave it a bonus, and we kept that pipeline. And then we went here, and this is the active site of the protein, and we tricked the computer. We said to the computer, look, there was something here, can you put it back? But now we have established a system that is 90% accurate in putting back the right cubes, and the computer using the Tetris algorithm. So you see, we played a lot of video games.
Probably we should release some people, and some people you want to stay, because many of them, they may not want to leave in order to be polite. And I say that because, for example, the organizing committee is from early in the morning until now, and they will stay longer, and they will come again. And we have also to consider these people. Okay. And, but I like very much your talk, especially this last part, because it's more relevant to biology. Sorry, I'm talking about that. And it's really, really, really important. And I was thinking probably we have to have this in a longer session, like a workshop. I mean, for all students, at least in Athens and everywhere in the world, I mean, through streaming. These are very, very important stuff. I'm really impressed personally. And as you go now to these descriptions of your work and so on, and I think it's really important that we get a grasp on it, and you really describe them in simply. Because if you get the, as you said, the patterns and previously the directions, we learn a lot about this. I mean, I'm learning a lot today from you. So I think we should release the people that need to go, because myself also I live in Kalivia, you know, the place with tavernas.
No.
It's 40 kilometers from here. My wife keeps sending me messages, so I really have to go.
So everyone that wants can live without problem. I think Dimitris will not.
Yes.
But I invite you somehow, also the students, to organize a workshop. This is really very, very, very important. I consider it, for all of us to learn more. But this cannot be achieved really at this time of the hour within 2 hours. I mean, we need to discuss about all these things and to intervene and ask questions. So I think either in your department at the biotechnology department of the School of
AUA, Agricultural
or
University.
here, the students of the Pasteur Institute, they have to have a workshop on this. Also in the Department of Biology where I am. I mean, this is really very
We have done a couple of workshops in the past. Anyway, we could do that. Of course, if you want to leave, you can leave. I have to warn you that there are some wild dogs that they have released outside. So, go. I have programmed them to leave when I show my last slide. So, at your own risk, go.
So because it's very interesting, just tell us how much
Yes.
time we have, more or less, for the people to know.
Listen, we don't need to cover everything. Let me finish this part and then we can have a session of questions and answers and this kind of stuff. So the important thing here is that we developed the pipeline that we can fill in gaps. And by doing that, we are closing a circle of drug design without doing the drug design. Because the algorithm now fills in the space with those little cubes. And those little cubes, they didn't come using the rational drug design approaches. So now we could deduce, reverse engineer those little cubes and screen the compounds that would have satisfied those little cubes and come up with the actual compound that would have created those representations in the protein without actually doing the drug design pipeline. The drug design pipeline takes 1 billion, takes 10 years. It's less than 1 in a million. So we thought that even though that path that I showed you has it's problematic in many ways. But I would dare someone to tell me if I stand more than less than 1 in a million chances to get something out of it, because the other way has proven itself and it's less than 1 in a million. So we try to think differently. And what we did is we came up with this compound, which this compound describes those cubes. And I have to tell you that we used 475 dimensions. Maybe in 10 years, 5 years, 15 years, there will be 2,000 dimensions, and those cubes will be different, maybe better. Okay? So we went back to the lab, because this is how you understand that you have achieved something. We took the compound, we went into the cell lines that overexpress STING. We checked that it works, it inhibits the protein. Then we went into in vivo, in zebrafish and mice, and first paper in Science Advances came out from those cubes 3 years ago in the lab, where we identified that AP4A is a modulator of STING. And then, having the first lead, which took about 1.5 years compared to more than 10 years doing the rational approach to drug design. And it took only a fraction of the money and the brains that it required. We went to networks and we said, look, now that we got a compound, can we look for analogs to that compound from a medicinal chemistry point of view? And by doing that, we identified a series of compounds that mimic, they would compete the original compound, and they were the polyunsaturated fatty acids, so PUFAs, actually modulate STING, and a paper in Cell Metabolism and another paper in Nature came about, and we managed to find how STING crosstalks and behaves in the cellular environment, the cellular context. And finally, now we had first lead, we had many other compounds, but now because we have many compounds but one protein, we went the other way around and we thought, now that you have all those compounds, can you find another protein that is similar to STING, using the triangles that I showed you before? And we identified a DNA protein kinase that has the exact same conformation and amino acids in the active site. And we believe that this protein kinase, any kinase that phosphorylates or does what a kinase was supposed to do, that kinase hydrolyzes the circular DNTPs that are substrates to STING, which kickstart the immune response. And we went to the lab and we confirmed that the 3', 2', 3' SIGAM does interact with DNA, PK, and we published that in bioRxiv and is now under consideration from Nature Immunology, and we received the first set of reviews which we're trying to address. So, see, but it was not that bad. It was 60 slides. So, what I'm trying to say here is that we could be brave enough to think differently. I am more than happy to come to the lab to talk to every one of you about what you do. If you have a data set that could be applied to this approach, if you have a specific protein and you want to find more ligands, or you have many ligands and you want to find some proteins that they could interact with, maybe you see some side effects and you don't know why your compound is doing side effects. Maybe it's doing it because there is another protein with the same catalytic site, active site somewhere else, and you don't know that it exists. Using that approach, maybe we can identify it. Many different, a lot of different people have collaborated to get here. This is the funding that we got. The highlights is that we did the in vivo work with our collaborators in CNRS in Montpellier, France. And with those people, we got an ERC, and then a couple of horizons and national funding, the Excellence and EDCTP. So there is a continuous funding that is needed to keep the people motivated and the reagents on the lab, on the bench, as it should be in any of those biological experiments. This is your president, my boss, and thank you very much. It's only 10 to 8, and I started at 7:15.
So let's see, thank you for the amazing talk, and let's see if we have questions. Who would like to ask something?
Next time I bring drinks and food and relax.
Thank you very much for your amazing talk. I think you don't need me to say that it's amazing, your work. And I would like to ask, why is it more efficient to represent the protein with cubes instead of the traditional points, which are the amino acids? And
the answer is very simple. I'm afraid it's not scientific, but the answer is very simple. A lot of smarter people than me in the past have done what you said. They have represented it with points and bonds, and this is where we came. We wanted to think, to not be biased by this way of thinking. I'm not saying that it is wrong. What I'm saying is we thought that maybe it's not the protein that is important, but it's the context of the protein, it's the space, the cellular environment. And what we did is we segmented space. So if we did that in this room, we can make a big cube here, nobody's there, I am here. If I go there, you cannot hear me properly. I'm still the same person, but now it's a different experience. But if I go back to my cube, it's me, but now I can communicate with you in a different way. And this is exactly what we wanted to achieve. it's not me that changes, it's where I am that is making a different experience to the self. And we wanted to do that in the most unbiased way that we could, and that is by defining space. And of course you can imagine that when you put those cubes and you start to move the protein in a molecular dynamic simulation, you can have an amino acid that belongs to 2 different cubes or 3 cubes, and then it moves to another cube, and that trajectory from where it goes to where it goes changes the physicochemical, the color of the cube. And that transition could be a biomarker, we hope, in the future. Not the molecule, what effect it has in the environment.
Thank you very much.
Thank you for your presentation. I'd like to ask if, as I understood, the use of the cube representation, you targeted more on the drug design or ligand design. Could that give information about structural motives and how we have amino acid motives of distinct binding sites or something like that? Could we draw information about structural motives or structural property motives to predict structures in the future?
Yes. Of course, from structural biology, crystallography, we know there are these barrels and helix, loop helix, and all these things that we learn. Think of that. You have a protein, the microphone is a protein, and you have something that binds here, so you have a protein-protein interaction, and something else that happens in a ligand or DNA binding domain, like a transcription factor or something like that. Do you think that the signal is wired through the backbone of the protein? So something binds here. How that structural change, conformational change in the other end of the protein will occur? Do you think that there is like, it's a wire, and it's like electricity or a telephone wire, and the signal will go through the backbone? We don't believe that. We think that something binds here, and it will go not via the backbone, but in another way, depending on the structure of the protein. Okay. So there are many different algorithms to do that. One of those is the traveling salesman algorithm, if you've heard, or the ant colony optimization algorithm. Ants, when they find, they just walk around in the field, but when they find food, they release some pheromones because they carry the food, and they leave them on the ground. And when another ant wants to go to eat something, it smells the pheromones, and even though they walk in random walks, when they find food, they go directly to where they need to leave to their nest. Okay? So we are now thinking of applying that algorithm to the cubes in order to come up with new structural motifs that are not defined by the backbone. They are defined across the protein. Yes. It's like tomography, like taking a slice of the protein.
Thank you.
You have another question?
I have plenty of time.
Okay. I just wanted to ask from a more moral and ethical point of view.
I replied your email, yes?
Yes. And yeah, I just wanted to say that since we have the definition of artificial intelligence, I just wanted to ask if it's responsible and if we should have AI to be accountable, so to speak, for the more serious medical and scientific purposes. If I could use geopolitics as an example, if 2 countries go to war and we have artificial intelligence to decide how to resolve the conflict, and it just says, let's nuke the places. So, you know, and quote-unquote logical solution, but not a very human one, not a very rational one in the long term, so to speak.
It's true, it's true. Please, yes.
Yeah, that's the question, I think.
It's true. There is a fantastic story, Κόμις Μοντεχρίστος, Αλέξανδρος Δουμάς, αν το έχετε διαβάσει, που έχουμε τον Νάντες, νομίζω λεγόταν, guy who is falling in love with a girl, Mercedes, and there is something like what you said. So there's a conspiracy. So someone wants to take away his girlfriend, so he puts them in jail. And then he goes to jail, and he meets a person, Faria is that person. He is sentenced to death. He's going to die in prison. So he decides that I will teach you everything that I know. So when I die in prison and you are released, my knowledge will go to the outside world. And he teaches that person everything. But back then he knew 150 books, they say. That's what is in the book. And at the end of the day, AI is a tool. It cannot teach you philosophy. It cannot teach you lust. It cannot teach you love. It cannot guide you. It cannot make you feel wonderful that you are in the lab doing your research. It doesn't have feelings. So yes, in a geopolitical thing, of course, the easy answer is the thermodynamically lowest entropy solution. That bomb costs €1,000, drop it, problem solved. Okay? My answer to your question is this. AI is a tool. It's the same as a scalpel that the surgeon is holding. If you give that tool to me to do the surgery, it will not go down very well. Okay? It will be a massacre. But on the hands of the right person, it can save lives. AI is exactly the same thing. It cannot be accountable. As the scalpel, the knife, cannot be accountable for killing the patient. It's the person who is using the knife that didn't perform the surgery properly and the patient died. The same with AI. If you use it respectfully, if you know your stuff, if you've done your homework, if you are not too stressed, what will happen to your final year dissertation, Andrea? Because you worked too hard. That was his problem today. He sent me an email. Take your time, relax, do your homework, study, learn. The important tools, the actual real weapons that we have is knowledge. It's not AI, it's not a Gilson pipette, it's not a good centrifuge, it's knowledge. Do your reading, do your homework, and do not stress about your ptychiaki, because it will go fine.
Okay, so actually two questions, if I,
Of course.
So let's go back to your initial slides where you showed us that there was an increase in the data amount. So do you believe that there will be a time, either sooner or later, that will reach to a plateau where there's no more data generated?
That's one question. The second?
The second one is why dark DNA?
Ah, yes. Okay. So here is my answer to your first question. I don't think that the technology that we own right now and will be, I hope, developed in the future will produce less data. I think that it will continue. But there is a huge problem in cloud computing, not with processing, but with storage. The bottleneck right now in cloud computing is not very fast processors, GPUs and CPUs that can perform the calculation. Is where do you store all? There are not enough hard drives. And there are big companies, and I'm not promoting those companies, I'm just going to say a name that I find wonderful. Please read about them. It's called Petagene. Petabyte gene, Petagene. And they have made billions in revenues. It's a startup company by 2 PhD students in Sweden. Billions, not millions, billions. Because they came up with like an algorithm, like how you zip a file and you reduce the size. Now they zipped sequences to 10%, 20% of the of the space they take on the hard drive, but still be accessible to the algorithm, compressed. How we generated the maps? So they generated a zip file, and then they aligned the zip files, and they came up with an alignment tool that aligns zipped sequences. So I think that the bottleneck is going to be storage.
Or the magic word, quantum computing.
Maybe. But again, I'm not sure how much storage there will be on a quantum computer. You know, you can have 6 different states or many different states. It's not a binary system, a quantum computer, of course. But I think that has to do with the processing, not the storage. I'm not sure about the storage. That's why I'm saying this. Dark DNA, I was very drunk, and I had to come up with a catchy name for the lab. So dark matter, we know it's there. We have the mathematics to prove it. We cannot touch it. We cannot see it. Dark energy, we cannot see it, but we know it shaped the world. There are parts of the DNA that are dark, that haven't been discovered yet, and we hope that someday, we will add a little bit to the knowledge that is out there about the dark part, the dark bits of our genomes and of life. That is why we call it dark DNA.
Good, thank you very much.
Also, the URL was available for $8.
So maybe one last question.
Again, very interesting talk. I want to go a little bit more, I guess, philosophical, perhaps, about AI. By the way you talked about it, because you kind of said ML is one thing and AI is a different thing, I assume your stance is more like that there's some emergent properties in AI or capabilities that you can have with, it's not just statistics, perhaps? Would you align with that view, maybe?
Yes.
I would like to antagonize you a little bit on that.
Yes.
Have you heard of the Chinese room argument?
No.
So, for the context, the Chinese room argument is basically a thought experiment. It says the following. It says, let's say there's a human in a room completely isolated, and the only thing he receives, his input, is, let's say, memos in Chinese, in Mandarin, whatever. He does not know Chinese, but he has access to a book which contains, let's say, a billion phrases, and for each input, he can look it up, and he can match it with an appropriate output.
Okay.
And he can send it back to the whoever. And you can see the analogy here. It's basically, he's inside the black box, right?
Yeah.
So, but that does not mean that he actually understands what he's doing. He's just really good at following the instructions or the he's learned the, let's say, motivo, pattern.
Yes.
And so I guess this is kind of my point. How could we prove that there is something more to a response than just really, really complex statistics?
Yeah, of course. That's a very valid. Thank you. I will look it up. I didn't know about this Chinese room thing. My thought about it is that, of course, this applies to many, many different scenarios in biology and medicine. I think that by applying the Mandarin memos and you having a dictionary where you can translate it while you are inside the box, if you have a very well annotated scenario, so if the memos are biased, in one case, in a second case, in a third case, so if you apply something like a pipeline, like the one I described, okay, but you know the outcome because you have done the experiment in the lab. It's not just theoretical, it's not just on a computer. And you recreate and you play again and again and again all the permutations of all the different Mandarin memos that you may receive. At some point, you may bring yourself to be able to predict a couple of the memos that will come to your hands in that little Chinese room. And maybe when you reach that point, you can apply that route to a different scenario. So I think that nothing on a computer works in the real world. It's sci-fi. You should never trust things that have not been proven experimentally. By applying those scenarios on real experimental data, then you can start trusting those scenarios for the data that you subjected them to. And then, if you do that many, many times, then you can just go one pixel further, one step further. Of course, you're right. You cannot, we would have won all the Nobel Prizes for the next 30 years if we could have come with an algorithm of AI that can be applied to cell biology. Little, small, manageable scenarios that can take science one step further. Είναι μεγάλη μπουκιαφάη, μεγάλη κουβέντα μικρής. Αυτή είναι η απάντηση.
So let's thank Dr. Lahaki.
Who else? Someone from there. You.
Last
And
question.
you.
Okay, last.
No, you.
You?
Ask something. Okay. Όχι, πλάκα κάνω.
Do you want to ask?
It was very similar to
Okay.
but
yeah. That's the first time I do that.
Yeah, but it was a very similar
Be prepared.
question.
I will ask random people to ask questions.
Yeah, but I was just wondering, as a biologist and a very enthusiastic PhD student, it's very easy to get in the trap, like, I want to use this tool to analyze my data, and then I'm not actually like trained as a data scientist, and it's so tempting to use ChatGPT and let it code for me, and we never know what's actually going on. And I was also going to ask about like a data validation and what's like caution points as a biologist if we want to use AI models for our data sets. Was my
which is pretty much the similar thing. You have to find a wealthy supervisor. So he can test your stuff. That's otherwise you're lost. Yes.
Okay, final. So it's the same. Okay. So let's thank Dr. Lahakis and guys, thank you very much for coming until late here.
Thank you very much for your time and patience. Vasily, thank you for the invitation because he called me. And I hope that we can collaborate, exchange. The code is open source. I can send it to anyone. Send me an email. Go to darkdna.gr and my credentials are there. I will send you everything that you like.
I hope you like the presentation and see you tomorrow at 9:30. See you guys. 9:30. 9:30.
I will be here tomorrow 9:30 to continue my talk.
We have a party tomorrow night. Δεν ξέρετε ότι δεν τον έχετε καλέσει για το πάρτι. Εγώ συγκινήθηκα, είναι ευτυχώς να πάω από το κόκκι ζητημένο. Είναι κάτι συγκίνηση στο τεσσερικό μου ήταν ερωτήσεις ποιότητας και ζωής. Εγώ είπατε το τέλος, είπατε και το concept. Only that ground. Με συγκινήσατε. Έχετε διαβάσει και οικονομικά έκθεση. Σας στέλνουμε πάλι.
Εξαιρετική και πολύ καλή πρόταση του κύριου Ευθυνίου, πώς μπορούμε να οργανώσουμε κάτι.
Ο οποίος προσπάθησε να σώσει το χάπι